Can you guys hear me? Hello, can you hear me?
Good morning, everyone. I request everyone assembled here and joining us online to kindly mute their cellular devices. All participants who have been allocated to display their posters today are kindly requested to do this at earliest, if not done yet. The internet access details are displayed all across the auditorium for easy login. We'll start the program momentarily. On the behalf of Symposium Organizing Committee of Translational Health Science and Technology Institute, I, Sneh Vajpayee,
And I, Bharat Lohia,
extend our warm welcome to all of you to the final 3-day symposium towards NTB, Achievements, Challenges, and Future Directions. Today, we have 2 pre-lunch sessions, sessions 7 and 8, on TB drug discovery and development, and 1 post-lunch session, session 9, on host pathogen interaction. TB treatment has come a long way from just good food, rest, warmth, and lots of sunlight, just as our age-old TB sanatoriums, to royal touch from kings and queen for quick healing, to cod liver oil, vinegar, and turpentine, to the current 4-drug cocktail for drug-susceptible TB. The 4-drug cocktail, though, has been successful in reducing the burden and enhancing the cure, has also caused several toxic side effects. With the emergence of MDR and XDR TB and the extended duration of treatment up to 2 years, came more side effects. It is in light of this that the next 2 sessions become very important. To give us a better perspective of where TB drug discovery stands, I warmly invite Dr. Bal Ganesh to deliver the keynote lecture titled 30 Years of TB Drug Discovery and What We Have Learned. Dr. Bal Ganesh is a highly accomplished scientist with research training from Brookhaven National Labs, USA, and the Max Planck Institute of Molecular Genetics, Germany. He has held numerous leadership positions in pharmaceutical industries and research institutions, including serving as the president of Gangajan Biotechnologies, where he oversees research and development activities in the identification, design, and progression of bacteriophages and phage-derived products for treatment of serious bacterial infections. He also led the open source drug discovery program of CSIR. He led the Bangalore unit of AstraZeneca R&D over a decade, where he contributed significantly to the discovery of neglected diseases and TB, respectively. Dr. Bal Ganesh has received several honors for his work, including honorary doctorate from University of Uppsala and Ivan Ostlund Medal for drug discovery. Following the keynote address, there'll be a 5-minute question and answer session. In the interest of time, I request the audience to keep your questions brief so that there is opportunities for others. I urge him to enlighten us with his valuable insights into TB drug discovery.
Can I?
Yes, sir.
Okay. You can see my slides. Hello. Can you guys see my slides?
Yes, sir.
Okay, thank you very much. Thank you for the nice introduction. I'm sorry I'm not there in the audience with you. Obviously, some health issues. I'm also very grateful to Krishna Mohan for more or less helping me out through the last 5-6 days of how to deal with my problem. I also sincerely hope that my voice doesn't trail off. So what should a first lecture on the last day be? And I've been thinking first it should be short. Second, I think it needs to be a little provocative, and I think that's what I'm trying to do. I'm going to present my perspective of 3 decades of drug discovery, TB drug discovery. It's after all a small window that I look at it, and there are so many aspects where you can always have a different opinion. But here's my chance to give you my opinion, and we'll be happy to hear what you think. So the title as it goes, 3 decades of drug discovery, what have we learned, was actually the title. And then when I put together the slides, I said maybe it should have been better titled, what have we not learned? So let's go over that. And in this next 15 or 20 minutes, we'll deal with both what we've learned and what we've not learned. Oh, my slides are not moving. Okay. What I'm going to talk to you today about is my favorite topic, evolution of the current therapy. I love this story. So you guys cannot help but listening to it. Hopefully it'll be short. And then I'm going to say something about the big unknowns and yet we dare. And then a few words on my own views of where are we going and the challenges. So talking about the evolution of the current therapy, we can talk an hour on this, but 3 important landmarks that we who sit in this audience, as well as all of us who are partaking in raising the knowledge bar of TB, the disease, as well as the treatment, I consider 3 of these landmarks as very important for us to remember as we go forward. The first one is a trial in 1947, and I point this out specifically because this was the first ever clinical trial on a drug. And this was actually, questioning the famous concept that, can we have bed rest or can we actually treat with a drug? At that time, streptomycin was available. And if you will look at this very small trial by today's standards, you will see that in the first 3 months, the streptomycin group clearly had 10 culture negative, while in the control, there was only 1. After 6 months, though, you had the streptomycin group had 8 culture negative and 2 in the control group. But the more important thing is, if you look at streptomycin resistance in a 5-year follow-up, everybody was streptomycin resistant, 35 out of 41, and the mortality between those who had only bed rest and those who had streptomycin was more or less same. But this is the first time that a drug was used for treatment of TB, and more importantly, the first ever known clinical trial for a drug. The second is another hallmark, and many of you must have read that famous book on the sanatorium. This is asking whether the concept of treating people at home, or do we really need a sanatorium? And this was a 12-month treatment where people were treated at home with, at that point, what was available apart from streptomycin was para-aminosalicylic acid and isoniazid. And we had 49 patients treated at home and 50 treated in the sanatorium. And if you will see that the relapse rates was equal among both of these, one was 7, one was 10. And in a 2-year follow-up, again, in terms of the contacts, they're very similar. Again, this was the first scientific proof of domiciliary treatment you can have with drugs, and you need not go and be confined to a sanatorium. The third slide, again, is another major landmark. It happened in 1976. By this time, we had isoniazid, rifampicin, pyrazinamide available. And this slide data is all from Mitchison's monograph. This is a culmination of a number of trials. I'll refer to one earlier before this towards the end of the talk. But what you will see is a patient group of giving 3 drugs for 2 months, followed by 10 months of isoniazid. It was a 12-month treatment. You had a relapse rate of only 3 after 2 years. Now, remember, relapse rate is measured as people who come back with TB symptoms, and the TB that is isolated is sensitive. Now, if you look at the last 2 rows, you can see that we had a sputum negative rate at about 80-85% and a relapse rate of 8 to 3%. So clearly saying a 6 months treatment with what is current therapy, we have replaced streptomycin with ethambutol, was very good in preventing relapse, and it was also very good in cure. What is also important here that, we started in 1950 sometime with the first drug. In 20 years, we went from despair to efficient treatment. And though you would today say that when I say translation science as it speak, that they actually went from a lab to some experiments in animals and directly into humans, and they could switch different groups. It was still very epoch-making in the sense that you could make some hypothesis and carry it out. The other important fact which came out in all these trials was, one is you needed combination to combat drug resistance. But there was clear indication that different drugs played different roles, because what's not here, which was in the next trial, was pyrazinamide, where they showed pyrazinamide had no effect if you put it in the continuation phase, and its maximum effect was only when you put it in the initial phase. Again, there was something more than just preventing drug resistance happening. And this is 76. And today we believe that there are different populations that these drugs target. All right, so that's my favorite storytelling. Let's then go to, if this was a slide which I modified a bit, but we made in around 2000, and tried to look at 3 different parameters. Just looked at the microbe along the different decades in the pre-80s, 90s, 2000, and we said 2010 and beyond, which probably should read 2020 and beyond. If you look at the microbe in the pre-1980s, they were all drug sensitive. In the 90s, we started seeing some amount of multiple drug resistance. By 2000, we not only saw a multiple drug resistance, we started seeing extremely drug resistant, as well as we went forward, we saw lots more cases of MDR and XDR. Let's look at the patient population. Has it actually changed? We all know, though I would hate to admit this, but this is true, that TB is still largely a disease of the lower socioeconomic strata, and that's because clearly poor nutrition and lack of pure air contributes to the immune response, so they are more susceptible. but most of the patients were immunocompetent. In the 90s, we started seeing HIV, we saw the same socioeconomic strata, and we started seeing increase in relapse rates. And this was, we started seeing lifestyle changes, and we also had an impact of HIV. In 2000, which is still quite prevalent today, we are seeing TB across different economic strata. There is a definite increase in the number of non-pulmonary TB, and the caveat in that statement is that clearly the awareness diagnosis is better, but there is an increase. We clearly saw a lot of reactivation due to HIV and treatment failures, and then we had this whole new therapeutic area of immunomodulators. And if you'll remember some of the famous ones like the IFN and gamma inhibitors, I forget the name. It is a thing about etanercept. It's a one like that, which inhibited TNF alpha, and then you started getting increased relapse rates, and so there is a warning label. And then if you look beyond, as we see today, we've got the diabetic epidemic, and it's more or less well established that predisposes to TB. We have a large aging population. We are seeing more and more of organ transplantation. We're going to see more of device implants and so on, which all require immunosuppression. So I remember when we started work on TB sometime at AstraZeneca, sometime in the 90s, you asked the question, what is the sort of target product profile or the minimum attributes of a drug? And when you spoke to the TB program authorities, they would come up with this long list: sterilizing, it should reduce duration of treatment, it should be combination compatible, it should be DOTS compatible, it should be cost effective. Then the 90s, it was more or less the similar picture, but it should be active on multiple drug resistance. It should be obviously compatible with the HIV treatment. And this was the time when rifampicin and some of the protease inhibitors were for anti-HIV were actually incompatible. Then around 2000, I remember this whole mood shifted and people said, give us any drug which works because we are desperate. And if you'll remember, 1996 or 97 is when WHO declared TB as a global epidemic. We all definitely wanted it compatible with HIV. So give us any drug, but let it have minimum side effects. And we continue with the same thing today, where we said we wanted MDR and XDR drugs. And in my view today, we have drugs. But what we are being challenged with is, can we reduce the duration of treatment? So with this background, if you then ask, what should be the target product profile for somebody who is getting into drug discovery? Because you are saying, excuse me, just drink a sip of water. Sorry. What is the target product profile that one would like to have? As you know, this is where how our compound needs to look at. And what we have done here is on the y-axis, we have value, where we're saying value to the patient, value to the doctor, value to the Stop TB program. And on the x-axis, we have risk in the sense that how easy is it to get this type of a drug. And if you will see on the left column, we've got compounds where we say something which is effective on drug resistance, but no cross resistance. And then we said something, if you look in the middle, effective on drug resistance, but parenteral use, obviously the value is limited. Then we said effective TPP3, which is effective on drug resistance, narrow spectrum. And of course, what we are differentiating 1 and 3 is asking the question, can broad spectrums be used versus narrow spectrum? And then if you looked at 4 and 5, they were basically effective on drug resistant, narrow spectrum versus broad spectrum, but we're looking at rapid sputum clearance. And we knew at this point the value is high in this group of TPPs, as we call them. And if you look at TPP 6 and 7, clearly the value is very high and the challenge is also very high because you're talking about reducing duration of therapy. So this is the sort of the broad picture, what we thought different compounds can actually play a role. And I wish I had more time on this, but we won't at this point. So when we were doing this, our biggest challenge, as we said, what is going to make the final difference that we can differentiate something which shortens duration, which is effective on drug resistance, rapid sputum clearance, so on and so forth. At that point, the only tool we have, and the only tool we have today, is what you call as early bactericidal activity. So we tried to correlate whatever data was available at that time, saying, can we look at the efficacy in mice in terms of dose and reduction of colony-forming units in the lung, vis-a-vis how does it behave in the the early bactericidal activity where you give multiple doses and you're measuring sputum across first 2 days, then up to 14 days. India allows only 7 days. I don't know if that has changed, but it's a monotherapy. So here is 0 to 2 on the x-axis in terms of sputum reduction, and that's the ratio which is used. And on the y-axis, you have effective in terms of the mouse. What you will stand out, you will see that isoniazid is clearly very good in the early bactericidal activity, and so is MOX. This is, remember, the first 2 days. Cipro is a complete, does not work in the mouse model at all, but does give you some EBA. Strikingly, pyrazinamide has no EBA and has moderate or low activity in the mouse. I have TMC 207, which is basically bedaquiline at that time. P824 is protonamide, which is the GATB compound. And both of them in the first 2 days don't seem to be very active, and you see rifampicin around in the middle. If you then move to the next, so we asked, we really don't see any correlation. Then you look to the 2 to 7 day, same type of correlation. Pyrazinamide remains where it is. Rifampicin clearly stands on top. And then you have MOX, isoniazid, and Cipro, but the other drugs, Ofla, protonamide, bedaquiline, pyrazinamide, they're all iffy in the sense, in terms of they're very good in the mice, but of limited activity in the EBA. So our biggest problem at that point is how do you actually, if my progression is from mouse to EBA, how do I choose a compound? And this will haunt us as we go forward in the next 10 minutes. This is a old slide, but I call it newer two drugs, and I have a specific reason why I'm putting it out. The lower ones, one is Cetsolid, AZD5 is another. These two are Oxas, which are no longer going forward. Sequela is still there in terms of on the list, but I don't have any new information. But the rest of the compounds, most of you are very aware of the Delametab, bedaquiline. There's a spelling problem here, sorry. Ofloxacin, Moxie, and P824. The reason I put OFL there is that the Moxie trials, which we've all heard of, started post-2000. But the first trial that we should try and include a quinolone in the treatment of TB was done at the Tuberculosis Research Center, Chennai, under the leadership of P.R. Narayan, where they used ofloxacin. And that trial actually showed that you could reduce the duration of treatment to 4 months. But there are a lot of holes which were found in the trial, but that's not important. What's important is that thought process that let's look at the quinolones as a way of bringing it into the treatment. The credit should go to TRC. Then you have this I picked up from the latest WHO review 2022. You have all these compounds are in phase 2. And I really don't see any pattern, a specific pattern in them, except to say that most of them have either come through screening of compound libraries or using what you call as analog screening, where you've had related compounds in other diseases or like in the antibacterial field, and then bring it into TB. So we've got a reasonable number. We've got about 8 of these in phase 2, or maybe 7. And then you say, even if 1 or 2 come in, so you totally have 5 or 6 drugs in your plate. And with that, then I said, let's look at your TPP matrix. This is the same slide, so I can move over. But what was striking is this, with this pipeline, I think TPP 1, 3, and 4, even though you'll have to shift the value, have and will start making the impact. They will make a difference. They are making a difference. So I'm not trying to say that we've got a full pipeline. It's like half glass full or half glass empty. We still need to find new compounds to manage resistance. But at least we have compounds by which we are able to deal with drug-resistant TB in an effective manner, even though we still talk about whatever months of treatment. I think the challenge is in TPP 6 and 7, where how do we reduce the duration of treatment? And this is where I go back to my first or second slide, where I said, what have we learned? And then we look at what have we learned and then say, what have we not learned? So I go to the next slide where I simply say, okay, let's step back and look at our drug discovery path for TB. We screen either target-based, compound-based, analogging, and we choose some compounds and bring it into a number of in vitro assays today. Previously, we used to do MIC, MBC, and kill kinetics. But today we're doing a lot more assays with different models, like replicating TB, non-replicating TB, TB growing in starving conditions, TB growing in reduced oxygen conditions. So we've got a whole list of those models. And then we go into ex vivo assays, like where we are looking at how good is our compound in dealing with MTB sitting inside the macrophage. And in fact, one of the compounds in that list actually came from screening a library in an ex vivo assay. Once we believe it has passed this and you think it has a reasonable pharmacokinetics, we go into in vivo assays, which is the acute model of 3-week treatment, or the chronic model of where we go into looking at relapse, and we do it in mice as well as in guinea pigs. Then we go into early bactericidal activity 0 to 7 or 0 to 14 days, and then we go to a phase 2 coverage. If you look at all this pathway and then you place your drugs today, and here you have Moxi, bedaquiline, Elemenib, P824, so bedaquiline has got repeated. But the first striking thing, if you see, I'm looking at replicating intracellular activity on non-replicating bug, mice-based infection models, or early bactericidal activity. I cannot differentiate them. I might say one is better than the other and so on, but really, at this stage, they're all very good. 2 compounds like citozolid had poor intracellular activity, and the AstraZeneca compound actually failed in EVA. That's a completely different story why it failed, but we won't get into that. So strikingly, I'm not able to make out a difference. Though I can say in one case, the MIC in a particular model is 0.1 microgram in one case and maybe 1 microgram in the other. But otherwise, I can't make out a difference. And then if I look at the current compounds, which are in the phase 2, I have the same problem. Some of this data is not available in the public domain, but they have completed EBA and the companies announced the results are looking positive. So I'm taking that they are positive. But again, the whole gambit of assays they may differentiate a bit, but they really don't, there is no striking difference. So we are not able to differentiate. So my problem here is then we are talking about the only way I can today get into is to do a phase 2 trial to look at shortening of therapy. All my earlier assays don't give me a clue of which compound or which compounds I should use to try and achieve that. I've got back, sorry. Now, what I've added in this slide is one compound which has always been an enigma. A lot of work has been done about it, but we've got nowhere. Is the last row, which is pyrazinamide, which is active only in a special medium. There's some sort of a black magic. It has no intercellular activity. As far as I know, there's no report on non-replicating activity. It is active, moderately to poor active, and it has in the mice model, and it has no EBA. What's so special about pyrazinamide? And we've always looked at pyrazinamide from a compound perspective, microbe perspective. What's its target? How does it get modified? Is it a prodrug? What differences does it make? But I think we need to look beyond in the sense that are there host specific challenges. So what I'm doing is mingling in terms of what I think is the problem as well as where I think we need to look at. And the next few slides is along the same lines. Treatment failure. I will stop in 5 minutes. Now, treatment failure, we've got data starting from the 1950s to as late as 2000s. So I just want to point out 2 facts. If you look at the standard relapse rate, it's about 10% to 16%, where you can call it as treatment failures. Now, and then if you look at another more recent study, which was done on Gatiflox, where it showed it was not good enough to the standard trial, we had the same 10 to 15% treatment failure. I don't want to use the word relapse, treatment failure. And so what is different in the 10 to 15% failing treatment? 80 to 85, 90% are cured, and this thought process is reflected in the new paper on a new trial. I hope somebody will ask me about it, we can talk about it. Is this 10% purely stochastic, like, the lac promoter operon in a population, some will always be expressing? Is it due to the host response? Is there a microbe heterogeneity, or all of the above, like a host microbe interaction? I think there has been less and less studies on this population which comes through versus a population where there is a treatment failure. And I think there's a great opportunity here. I think there's lots and lots of information that has been developed in terms of how host responses, looking at various models as well as macrophages, different macrophages, how they behave. Sorry, excuse me. How they behave and the reflection on, a whole lot of data. We need to get this data to converge. Where should I think we should be going? We should look at patient cohort studies. Clearly, this type of that percent of patients who are relapsing, patients who are treatment failure. We have a lot of sorry again. We have a lot of information on the tubercle compartment, especially the drug penetration. How does it look? this is my personal opinion, and I've said this for many years, that, I know we have to do mass treatment. But right at the beginning, we apply the rule that, it has to treat all sorts of patients, whether it's extrapulmonary treatment, 3+ treatment, high deep cavity patient with comorbidities, everybody is put into one bracket. The last point which I wanted to make was adjunct therapy, and I think this is very serious. Efforts should be made. Was a recent trial which has been published on metformin, which was we were involved in it from the open source. This was led by Padma from TRC and Soumya Swaminathan at TRC, where metformin clearly, I mean, it did not bring down the CFU. This was done on normal patients, not the diabetic. It was done for 8 weeks, 502. Clearly did not bring out reduction in CFU, did not have an effect, but it had a profound effect on the lung pathology. And there are other compounds which are very close to clinical trials or in trials from an adjunct therapy perspective. So, ladies and gentlemen, I will stop here and will be happy to answer any questions. And I hope I may not have stuck to the time, but I hope we have not gone over too much. I'm sorry again for my interruptions. Thank you.
The floor is open for any questions. Biji, can you hear me?
I can, loud and clear. I'm just getting back to, yeah.
Hello, sir. Sir, I wanted to ask, how do you differentiate between treatment failure and relapse?
Okay. This is a standard definition, which is used for classifying patients. So treatment failure is at the end of 6 months if you still find the sputum positive. Relapse is usually a follow-up of the patient post stopping of treatment for 4 to 6 months. And then you see whether the patient comes back with symptoms. And then you look at the microbe and you still find it sensitive. So that's the definition of relapse. And treatment failure is post 6 months. And in this case, obviously, it could be treatment failure could be the drug resistant failure, or could be a drug sensitive failure.
So first one is
Does that answer your question?
Yes.
Hi, so my question is on host-directed therapy. And I mean, where do you see it? Host-directed therapy is going to, we should focus more on shortening therapy time, or would it be like an immunomodulator? So look at host function and lung function post-therapy. Where do you see host-directed therapy is going to fit?
We can have a seminar on that, but let's first start with saying what if you look at the preclinical data that we have and what we're trying to look at, we are actually more or less looking at colony forming units reduction. And so we're asking the question that how does host therapy modulate, influence, ability to reduce colony forming units, either increased or rapid, whichever way you want to do it. But I think, I don't know, I hope many of you have read the newspaper last week by Prasad. There was an article on TB in the Tamil Nadu health system. And it was a really amazing article for me, because there was a 50% reduction in mortality rates in the primary health centers. And it was driven purely by three criteria that they said. They segregated patients on patients who were ambulatory, patients who had a reasonable lung capacity, and patients whose cavity was not very extensive. These three patients were not put in the hospital, while only these three category patients were not put, the rest were put in the hospital. So what I'm trying to say is the patient's well-being makes a huge difference in the outcome of the disease. And if you look at the new NEJM clinical trial paper, I hope somebody will ask me about it, you can clearly see that the choice of the patients made a big difference. And though it involves a lot of infrastructure if you're going to choice. So what I'm trying to tell you, or what I think I'm trying to tell you, is that host-directed therapy can make a difference across. And the window in which we are measuring makes the difference.
All right, thank you.
Yeah, thanks for the nice talk. So today we have totally different treatment regimens for drug sensitive and drug resistant TB. And there are discussions on new drugs. Should we have something that encompasses all of this? Your thoughts on that?
Okay, so if you look at TB treatment, you've got, I think today, 60% is private and 40% is through the government channels. My numbers may be slightly here or there off. Now, from the government channel perspective, the more you simplify the treatment, the more efficacious they are in terms of ability to administer it. Simply because you need so much more resources if you try to personalize treatment. On the other hand, even today, if you look at the private treatment, you will see it's more or less personalized to the patient's characteristics. So I think there is room for both. And going forward, we will see that. I am not a strong believer of one size fits all.
Thank you. Can I ask? So, hi, Biji. I'm Nishit from THSTI.
Hi.
Talk. Thanks for accepting our invitation. So, I have one question that, as you have been showing, and we have seen that no matter what we do, what drugs we develop, but it takes bacteria not a fraction of time to develop resistance. So, what's your take on that? Like earlier, we used to think that it's non-compliance or adherence, but now we are seeing that those are playing not a major role in the drug resistance. So what's your take on that? How to tackle that?
Vishit, I think there's good evidence to tell us at least glimpses of what we think is happening. Yes, you're right. We used to think of compliance as an issue, and still compliance is an issue. There's no question about it. But if you look at the individual drug resistant frequency, you will reach a number which is phenomenal. So you never will have that higher population of bacteria to say that one among so many 10 to the minus whatever will give you a mutation or resistance across all the 4 drugs. But we're still getting resistance on it. I think there are 3 really, really good papers where one was by Cliff Barry's group, one was by Camila Rodriguez's group from Mumbai, and one recently, Veronica had published a paper. In all these 3, when people took bacteria out of individual tubercles, and this is cases where they had done a lung lobectomy and then you look at bacteria in the individual tubercles, you find they have very different drug sensitive profiles. This was then followed by Veronica's paper, where she actually studied drug concentration in the different tubercles. This is with the frontline drugs, and showed clearly there was a difference. But if you looked at 4 different patients, you had 4 different patterns. So I think the problem we're seeing is that I don't know how you're going to treat with that. The different tubercles, you are having different pharmacokinetics, and so you're going to face that issue. Now, apart from that, I think it's also the question of number of bacilli, like we know in the HIV case. In the miliary TB case, there's a huge number, so we do develop, reach levels of frequency where you can get drug resistance.
So do you think that what Amit has just pointed out, that if we combine the host-directed therapy along with antimicrobials, that may solve to some extent the problem of the drug resistance also?
My only answer to that, Vishwajit, is it's worth studying.
Yeah, I know. Thank you.
Absolutely.
Abhijit, this is Balaji.
Hey, Balaji.
One quick question we discussed earlier, the location matters. So in case of host-directed therapy, if you take ocular TB versus a bone TB, and you bring in, for example, if you bring in a checkpoint inhibitors there, for argument's sake, the location makes a huge difference. I was just curious, one has to fine tune based on the location. As you said, metformin improves the lung pathology, but doesn't affect the CFU. So I was just wondering, what is your views on that?
Balaji, I don't have an answer to your question. All I can say is that while so much is being done on animal models and different animal models, I think there's a real paucity of data in terms of, what Soumya introduced, the academic trials, where we get some information from either normal individuals or some patients. It's very difficult for me to say what will happen if we are always worried that if you take one of these host modulators, how will it affect the host function, and will we see more issues? I can't answer that question, Balaji. All I can say is something to think about. Sorry if you expected more.
I think we'll stop in the interest of time, and also I think BG needs to take rest.
Thank you, sir, for your thought-provoking presentation and providing us valuable takeaways for the future of TB drug discovery and development. Continuing with the area of TB drug discovery and development, the 7th session will be chaired by Professor William Vishai and Professor Balasubramaniam Gopal. Dr. William Vishai is currently a professor of medicine at Johns Hopkins University, USA. He also serves as the co-director for JHU Center for Tuberculosis Research. He served as the founding director of Howard Hughes Medical Institute, funded KwaZulu-Natal Research Institute for TB and HIV, South Africa. Dr. Vishai's interest involved TB pathogenesis and animal models of pulmonary infections. His work has led to a second-generation BCG vaccine, which is being advanced towards clinical trials. He was recently in the review panel of WHO Stop TB Partnership Coordinating Board. Dr. Balasubramaniam Gopal is currently at Indian Institute of Science, Bengaluru. With his expertise in structural biology, his research interests include gene expression, analysis in bacterial pathogens, and synthetic microbiology. He is associated as co-founder, advisor, or consultant to various startups and mid-sized biotechnology and pharmaceutical industries involved in production of biosimilars and active pharmaceutical ingredients. He is a fellow of Science Academies in India and is the recipient of the prestigious Shanti Swarup Bhatnagar Award. We have 5 talks in the session 7, each for 20 minutes. Following the talks, there'll be a brief question and answer session with the session speakers. I request the audience to hold their questions until then. I urge the speakers to please stick to their time limits. The buzzer will ring at 18 minutes. I request the speakers after their talks to be please seated on the dais for the question and answer session. Now I request the session chairs to kindly lead the session.
Good morning. So Bill, would you like to start off?
Welcome, everybody, to this session, session 7. I'm pleased to introduce Dr. Vinay Nandakori. He's an Indian immunologist, biotechnologist, and currently serves as the director for the CCMB India. He is known for his studies on kinase-mediated signaling networks on MTB. He received one of the highest Indian science awards, the National Bioscience Award in 2010, among many other awards. Welcome, Vinay.
You have a caller there. Yes, okay. 18 minutes, no problem. Thank you, Bill. Thank you, BG. What I'm going to talk about is the work that we started when I was at NII, kind of completed once I moved to CCMB. And this work, almost everything that I'm going to talk about is done in collaboration with my PhD mentor, Professor Umesh Vashne, who's sitting in the first row. So the project is started because at our conversation in 2012, and eventually we ended up publishing 2 papers on this. So I'll talk about that. So this is my current group, and I start always with acknowledgements, and all the work that is done by Sabanaz, who's a student who came to my lab around 7 or 8 years ago and done all the work in this. And most of my lab has now moved to CCMB, with other than Sidra Khan, who will be in the audience here somewhere. Lab used to and continues to work on serine threonine protein kinases and phosphatases, and which are direct, like PKNA, PKNB, PKNG are where most of our work is. Since we started off with that, but we had interest in substrates of kinases, so we moved into various areas like peptidoglycan synthesis, regulation of cell division, regulation of transcription, and at a little bit later on the host-directed therapy. And now in the recent past, we have also worked on evolution of drug resistance and role of DNA repair. For today, I will completely stick to this part. That done is completely, work is done by the one student only mostly. So we all know about the anti-TB drugs, and this is first and second line drugs. And in this particular audience, I shouldn't be talking about it anyway. If you are resistant to one of these drugs, it's called monodrug resistant tuberculosis. And if it is resistant to both of them, and then it's called MDR. And if it is resistant to 2 of them, followed by one of the second line drugs, it is XDR. And in all these things are fail, and it becomes so many drugs it is resistant to, we end up going to bedaquiline or delenamide for treating the XDR tuberculosis. So the point is, what is the mechanisms of the drug resistance? In many cases, it's kind of well understood that you have mutations in the target for the drug that we are giving. For example, if you are giving isoniazid, we are talking about mutations in INH or mutations in CATG. Such kind of things happen that is one way of getting resistance. But then you can also have mutations in efflux pumps, which are somehow decrease the concentration of the intracellular drug inside the bacteria, either by pumping it out more effectively or allowing or not allowing it to come in the first place. But then you can also have compensatory mutations elsewhere that sometimes lead to drug resistance. And sometimes some things, we can also have changes in the drug. For example, modifications like a methylation of the drug itself can change the way a drug works. So, there are examples for each of them. For example, mutations in the RPOZ for rifampicin resistance is a good example of modification of the drug, modification of the target. Overexpression of efflux pumps can change the intracellular concentration. This is another one which is commonly appears. And degradation of the drug or drug modifications is the third one. These are the mechanisms which I'm kind of giving you examples here, but essentially these are the most prevalent mechanisms. But despite all the mechanisms that are known, there are so many unknown mechanisms. People are looking into various other aspects, like, what are the other mutations which are not usual suspects. So this is indeed a problem because of the burden of MDR is quite high. Recently, we have sequenced around 200 samples, clinical samples that are available across India, and out of which around 40 odd were actually drug resistant, based on the bioinformatic analysis done by Dev's group at NII. So this is a serious problem, and we need to find out what are the other mechanisms that are out there which are not usual suspects. In 10 to 40% of the cases, we don't know what exactly is giving resistance. So that's exactly what our work is about. And as I said, this is done in collaboration completely with my PhD mentor, Umesh Vashne, and done mostly by, almost seriously by this girl, Sabha Naz. I've also collaborated with Dheeraj. Paritosh is one guy who did a lot of analysis and is an equal author in a paper with Sabha. Many other people across, and Yogendra Singh, my long-standing collaborator for almost everything that I do. So we were looking at DNA repair, as I said, with boss Umesh Vashne, and this was about, there are two mechanisms that will remove uracils. And what we identified is that, like, basically UNG-UDGB. So we made double mutants of it, and we went on to show that if you have mutations in DNA repair genes, you can actually get much higher levels of mutation accumulation, and that can actually lead to drug resistance. So this work was done almost last year, it was published. And at that point of time, we started asking a different kind of a question. So this is fine, where we are actually artificially introducing mutations in DNA repair gene. And that seems to be giving a certain benefits for the pathogen to survive better, number one, and also evolve drug resistance, because mutations that happen because of DNA repair genes are not getting corrected. So is it really there in the clinical samples? That was the question that we asked after this, right? And for that, Sabha has taken up this work of analyzing all the sequence data at that point of time that were available to her, around 2,600 of them, and coming from various parts of the world, for coming into every lineage possible. So what she has done is basically she has divided them into drug susceptible, MDR, XDR, based on the data that was already available. It is not something that we have done. Basically, and in each case, then she started looking for mutations along with Paritosh on in sequences that were there, like coming from an MDR strain or an XDR strain, but not sensitive strain. Means that is, these mutations are not present in drug sensitive bacteria, but only present in either MDR or XDR. With this particular thing and putting a stringent cutoffs, we ended up getting around 86 mutations, 86 different genes. And out of them, we had actually the known direct targets, which are usual suspects, the red ones. But what is interesting for us is that, our repair work, which was published in PLOS Path, kind of told us that repair genes are important and they may be having some role in drug resistance. Lo and behold, we actually ended up getting 5 DNA repair genes, but not UDG or UNG, but 5 different DNA repair genes to be associated with drug resistant strains, but not with drug sensitive strains. So our hypothesis is very simple. DNA repair is an important process. And if you do not have repair, what would happen is you will accumulate mutations. Some of these mutations may be more beneficial, and that may be contributing to a better drug resistance pattern. Means DNA repair mutations are most probably prelude to drug resistance is our hypothesis. To test this hypothesis, first of all, we had to validate that the mutations that we have identified in a GWAS study are even relevant. How do we know that just because a GWAS study gives mutations, they're actually important enough in the context of functionality of the protein, and also in the context of its, in some way it contributes to. So we took two candidates in this case. One is MUT-Y, the other one is UVRB. And both of them are involved in different base excision repair. And here you have the other one. So these two genes, we basically had mutations identified. They're involved in different DNA repair processes. I'm trying to not to go into the molecular details, straight to keep into this. So what we did was a very simple concept, where what we did is we knocked out these genes from the genome and replaced it either with a wild type copy of the MUT-Y or a mutant copy of the MUT-Y, which is involved in base excision repair. The mutation is the one that we identified in GWAS studies. So we at any point of time have 4 strains: wild type strain, a mutant strain in TB, a TB mutant strain complemented with an integrative copy of MUT-Y that is wild type, or a mutant copy of a MUT-Y that is identified in GWAS studies, the mutation that was identified in GWAS studies. So prior to this, we have using the mutants that were already available in Professor Varshney's lab, we have validated that the functionalities do get compromised. Not in vitro, but in smegmatis mutants that we have done this. But at a later point of time, we focused only on MUT-Y and we only focused on tuberculosis to do the further work. The first experiment that one does in any of these things is called mutation rate analysis. What we do in such kind of an experiment is basically we take, I mean, this is written with respect to Ang, but basically we take the strains and we eventually played them on antibiotic and calculate the mutation rate. So keeping this short, what we have showing is that here, this is a smegmatis work that I was talking about. You have both MUT-Y and complemented with the MUT-Y mutation that we've identified in GWAS, and UVRB and a mutant complemented identified. In both cases, what you see is the mutation frequency goes up when you have when you take away the base excision repair, I mean, any of these things, and you can see it going up. But when you complement it, it comes back down. But when you complement with the mutation that we identified in our GWAS studies, it does not come down anymore. So same thing is the case with UVRB, you can NER pathway. And in this case also the number of mutations are higher. Complementation brings it down, but not with the gene, I mean, with the mutation that we identified in GWAS. What it tells us that the mutations that we have identified in our GWAS studies actually are compromising the functionality of DNA repair proteins. So as I said, after this, we focused mostly on MUT-Y and we focused only on tuberculosis for going forward. And what we noticed is that this holds true even in case of tuberculosis. And we have done in this case with the tuberculosis mutant, you can see that the mutant is having a higher levels of mutation frequency. And when you complement it with the wild type, it comes down. Complemented with the GWAS mutant, it does not come down. All these things are what we call, from the context of tuberculosis, is in vitro studies, because you are actually growing it in a bacteria in the in BSL-3 and performing these kind of mutation studies. The next level that we obviously anybody will ask is what happens when you start seeing the host, what we call as ex vivo kind of studies, where we take in the peritoneal macrophages from the mice and they infect it with any of these 4 strains: wild type, mutant, complemented with the wild type, complemented with the GWAS mutation that we identified. And after that, basically, we looked at and performed this experiment. We did not find any difference whatsoever between any of these strains when we performed this experiment. So this was kind of baffling at that point of time. Maybe, you know, we are seeing a difference here. Then, in fact, this idea is completely, it came in a discussion between 3 of us, but Sabha is a highly motivated girl to perform an experiment of this kind, what we considered as ex vivo evolution kind of an experiment. So the experiment is simple. You infect, and after infecting, around 72 hours later, you lyse the cells, and you basically are 120 hours, we get the strain back and we grow them in vitro again. And we use that grown strain to infect again and do these 4 full rounds. And at the end of 4 rounds, we actually enumerate the CFUs, so we are evolving for a selection. And this is done with without antibiotics or with antibiotics. Essentially, it is 16 different ex vivo infection studies performed 4 rounds. And the whole thing was, it's like days and days of work. And most importantly, you can't make mistakes. It's a pretty huge experiment. Here is the experiment done. We have done it in the presence of 3 different antibiotics and no antibiotics. You can see very clearly that after these doing ex vivo evolution experiment, we find that the survival is way better in the case of MUT1 mutant. But when you complement it with the wild type, it comes down. When you complement it with the mutant that is found in GWAS studies, it is not giving you, so abrogating that particular complementation. And this difference becomes more pronounced when you have stronger antibiotics like either rifampicin or ciprofloxacin. Isoniazid not as much, but you can very clearly see that if you have mutations in the DNA repair gene like MUT1, it actually contributes to better survival in the presence of antibiotic or even in the absence of antibiotic, means it is putting a selective advantage. When we sent out this paper, the obvious question came that when you are saying it is giving you advantage, does it have the mutations that are necessary for it to, like, are there mutations in the target? And we have done the genome sequence of almost 10 colonies in each case. And what we identify is that it indeed accumulates mutations in the wild type, in the complemented strain. In one of these two things, we get a better accumulation of these mutations in when you don't have MUT1 or when you're complemented with GWAS mutant compared with the other 2 strains. So this essentially told us that having a DNA repair gene mutations actually help you better accumulate the mutations in when you subject them to stress of a different kind, which is antibiotics in this case. So, but all these things are good at again, this is an ex vivo evolution. The next best thing one has to do is some kind of a guinea pig kind of an experiment. We tend to use guinea pigs over mice because guinea pigs give us much better output compared to this. So, much better differences when it comes to marginal, strains which are not so drastically different. We have not done any mice experiment in this case. You can very clearly see MUT1 mutant actually gives you a slightly better survival rates in guinea pigs. And it comes down when you add MUT1, give the MUT1 back. It doesn't come down as much when you put MUT1, which is complemented with the GWAS, the mutation that we identified in GWAS. So all well and good, but these are experiments where we have done this, we have done this experiment, what you call as, independently. But in vivo, let's assume you have a situation, you have over a period of time accumulated a mutation, and then it is competing with the wild type in the same scenario and the same niche. So to understand this, what we have done is we have done a guinea pig competition experiment where we competed either the wild type with the MUT1 mutant or competed with complemented or completed with the GWAS mutation that we identified. And all I'm basically saying, the load is similar. This is almost my last slide. So what you can see is when you compete it with the MUT1 mutant, you actually get MUT1 mutant showing much better survival compared to the wild type, means it outcompetes the wild type. And when you do it with the complemented strain, that competitive advantage MUT1 mutant had is lost. But when you do the same with the GWAS, the mutation that we identified in GWAS studies, it is back. What it basically means that in a same niche, the DNA repair mutant that we identified, a DNA repair mutation that we identified in a repair, in GWAS studies actually provides it a competitive advantage to survive in different kind of niche. So that's basically what our story is about. And this was published just about 1 or 2 months ago in eLife. So thank you.
This is what.
So it's a pleasure to introduce the next speaker, and that's Dr. Dhananjay. Go there. Sorry about that. So the next speaker is Dhananjay Barclay, who's the Executive Vice President of Medical Research at Lupin. His specialization is in clinical pharmacology and clinical research. He has worked earlier with some of the best pharma companies in the world, including Hest, Glaxo, Novartis, Aventis, and Sanofi. He conducts global clinical studies for different novel chemical entities and biosimilars. He's a member of several regulatory committees that frame guidelines for GCP, bioequivalence, IPR phase 1, and ethical marketing of pharmaceuticals. He also is profiled by Tata Macro Hill as thought leaders among 22 corporate leaders. Dr. Barclay, please.
Thank you. First of all, I want to confirm whether you're able to hear me all right and see the slides. Okay. So at the outset, I would like to thank the organizers for inviting me, and I'm sorry, my apologies that I could not be present face to face. And thanks everyone at THISTI for giving me this online opportunity. Thanks to Dr. Dinesh Mahajan for coordinating with me. And my first disclaimer is that I'm not a TB researcher any longer. I used to be, more than a decade ago. But, I would like to talk about some of the new technologies that can be repurposed for the benefit of anti-TB drugs. So my focus will be on what are the areas where new innovations can be brought with respect to anti-TB drugs. So the government, has made a very ambitious aspiration announced a few years back that they want to eliminate TB by 2025, which is only 2 years from now. The challenge is very, very daunting, as all of us know, because the problems for using anti-TB drugs particularly have remained the same for the last few decades. The longer regimens, higher dosage, non-compliance, drug-induced liver toxicity, inadequate access with respect to extrapulmonary sites, and rather fewer technologies being brought into this particular field, particularly because of the high cost that are involved in bringing them. So, if you want to really talk of different types of innovations, mostly incremental innovations drive the new drug research. Rarely you get the breakthrough innovations. Sometimes they're disruptive. Now, the best thing to differentiate is that if there is a high market impact but low technology progress, as described by Jim Kalbach, it's called disruptive. But when it is high technology progress but low impact, it is generally breakthrough. And when both are present, it is called the game changer. So some of the innovations that we will see in the next few minutes will be of the game changing type also, but mostly breakthrough or disruptive. So if you talk of repurposing drugs and technologies, I think the best example is INH. We have seen moxifloxacin is another example which can shorten treatment. But if you look at some of these areas, they may look wild. For example, digitally observed treatment, a new definition of DOTS to ensure compliance, topical treatment for extrapulmonary TB, can we counter liver toxicity with of anti-TB drugs? And the last area I'll touch upon is battleground macrophage, which is very, very important. And some of these things, I want to give this quote of Isaac Asimov, who said the most exciting phrase to hear in science, the one that heralds the most discoveries, is not necessarily eureka, but people say it's funny. So many innovations have started as funny innovations, but then they have changed the particular therapy areas in the past. So if you want to really improve TB compliance in both public-private sector, what are the ways in which we can do it? Obviously, all of us know that the main challenge continues to remain the compliance, and DOTS was mainly born to tackle that problem. Still, we don't have satisfactory cure rate, mainly because of non-compliance. So can we use some of the recent technologies that are available for other fields? For example, if you look at some of the drugs that have been introduced in the recent past, one such product is already available. Because of the availability of new sensors that can be swallowed, it is possible to track the ingestion of anti-TB drugs in the real time. And this has been approved as ingestion event marker. And these IEMs are grain or a grain of sand size sensors, which can be embedded or over encapsulated with the tablets. They're designed to communicate a signal in a time-stamped confirmation way for the ingestion of the external device. And the first drug that was introduced in 2017 was by Otsuka, which was the drug aripiprazole, Abilify in psychotic patients, in conjunction with a digital therapeutics company called Proteus. So this was a very, very innovative device where the patient swallowed a pill containing aripiprazole. And when it came in contact with gastric fluid, the magnesium and cuprous chloride, which were added as part of the IEM, they reacted to reactivate and power a small device which communicated a signal to an external patch that you can see here, which can be stuck on the body. And then there is a mobile app which picks up that signal, and the doctor can review the medication of the ingestion event that is recorded as a timestamp. So you can make out every drug administration and compliance can be tracked by healthcare workers and by the caregivers. I want to show you a small video now, which is a latest technology which is available, approved by FDA, although it has not been put for any particular drug use. It's just a 1-minute clip.
This digital pill is ready to revolutionize remote patient monitoring. Containing an adjustable sensor, this capsule provides convenience for patients and accuracy for clinicians. Inside is any oral medicine that a trained and licensed pharmacy can fit into a pharmaceutical-grade capsule. Shortly after being swallowed, in as little as 1 minute, the pill sends a message to a small, lightweight, wearable reader. Once the reader receives notification that the pill has been ingested, Bluetooth technology forwards its message onto the app, which has been downloaded onto the patient's smartphone. The patient wears the reader until the signal is detected. It can be removed between doses and is easily recharged by being placed on the wireless charging pad. The patient receives additional support because their healthcare provider is able to view, track, and trend ingestion data in the accompanying dashboard. The feature is especially valuable as remote real-time monitoring is evolving and developing the standard of care for patient engagement. If the clinician sees on their dashboard that a patient has veered from their prescribed medication regimen, immediate follow-up can provide adherence support or other interventions that help patients in real time. Accuracy.
So this is the shape of things to come. If although this technology will be expensive to begin with, but if the aid agencies get together and try to develop something like that for the anti-TB, particularly fixed dose combinations, I think things can revolutionize. The next thing I want to talk about is the intralesional TB therapy as a potential exploration, something like a topical warfare. And I think this technology is again not new. There are products available which I will show, but these are in situ forming implants, which is primarily meant for extrapulmonary tuberculosis. I think all of us know the challenges involved with extrapulmonary TB, where there is a granuloma formation. And because of that, the drug penetration inside the TB focus is not very satisfactory. And therefore, the treatment duration for extrapulmonary TB is far longer than a pulmonary TB. Some work has been already done in the University of North Carolina on rifabutin ISFI, and they have shown that long-acting biodegradable ISFI with rifabutin was able to maintain high plasma concentration for almost 4 months, and it prevented MTB infection and cleared the acute MTB infection in animals. So animal studies are done. This mechanism is very simple. you inject a drug, a biodegradable polymer in a liquid phase inside the skin. It is a subcutaneous injection. It is generally PLGA based ISFI, which are long acting, and then it forms something like a solid implant, which will remain inside the skin, and it will keep delivering drugs for a long duration. So 4 months has already been tested, as I mentioned. So think of, a lymphadenitis, for example, being treated. So these are two examples of drugs which are already being used for HIV. Rilpivirine is one example. The product is called Eligard, and it uses an arterial delivery system. The advantages are obvious. This technique does not require surgical removal because they slowly degrade into non-toxic byproducts. And the fine-tuning of release kinetics is possible by modifying the ratio of polymer to solvent or by rate-controlled additives, which reduce the side effects also. Basically, they provide ultra-long release of drugs, flexibility, and choice of active ingredients, and number of combinations also is possible. And the ability of surgical removal in case of, suppose if the patient is having high liver enzymes and you need to stop, obviously you can just surgically remove the implant. That is also a big advantage with that. And ability to integrate multiple drugs is what I mentioned. So obviously this technology, if used, will make it possible to release anti-TB drugs to extrapulmonary sites, particularly lymph nodes around the pleura, for example, even inside the vein for tuberculoma, to provide 6 full months of local treatment or even longer if it is required. Lymphadenitis seems to be the low-hanging fruit with respect to ISFI because these are easily accessible. They are subcutaneous, so injecting into that site where implant is formed will be a very easy thing to do, and it will slowly release the drug. It will also ensure that the other lymph nodes nearby through the lymphatic network we carry the drugs, and the systemic toxicity of the drugs will definitely reduce. So, we have liver toxicity as the problem for some of the drugs, and I think we know that bedaquiline has issues like QT prolongation, hepatic dysfunction, and metabolism by 3A4. 3A4 liability is well known, and there is a formation of a metabolite called M2, which doesn't have add to the efficacy, but it leads to QT prolongation. So there is some work I've seen going on, but I think if bedaquiline can be developed as an inhalation formulation, then it will dramatically reduce the toxicity, but provide all the benefits for a pulmonary TB. There are other ways of reducing the liver toxicity, and N-acetylcysteine is something that is, projected to be one such drug. Currently we know that 11 to 15% of patients, discontinue the anti-TB drugs because of hepatotoxicity. So this is one of the challenges faced when the treatment is started in the first few months. It is known that it produces oxidative stress, lipid peroxidation, and decreased glutathione levels. And there is enough evidence to show that the role of GSH in T cell activation, phagocytosis, etc. So when there is a reduced level, that doesn't help. And N-acetylcysteine is shown to increase the GSH levels. At the same time, it is also beneficial in conditions associated with lipid peroxidation and decreased GSH. So the hepatoprotective effects are well known, although there have been some studies, but they are not properly randomized controlled trials to show that addition of N-acetylcysteine to DOTS regimen, for example, in the newly diagnosed cases will provide some relief. And last few minutes, I want to talk about the battleground, that is macrophage. And I think this is a very, very important aspect of tuberculosis, which has not been taken so much into account. I heard some questions about the host-directed therapies. And I think in case of COVID, we saw that happening with ivermectin, hydroxychloroquine, did produce some effects in patients, which was a host-directed mechanism, and therefore it was not strain-specific for COVID-19. So there is a definite distinct role of macrophages, which is well known in case of Mycobacterium tuberculosis, because they use the Trojan horse strategy. And I think glucon particle embedded therapies are very much possible. So I think some work is already being done. I'm going to talk about that. I think many of you may be familiar. Dr. Amit Mishra at CDRI has tried, various different things. There is a Integral University of Lucknow. I saw some papers, talking about this area. So the targeted delivery of anti-TB drugs using macrophages has not been into clinical trials as such. There's only experimental work. But rifampicin, being targeted delivery through glucon particles, there are some studies and also with rifabutin. So what are these glucon particles? They are 2 to 4 micron spherical hollow shells isolated from Saccharomyces, and the hollow cavity of the particle will allow encapsulation and absorption of the drug. And while the surface contains a specific type of beta glucon, which is coming under the GRAS list, so therefore clinical trial permissions may be easily possible, but which facilitates receptor-mediated phagocytic uptake also by macrophages. So the efficacy of glucon particle containing rifampicin has been seen at a sub-minimum MIC levels, and they have demonstrated significance of the targeted rifampicin release. And I've seen some even photographs of the organs in a placebo-controlled trials in mice. And this was a paper, I think, which was published, I think last year, which is a combination of a Korean university and I think University of Worcester in Massachusetts. So I'm sure there is some work going on in India, and I mentioned about the Integral Lucknow University. So if, say, for example, the glucagon particles containing rifabutin has also being studied. So such particles containing anti-TB drugs also will lead to reactive oxygen species and nitric oxide induction of innate immune response in the macrophages, increasing the mycobactericidal activity of the drug. So autophagy also has been seen. So the basically Trojan horse strategy that the Mycobacterium is using, this is a way of reversing that by sending in impregnated soldiers inside the horse containing anti-TB drugs. So strategically, it looks a very, very appealing idea, and there should be some work done in that respect. So using glucon particle for targeted delivery of anti-TB drugs will definitely help in shortening treatment, because we know that it is the remnant mycobacteria within the macrophages which lead to the lengthy therapy that is required, because they remain safe within macrophages. And I think Thisti had done some work which brought me there a few years back to look at some of the molecules that work on the host-directed mechanisms, dealing with macrophages. So I think work is happening there, but I think it has to be put on the pedestal, and people have to come together. Government has to fund such efforts. And if that happens, I think the battleground macrophage can be easily won. That will tremendously make a boost. It can be a game changer simply by reducing and shortening the therapy to, say, imagine 1 month, 2 month type therapies. Of course, the time will tell. So many times I've seen conducting research in various therapy areas is that it's important to, everybody sees, what is in front of you, but thinking what nobody else has thought becomes very, very important in conducting research projects. And I think that if you apply, for example, this picture is seen as a nice picture of a sunrise, but few will see a small fetus hidden inside. And, so that can really differentiate a good research, which can be a game-changing type of a research. And I think that can be the dawn of TB-free India. In my opinion, if some of these areas, and I think there are multiple more areas that are possible, but we really need some game-changing breakthrough type of innovations, which will make a deep impact on the way we treat tuberculosis, both of the pulmonary type and the extrapulmonary type. And I think institutes such as yours can really translate that into clinical medicine. So thank you for your attention, and again, thanks the organizer for inviting me here.
Thank you, Dr. Buckley. We have time for one or maximum of two quick questions. We're running 20 minutes late. Amit Mishra.
Hi.
I would like to take you back to 2012, when you walked out of the project of making inhalations of a drug combination of isoniazid and rifabutin in vicarial sutures. And I would request you to share as much as possible publicly about the reasons why Lupin took this decision.
Yeah, so first of all, I'm not here representing Lupin's policies for tuberculosis, but I think we have seen worldwide that most pharma companies, who started with tuberculosis anti-infectives, and then they branched into number of newer areas and therapy areas. And at that time, Dr. Mishra, when we interacted, I was very new to Lupin. That was my second stint, and I was more known for my first stint, which was mainly in tuberculosis, where we did a lot of work internationally. Lupin at that time was branching into new chemical entities in which we had invested a lot of money. We wanted to get into biotech research, and this particular project was going on for quite a few years. And pharma industry bets on some research projects for a short time, and if things do not materialize into commercial applications. And one of the reasons why it was a brilliant project, excellent academic impact, but when we did the commercial viability was one of the issues, because the cost of the particles was more than rifabutin, if you remember, Dr. Mishra. So therefore, when management saw that entire perspective, that it is going to cost us more for the delivery of the drugs, and then there is a long, it would be seen by most regulatory authorities as a complete new invention. And as a result, requiring at least 10 to 12 years of development through all the phases, I think it was decided not to commercially take it forward. As I said, that even the digitized therapy that I talked about, if you look at the cost per tablet is going to be high. But my point here was about government institutes coming together, getting government funding, identifying one or two key projects, and commercializing it is the key for a game-changing innovation. I hope I answered that, Dr. Amit.
Yeah. Hi. So regarding the digital sensing platform that you showed, what is the life of the sensors? Because the TB therapy is going to be long. And secondly, once the therapy will be over, how would they get eliminated from the body?
No, it contains actually dietary, grade copper and magnesium, as I mentioned, and it gets passed out through the tablet. So it doesn't remain in the body. So it is the ingestion event marker. So the moment you swallow and it goes into contact with gastric juice, it gets converted to a signal that is picked up. So therefore, every single ingestion of the drug will be marked on a timestamp. So nothing remains in the body.
Okay, thank you.
Go on to the next speaker. Krishna has got an announcement.
I request all the poster presentees to basically go back to the poster boards and install your posters, because otherwise it will not get evaluated. And the whole effort of yours will be gone waste. Thank you.
It's a privilege to introduce our third speaker, the hardworking co-organizer of this wonderful symposium, Dr. Ramandeep Singh, who's a professor here at THSTI. His research has made significant contributions to key metabolic pathways, including toxin, antitoxin systems in organic polyphosphate, itaconate assimilation, pathways and key other pathogenesis mechanisms of MTB. His targeted phenotypic studies have led to key screening assays and led to novel drug targets and scaffolds against MTB, which show novel mechanisms of action and have great potential for first line and synergy with drug-resistant treatment regimens. Welcome, Ramandeep.
I hope I am audible. So good morning, everyone. Today I would be talking about our work on inorganic polyphosphate. So what my lab works on is that we using different genetic tools. We have tried to identify the role of, understand the role of various metabolic pathways in physiology and pathogenesis of Mycobacterium tuberculosis. As a second theme of the lab, we have tried to standardize various high throughput phenotypic target-based and macrophage-based screens to identify small molecule inhibitors against Mycobacterium tuberculosis. And we also have done some work evaluating new vaccine candidates. So in my lab, these are the various metabolic pathways that we work on. We work on toxin antitoxin systems in collaboration with Krishan. We work on inorganic polyphosphate metabolism. We also work on itaconate dissimilation pathway, and we also have some work on a few transcription factors which have a role in antibiotic resistance. And we also have started some work on the role of small RNA-mediated gene regulation and its role in the physiology and pathogenesis of Mycobacterium tuberculosis. And few of the conserved hypothetical proteins, we are also working on them. So today, I would be talking about the work that we have done on inorganic polyphosphate metabolic pathway of Mycobacterium tuberculosis. So what is inorganic polyphosphate? Inorganic polyphosphate is a linear polymer of phosphate, which is linked by high energy phosphoanhydride bond. And there is a lot of literature available which shows that it has multiple roles. It acts as a reservoir of energy. It also substitutes for ATP in various metabolic enzymes. It also interacts with chaperone proteins and prevents protein degradation. It also interacts with various transcription factors and regulates the expression of various virulence-associated proteins. Polyphosphate in other microorganisms also has a role in biofilm formation. It also has a role in metal detoxification. And in other bacterial system, it had been shown that polyp dysregulation also impairs the virulence of various microorganisms. And polyphosphate also acts as a regulator of innate and adaptive immune response. So when we started this work, so these are the enzymes which are involved in polyphosphate metabolism. Polyphosphate kinase is the main enzyme which is involved in the synthesis of polyphosphate. And this is a reversible reaction. Polyphosphate kinase can also use ADP and poly-P to form ATP. And polyphosphate is degraded by polyphosphatase enzyme into inorganic phosphate. And it has been shown in other bacterial systems that PPP-GPP, which is a stringent response molecule, is an inhibitor of polyphosphatases. So the genome of Mycobacterium tuberculosis, it encodes for the homolog of polyphosphate kinase 1, which is involved in the biosynthesis of polyphosphate. And it also encodes for enzymes which are involved in poly-P degradation. Polyphosphate kinase 2 is the enzyme which uses polyphosphate as a substrate for synthesis of ATP, GTP. And polyphosphatase is the enzyme which degrades polyphosphate into inorganic phosphate. So the questions that we wanted to ask was, is there any poly-P accumulation in mycobacteria during later stages of growth or upon exposure to stress conditions? The second question was, is polyphosphate homeostasis essential for pathogenesis of Mycobacterium tuberculosis? What are the metabolic pathways that are regulated by inorganic polyphosphate? And are these druggable targets so that we can identify a small molecule which has a novel mechanism of action? So we published a paper in 2013 in JBAC where we had developed a luciferase-based assay system and showed that there is accumulation of polyphosphate at later stages of growth, as well as upon exposure to different stress conditions such as acidic stress, oxidative stress, nitrosative stress, and nutritional stress. And we also showed that this accumulation of polyp was PPP, GPP dependent, as there was less accumulation of polyp in the Rayleigh mutation. Subsequently, there was another study from a group in Johns Hopkins which showed that there was accumulation of polyp in Mycobacterium tuberculosis at later stages of growth during nutritional stress, as well as upon exposure to low oxygen stress conditions. So as I mentioned earlier, Mycobacterium tuberculosis encodes for 2 homologs of exopolyphosphatases. So we and others have shown that using purified enzymes, we have shown that these enzymes, they indeed possess exopolyphosphatase activity, and in addition to exopolyphosphatase activity, they have ATPase and GTPase activity. And the activity of PPX2 enzyme was slightly better than the activity of PPX1 enzyme. And we, along with others, also showed that PPP, GPP inhibits the exopolyphosphatase enzymatic activity in vitro. So to understand the role of these exopolyphosphatase in physiology of Mycobacterium tuberculosis, so we generated both single exopolyphosphatase strain as well as a double mutant exopolyphosphatase strain in which both PPX1 and PPX2 were deleted. And we showed that there was more accumulation of polyphosphate in the double mutant strain in comparison to the single mutant strain, and we did not see much difference in polyp accumulation in the single mutant strains. So, so to understand what happens if polyp accumulates, how does bacteria adapt to different stress conditions? We checked a number of stress conditions, and I'm just showing some results. We found that the double mutant strain had a defect in biofilm formation, and it also had a defect upon exposure to nutritional stress. And the double mutant strain also had a defect in macrophages. Then we have also checked the survival of these strain in guinea pig model of infection. And we observed that the exopolyphosphatase double mutant strain was significantly attenuated for growth at both 4 weeks and 8 weeks post-infection in both lungs and spleens of infected animals. And similar observations were also observed in our histopathology analysis, where we observed less tissue damage in sections of animals infected with the double mutant strain in comparison to the wild type strain. So, and this was a cumulative effect because the differences that we observed were not as drastic in the case of single mutant strain, which we observed for the double mutant strain. So the next question that we wanted to ask was, what are the mechanisms by which these exopolyphosphatases, they contribute to the pathogenesis of Mycobacterium tuberculosis? So we did a RNA-seq experiment from isolated RNA from mid-log phase culture of both wild type and double mutant strain, and observed that around 200 genes were differentially expressed in the double mutant strain when you compare it to the wild type strain. What was interesting to observe was that most of the genes which were down-regulated in the double mutant strain were known to be upregulated in bacteria when they are exposed to low oxygen growth conditions. So which means that they belong to the dormancy response. So what is a dormancy regulon? So Dr. Jayathyagi had identified a 2-component signal system, which is called DEVA-DEV-S or DOS-S or DOS-T. A lot of ligands are known which are known to activate these sensor kinases, which we call DOS-S and DOS-T, which leads to activation of DOS-R. And then DOS-R activation results in upregulation of this DOS-R regulon. So the question that we wanted to ask, that DOS-P is also one of the signal for this, polyp is one of the signal for this 2-component system. And does polyp accumulation leads to inhibition of activity of DOS or DOS3 sensor kinases. So then in collaboration with Deepak, who is here, what we showed was that poly-P was indeed able to inhibit the autophosphorylation of two sensor kinases, DOS-T and DOS-S. And this was a very specific observation because other sensor kinases that we use in our assay did not see any autophosphorylation effect. And then we also did some MST assays and showed that DOS-S and DOS-T, they bind to the poly-P. And as I said, it is a very specific effect, and this only inhibits the two component signaling of DOS-S and DOS-T. So, so we published this story, and this is the model where we showed that when there is accumulation of poly-P, it binds to the two sensor kinases, DOS-T and DOS-S, which inhibits its autophosphorylation and which in turn leads to suppression of the dormancy regulated genes, which are also virulence factors. We have also done some host RNA-seq analysis, which I am not going to present today, but we observed that the expression of various antimicrobial pathways were down-regulated in lung tissues of animals infected with the double mutant strain. So in addition to the exopolyphosphatases, we have also done experiments to understand the role of polyphosphate kinase 1 in the physiology and pathogenesis of Mycobacterium tuberculosis. So before we started, there was a lot of literature available on the biochemical characterization of polyphosphate kinase 1. So what was known was that polyphosphate kinase 1 undergoes autophosphorylation, and histidine at 491 residue was involved in the autophosphorylation of PPK1 enzyme. And in the same study, they had generated a lot of mutants, and they showed that this purified protein, histidine 491 alanine, there was neither any poly-P synthesis or ATP synthesis, thereby suggesting that this is the site involved in the catalytic activity of this enzyme. And there was also literature available where it has been shown that the two component signaling system, regx3, senx3, when it is activated, it leads to activation of transcription of PPK1. PPK1 then results in the synthesis of inorganic polyphosphate. This can act as a phosphate donor for MPRB, which is another sensor kinase. And activation of this sensor kinase leads to activation of MPRA, which in turn leads to activation of sigma factor E. And there's a feedback mechanism which is going on here, which results in synthesis of more and more poly-P. And there was another study which had shown that SIG-B in interaction with RPPA, it binds to the promoter region of PPK1 and drives the synthesis of PPK1, which results in formation of inorganic polyphosphate. So in the paper which we published in JPAC, we had constructed the mutant chain for PPK1 and shown that there was no poly-P synthesis in the mutant chain, suggesting that this is the main enzyme which is involved in poly-P synthesis. And we did a bunch of stress experiments and pathogenesis experiment and observed that the mutant strain was indeed attenuated for growth when you compare the cross pathology in comparison to animals infected with the wild type strain and complementary strain, and same was reflected in our CFU analysis where we observed there was a 100-fold growth defect, as well as in histopathology we observed very minimal tissue damage. So in the same study, we had shown that there was a growth defect in stationary phase of growth, and the mutant was more susceptible when you expose it to nitrosative stress or in macrophages. And we had also shown, which was verified later by others, that poly-P deficiency enhances the susceptibility of Mycobacterium tuberculosis towards isoniazid. So, but at this point of time, we did not know what are the exact mechanisms by which deficiency of poly P results in attenuation of Mycobacterium tuberculosis. So that is the time when Saurabh joined the lab, and this was his question for his PhD. So his work helped us to understand the mechanisms by which polyphosphate deficiency results in attenuation. So the first thing that we tried was to try to find out what are the poly P interacting proteins. So we did a pull down assay using biotinylated poly P, and after a pull down we subjected to mass spec analysis, and we observed that there are around 25 proteins which were interacting with polyphosphate. We used the selfie extract prepared from either mid-log phase culture or late log phase culture. Interestingly, 7 proteins were common between both the conditions, and these proteins, they belong to the family which we call acyl-CoA carboxylase enzyme. So then what we did was we did biophysical characterization experiments and showed that both ACC-A1 and ACC-D1 indeed interact with poly P. And we also did some molecular docking studies that identified the key interacting residues which are involved in binding of poly P with these enzymes. So these enzymes are involved in synthesis of malonyl-CoA and methyl-malonyl-CoA, which are essential for the synthesis of various lipids in Mycobacterium tuberculosis. And I would also like to mention that 9 of these proteins were known to be ATP binding proteins, which suggests that poly P might be able to replace ATP for the function of these enzymes. Then in addition to the pull down assays, we also did some RNA-seq analysis, again from the mid-log phase cultures of RNA isolated from the wild type strain as well as the mutant strain. And we observed that the transcription of 10% of the genes were differentially expressed in the mutant strain in comparison to the wild type strain. And interestingly, the expression of genes involved in type 7 secretion or PDM biosynthesis were down-regulated in the mutant strain. So we verified this by Western blot analysis and observed that the expression of CFP10, which is a type 7 effector molecule of a type 7 secretion system, was indeed reduced in the mutant strain in comparison to the wild type strain, as well as the PDM biosynthesis was reduced in the mutant strain in comparison to the wild type strain and complementary strain. This also correlated with our pull down studies because acyl-CoA carboxylase is involved in synthesis of the precursors which are involved in lipid biosynthesis. So both these factors are known to cause phagosomal rupture, which results in dissemination of disease, and they modulate phagosome maturation function. So we then did some confocal experiments and observed that there was more phagosomal lysosomal fusion in macrophages which were infected with the PPK1 mutant strain in comparison to the wild type and the complementary strain. So, so what we concluded was there was a reduced expression of virulence associated factors in the mutant strain, and the enhanced phagosome lysosome fusion in the mutant strain might be responsible for the attenuated phenotype that we observed for the mutant strain. So when while we were doing this study, there was another study which came in PNAS which showed that polyphosphate is secreted by Mycobacterium tuberculosis and is recognized by GPCR receptor, which facilitates the survival of Mycobacterium tuberculosis in eukaryotic cells. So we have also done some host RNA-seq analysis, and we observed that a number of pathways, antimicrobial pathways such as expression of inflammatory cytokines involved in recruitment of monocytes and neutrophils were indeed down-regulated in the mutant strain, and we then did some Nile rat staining and showed that there was less neutral lipid accumulation in macrophages infected with the mutant strain in comparison to the wild type strain. So this is just the model which we showed that there is accumulation of poly P, which results in synthesis of PDM and secretion of ESET6 and other type 7 secretion system, which is involved in secretion of pro-inflammatory cytokines, and this indeed is required for the virulence of Mycobacterium tuberculosis. So using this assay system, we then developed a high throughput screening assay where we screened a library of around 2,300 compounds and identified 11 FDA approved drugs, which we showed were specific in their ability to inhibit the PPK1 enzyme, and they inhibited this enzyme in a dose dependent manner. Then we also did some biochemical and biophysical methods, and we showed that these drugs indeed bind to PPK1 enzyme, and maximum binding was found in this compound 11, which was able to bind to PPK1 enzyme in a dose-dependent manner, and it also inhibited both poly-P synthesis and ATP synthesis activity of PPK1 enzyme in a dose-dependent manner. So then we collaborated with Shailendra, and he developed a three-dimensional model for this PPK1 enzyme and did some molecular docking studies. And we identified some key interacting residues, which we verified later by mutational analysis, and that these residues are indeed involved for the binding of this inhibitor with PPK1 enzyme. So since PPK1 is a highly conserved enzyme present in various bacterial pathogens, we purified PPK1 enzyme from other bacterial pathogens and observed that this inhibitor was also able to inhibit the enzymatic activity of PPK1 homolog isolated from E. coli and Klebsiella. And we also performed molecular docking studies and showed that the binding pocket for this inhibitor between homologs from either Mycobacterium tuberculosis or E. coli or Klebsiella were highly similar to each other. And we also did some MD stimulation studies. We showed that the complex between the protein and complex inhibitor was stable for a very long time. So while we were doing this study, there were other known inhibitors for PPK1 enzyme for other bacterial pathogens. So we have tested them also. And Gallene was one of them. And we found that Gallene was able to inhibit the PPK1 enzymatic activity associated with the MTB homolog, whereas no inhibition was seen with the other known drug, which is mesalamine. Then we have tested this molecule in macrophage model of infection and observed that this molecule was able to inhibit the intracellular growth of Mycobacterium tuberculosis, and it also showed synergy with isoniazid, betaquolin, and protamanid. However, in our mice study, we saw a very marginal growth defect where we observed a defect of around 7-fold in spleen and 4-fold in lungs. So the molecule that we have identified was showing good activity in macrophages, but not in mice. But now we have other molecules also, which we will evaluate in future. So I will skip these two slides, but what I wanted to show here was that these both these mutant strains were showing efficacy better than BCG in our guinea pig model of infection, and they were as safe as BCG in SCID model of infection. So in the end, I would summarize that what we have shown is that poly-P homeostasis is essential for the virulence. We have identified the metabolic pathways which are regulated by polyphosphate levels. We have a molecule which has a novel mechanism of action. We observed synergy with both first line and second line TB drugs. And we have some vaccine strains which are as efficacious or better than BCG in guinea pig model of infection. So most of the work which I presented was done by Saurabh, who was a poster today. I would request everyone to visit that poster and interact with him. And in the end, I would like to acknowledge collaborators. Deepak helped us with the 2-component signaling work. Shailendra helped us with the molecular docking work, Amit Zygam with the immunology work, Sam for providing us for the SCID mice, and we had also done some metabolomic studies, which I have not shown today. I would also like to acknowledge my students. They have lot of poster presentations. We had 6 posters yesterday and 2 posters today. I would request everyone to interact with them and guide them about their work. And in the end, on behalf of the organizers, I would thank everyone for coming here and participating in the conference. And I hope that we will have many similar conferences in future. Thank you.
So it's a pleasure to introduce the next speaker, and that's Dr. Bichitra Biswal. So Bichitra is a scientist at the National Institute of Immunology in New Delhi. His research interests include structural biology and X-ray crystallography. His laboratory at NII aims at understanding the structural, functional, and essentiality aspects of proteins in metabolic pathways using structural biology, biochemistry, and in vivo studies. He utilizes the outcome of these basic studies for drug discovery, particularly designing anti-TB molecules through a target-based approach. Dr. Biswal has published several research articles in peer-reviewed journals. He has received numerous awards, honors, and fellowships. Dr. Biswal, please.
Good morning, everybody. Can you hear me? Yes. So by training, I am a structural biologist, more precisely a crystallographer. So my laboratory currently works on 2 aspects of mycobacterium tuberculosis proteins. On 2 aspects. One aspect is we are trying to understand mechanistically how MTB makes small molecules, such as amino acids. And the second aspect is also the mechanistic studies, how virulent factors are secreted out from the type 7 secretion system. Okay. And I will be able to explain you how using X-ray crystallography or structural biology, one can design new anti-TB molecules. Okay, so, and one of the major theme of this conference is to discuss science which could potentially lead us to identify molecules that could shorten the current TB therapy, which goes as high as 24 months. So in my view, understanding or harnessing the TB biology perhaps is a major way to achieve this. And when mycobacterium tuberculosis is concerned, there is a fundamental difference. We all know proteins or enzymes are the working horses of cell. Every cellular process in our body either works through proteins or through enzymes. So therefore, an amino acids are building blocks of proteins or enzymes. And there is a difference here. A small microorganism such as Mycobacterium tuberculosis, which causes TB in humans, can make all 20 amino acids de novo, all 20. However, a complex organism and most brilliant organism in this planet cannot make 9 amino acids. They are F, H, I, K, L, M, T, V, and W. So very basic question asked as a physics and mathematics person, a very basic question. If you deprive MTB with one of these amino acids, the organism will die. It's very simple, but can that be done? And this is where my laboratory is putting efforts to block the synthesis of one of the essential amino acids, particularly histidine. And on this pathway, I have been working for last 15 years. And I have characterized this pathway with all possible ways, except a couple of papers from here, which are on largely on COVID and monkeypox. All the papers that I have shown here largely involves in understanding the pathway of histidine. And MTB makes histidine from a precursor molecule PRPP, pyrophosphate PRPP, phosphoribosyl pyrophosphate, by utilizing 10 enzymes in 10 steps. And among all the amino acid biosynthesis pathways, this is perhaps the most expensive pathway in terms of employing number of enzymes, and also energetically it is very expensive. It consumes around 12 ATPs to make histidine from PRPP. And with collaboration with Ramandeep, Nisit, Amit, and Ranjan from ICGB, and all three are from THSTI, and I have produced 7 PhD on understanding this pathway. I am going to present the conclusion of my talk. And the conclusion is that what we are seeing now, after 15 years research on various aspects, that if you chemically inhibit this pathway, and there is a chance that we can clear MTB both in vitro and in vivo model. Okay, and how we have designed such molecules through structural biology approach, this is the theme of my talk today. And in addition to histidine, which my laboratory largely working on, there are other amino acid biosynthesis pathways currently are in focus in terms of designing target-based anti-TB drugs. And one of other studies that is going very well is from Professor Surya's lab from IISc Bangalore. He has shown that a molecule called PRP, a repurposed drug PRK, and that allosterically inhibits the arginine J of this pathway. And he has shown that there is a good amount of bacterial clearance in mouse model. And here there is another group in Albert Einstein College of Medicine, Professor Bill Jacobs group, and Ramandeep here also works, does some work on methionine pathway. And there is a group in from Eric Rubin group. They also work on tryptophan biosynthesis pathway. And my laboratory at the National Institute of Immunology works on the histidine biosynthesis pathway. And over the years, we have determined a lot of high resolution structures of the histidine biosynthesis pathway enzymes. Only one structure left. We have not been able to crystallize that protein, but we have been trying. Once we get that structure done, then all the structure from the pathway is done. All 10 structures are done. And that will give us more opportunity to design inhibitor through structural biology approach. And I want to put one point here. Before that cell paper from Bill Jacobs, from Eric Rubin's group came, it was largely perceived that amino acid biosynthesis pathways cannot be targeted because the bacillus will take this metabolite from the host. But that paper, it's a revolutionary paper in my view. That paper says that host has a mechanism to kill any pathogen. One of the mechanisms or one of the ways that to deprive the pathogen with amino acids. That is the one of the mechanisms. And the host, and they have shown in that paper that the IDO is upregulated, and that IDO sequesters the extra tryptophan from the cell, thereby in a way to kill the pathogen and or compels the pathogen to make its own tryptophan. And that's because of that, because of the de novo pathway of the tryptophan, the bacillus is able to escape the immune evasion. And the similar mechanism we have shown here in the case of histidine. And host also try to kill the pathogen by sequestering or by starving the pathogen with histidine. But the pathogen de novo, it prepares its de novo biosynthesis pathway to escape this mechanism. How host does this? It upregulates two enzymes, HAL and HDC, histidine ammonia ligase and histidine decarboxylase. So they sequester the extra histidine present in the cellular milieu in order to kill the bacteria. But bacteria, because it has a de novo pathway, it escapes that mechanism and prepares. Okay, and we have, for the first time through mass spec, we have shown that host tryptophan histidine level goes down with respect to the time. And this is where the adaptive immune response is very, very important in upregulating these enzymes through IFN gamma pathway and try to kill the pathogen. Therefore, in our view, if you can host immune response combined with blocking the His pathway is perhaps a new way to curtail MTB infection. So I am going to present one enzyme through which, or by studying that enzyme, we are able to design some inhibitor, and that enzyme is imidazole glycerol phosphate dehydratase, which converts the substrate imidazole glycerol phosphate to imidazole acetyl phosphate. Basically, the name is dehydratase, or this is also known as HIS-B. And this dehydratase means one water molecule from the substrate is taken out so that it becomes, imidazole acetyl phosphate. And this enzyme has been a traditional drug target for developing herbicide because this enzyme is present in plant, present in lower eukaryotes, present in bacteria, but absent in humans. So there has been a lot of work on targeting this enzyme. In fact, as recently a group has shown, if you block HIS-B, a fungal pathogen known as Aspergillus fumigatus cannot make eye infection. Okay, so these enzymes are very, very important drug target, but nobody has seen this or studied this enzyme from MTB so far. So as a crystallographer, the first thing to do is purify the enzyme to a degree of homogeneity as high as possible and crystallize them. And in this case, crystallizing malaria or TB proteins are always difficult because of the various factors. But we are very lucky. When I was a postdoc with Professor Michael James, that time Bill Jacob was developing a overexpression system of Mycobacterium smegmatis, where we can very easily overexpress Mycobacterium tuberculosis protein in MSMEC. So I got that strain from him and set up standardized in my laboratory. After that, our purification and overexpression processes were relatively easy. So we purified this enzyme, HIS-B. You can see it is very, very highly purified enzyme, and it exists as a 24 more in solution. As you can see, the 20 kilodalton is the molecular weight of the native protein, and you can see the molecular weight, as we see from a DLS experiment, is around 400 kilodalton, and also the diameter is around 14 angstrom, 140 angstrom. And as a crystallographer, it's always tempting to, before you go for the structure solution, first you characterize the crystal. That's why Professor Bijan used to teach us that never jump into technology. First understand the basics, then you use the technology, then in both way you are advantage. You know what you are doing and you will be able to do it faster. Okay. And this molecule has a high tendency to crystallize. It crystallizes in 3 crystal forms. This is a cubic form, this is a tetragonal form, and this is a monoclinic form. And if you can see also from there was a poster yesterday I saw, I was asking about the asymmetric unit. You can see in the asymmetric unit content, there are a lot of symmetry. There are a lot of symmetry, and this is, you can see from the self rotation discovered by Professor Michael Russman, and from the crystal, you can see it has a lot of symmetry. Because then when you solve the structure, indeed it's a 24-meric molecule. It's indeed 24 molecule, and each monomer is itself is a symmetric molecule. You can see each monomer is made up of a 4-helix bundle sandwiched between 2 parallel beta sheets. Okay, and this monomer, it's not working. Yeah, this monomer is arranged in a 4-3-2 manner to form this biological complex molecule. Okay, and this is not a crystallographic artifact. We did the cryo-EM structure with Vinod, it's 24 more, and there are other groups also, various parts of the world, they also work on this enzyme. That's also a 24 more. Okay. This is how the 24 more is arranged. Okay, it's a really very nice complex molecule. It's like a football. And on the football, you have 24 molecules are setting in a periodic manner, not randomly, periodic, with 4-3-2 symmetry. Okay, and as a crystallographer, if you have one molecule in the asymmetric unit, if you apply 4-fold symmetry, you get the first 4, then apply 3 to symmetry, you get 12, from 4 to 12, then 2-fold symmetry, you get 12 to 24. Okay, and this is how this molecule and all the substrate you are seeing, they are all sitting on the surface of this molecule, and there are 24 identical substrate binding sites. 24. Okay, Professor Raghavan Varadarajan from MBU wants to collaborate with us because he wants to use this model to present the antigen. He thinks this could be a very good model for efficient presentation of antigen. And this is only, I have taken 4 molecules from the top. So you can see, it's basically a hollow sphere. It's a hollow sphere, and 24 molecules are sitting arranged on the surface of the sphere in a 4-3-2 symmetric manner. Okay, and also see here, the active site here made up of from 3 molecules. You can see molecule A. This is not working. Can I get a better one? So molecule A, molecule B, and molecule C. Look at this. The active site is made up of from 3 molecules, 3 protomers, 3 monomers. But the molecule is symmetric. Active site is here is asymmetric. Those who can see the symmetry, you can see here. Active site is asymmetric. The molecule is asymmetric. That means it has to take a closed structure. Okay. Yeah. And we also characterize the active site by soaking the substrate imidazole glycerol phosphate. And here you can see the electron density. Electron density means when you take a X-ray picture of a crystal, it's the electrons, they interact with the electromagnetic wave X-ray. And when the diffracted beam carries the signature of the diffractor. That's why when you say electron density means basically these were the electrons, they diffracted the X-ray, and from there when you deconvolute, you will get to see the substrate. So here you can see each and every atom of the substrate is very clearly well defined. Okay, meaning that this is the real substrate binding site, and you can map the interaction to a high degree of accuracy. And you see here in each monomer there are 2 manganese holding the imidazole ring of the substrate, and other part of the substrates are interacting with the other part of the molecule. You can see molecule A, molecule B, molecule C, and here I have shown the interactions. So a high resolution accurate structure has led to not only understanding the enzyme fold better, but also it enabled us to map the interaction between the substrate and the enzyme, and that is the basis of drug discovery. Those who do structure-based drug discovery, if you have a good structure, accurate structure, the active site is very, very well defined, then you take that model, then you do the subsequent drug discovery. Okay, and as I said, this is the electrostatic potential surface of the molecule. It has 24 active sites. All 24 molecule substrates are sitting on the surface. Okay, and you can see here the active site is largely, you know, wherever the imidazole ring there is a relatively negatively charged, and wherever the phosphate group you have relatively positive. So there is a you know charge complementarity between the interaction between the substrate and the enzyme. Okay, this structure is amazingly conserved, and now that this is what my main point is. Now taking this high resolution structure, and we know that this is already been a drug target for herbicide, so we know the compound, there's some library existing. So we took some of the scaffold from that library and made a huge library of around 2000, and in every you know docking we very, very carefully analyze the interaction, and you can see some of the molecules that you have found, they indeed show the specific binding with the enzymes, and this is the Fourier map, different Fourier map, assuming or giving us the confidence that the molecule that we had obtained through small molecule library screening, you see these molecules indeed bind to the enzyme with very, very high specificity. You can see from the electron density. Okay, very, very. So this is, as a crystallographer, this is the first thing to do, that you design one inhibitor through structure and whether this inhibitor indeed binds to the structure. You do the experimental crystal structure, and see that indeed you see this binding up. So this is the first stage. Then you go to the next step, that you see compare the inhibitor versus the substrate and see that they are binding. And we have done the experiment in MTB infect macrophage. You can see with respect to isoniazid, although our compounds are need to be improved, but you see substantial killing of MTB through this compound in macrophage. And also we saw that in animal model, recently we have obtained this data. We have a compound which shows good activity. We said structural SF2 in both in spleen as well as in lungs. And we have now a lot of compounds coming from Spiropharm Manesar. We have a very good collaboration, and now we are getting at least half dozen of molecule. And we have also done the toxicity both in macrophage and also at animal stage. So we are thinking that by blocking the His pathway, certainly there is a new way forward. Thank you.
Please wrap up. Oh. Join us. Our fifth speaker in this session is Dr. Krishnan Gopal Thakur, who's a principal investigator at CSIR IMTech. He has over two decades of experience with molecular biophysics and X-ray crystallography. His team has targeted over 100 bacterial, viral, and eukaryotic proteins. Since the COVID pandemic, he's worked actively to make resources against for the interventions against SARS-CoV-2, and he's worked extensively on VAPC and other key MTB proteins to study physiology and virulence of MTB. He's received multiple honors and published extensively on pathogens like SARS-CoV-2, MTB, and Staph aureus. Welcome, Dr. Gopal.
A warm good morning to everyone. And I thank Bill for such a nice introduction. And I also take this opportunity to thank organizers to provide me an opportunity to present some work that we have done in on TB proteins. So today I'll be describing a project that we have been working on, TA systems from tuberculosis. Just to give you a glimpse of what kind of work we do in our lab, this is for the students. So my lab works in the area of structural biology, and we determine three-dimensional structures of proteins and protein-protein complexes to understand their molecular functions. And I thank Biswal for giving a nice introduction about crystallography as a subject. So I don't need to explain too much about it, but yeah, so my lab works on various aspects of biology. And we use structural biology as key tools to understand the functions. Last 2 years, 3 years, we have been actively working on the discovery of antivirals and also developing molecules for combating AMR. But today's work, I will be focusing on the studies that we have done on toxin-antitoxin systems from Mycobacterium tuberculosis. So when we talk about toxins, they were discovered quite late in 1980s, but of late in a recent decade or so, they have gained a lot of importance in studying their roles in physiology, in bacterial physiology, competition, and virulence. So if I can classify these toxins, they can be classified into two broad categories. One of them is the ones which stay within the cell, we call it cytosolic. And there is another class of toxins which can be presented on the cells. They are extracellular, they are exported out of the cell, and they are actually involved in kin selection or competition. So they kind of shape the niche where the bacteria lives. So it's basically interbacterial competition that they take care of. And the cytosolic ones are the ones which are localized in the cytosols. They are actually molecular breaks that inhibit growth of the bacteria, and which is usually helpful in adaptation or virulence or persistence. So why we are so interested in these toxins is basically they are providing us an opportunity to understand how bacteria can apply brakes at different stages of growth and the kind of variety of mechanisms that they utilize to perform these functions. And these toxins are widely present in bacteria. And different types have been classified so far. We know 8 different types of these TS systems. And toxins are usually proteins, and antitoxins can be either protein or RNA molecules. So, so these toxins are actually targeting several molecular targets in a cell. They basically work on very fundamental biological processes, including DNA replication, protein translation, even membrane potential. So, so the toxins that we have been working so far are either targeting DNA as a molecular target or RNA. So when we look at the distribution of these TS systems in bacteria, we see M. tuberculosis has unusually very high number of these TA modules in their genomes. And this is true for the pathogenic M. tuberculosis or Mycobacterium microti and other Mycobacterium pathogens. But in the close relatives like Smegmatis have very less number. I mean, total 5, in total 5, but then if we talk about Vapsi family, it's just 1 member. So that actually prompted us to look into these systems and why they are so abundant in a pathogen. So this is just an overview of different kinds of toxins that are there in M. tuberculosis, and especially the VABC family, which is virulence-associated protein B and C, is highly abundant. So coming to this aspect, so we started working on VABC family of proteins. And the first thing to understand was, even though they are in the genome, how many of them are really functional? So in Ramandeep's lab, they characterized they cloned all these genes and identified which one of those are actually active, because for our structural and functional studies, later on, we wanted to see which one are actually functional in bacteria. And we selected around 13 such toxins, which are either having a moderate growth inhibition or they had no, and they had a very strong, significant growth inhibition. So among these, for structural functional studies, we took 13 of these, and some of them we failed to clone, or not clone, basically to get them in soluble fraction. We see no protein expression, or in some cases, some of them we were successful in producing proteins. So by nature, if you talk about toxin, it means growth inhibition. So if you take heterologous expression system like E. coli, it generally means it will the cells will not grow. So it was a challenge to produce these proteins for our studies. So initially we worked on VABC20, which was shown to cleave SRL loop in ribosomes. And then there are several of these TA systems where molecular targets have been identified. But we focused on VABC11 because no structural details were available. And we knew the molecular target of that toxin. And so we took forward this study. And it was also a highly toxic protein. So we have shown that this VABC11, if you overexpress VABC toxin, it actually, VABC toxin, it actually causes growth inhibition. And we also showed that this toxin is actually a metal ion dependent tRNAs. Okay, it's very light. So you look at it, this is in the presence of magnesium, it cleaves the tRNA, but if you add manganese, it cleaves even more. And we then also did RNA-seq analysis and identified that when you overexpress VABC11 in a host, there are almost 700 genes that are differentially expressed, where 400 of them gets upregulated, and I think I'll use this cursor, and 281 gets downregulated. In Raman's lab, they also showed that if you make a knockout, so there is basically the host loses its ability to grow in vivo, and almost 2 to 3 log difference is seen in lungs and in the spleen tissue when you use the knockout, but when you complement, it behaves like a wild type. So this data actually suggested that this TA module is important for in vivo survival of the pathogen, that is M. tuberculosis. And comparative gene analysis also suggested that most of the genes that are actually downregulated are involved in ATP synthesis, energy metabolism, and also ribosome, which is protein translation. So which actually correlates with the observation that it slows down the growth of bacteria. So we then determined the crystal structure of this TA complex, which is a heterooctamery complex. And the role of antitoxin basically is to inhibit toxin, and also it also binds its own promoter. So it kind of inhibits its transcription as well. So it works 2 ways, the antitoxin part. So this was the structure. And then since there is no crystal structure for any crystal or any three-dimensional structure reported for toxin RNA complex, so we just did a modeling study, and we somehow we got one of the poses where we thought this is basically an anticodon region where an RNA is cleaved, and this is how it was docked. And there are, you can see, positively charged residues which were lining this protein, and we thought they may be involved in the binding of tRNA. And when we did BLI-based studies, we see that the tRNA, this substrate trapping mutant that we created, binds with nanomolar range affinity. And when you actually make these mutants on these arginine mutants, we see that when you mutate arginine 94 to glutamate, it actually loses its activity. So we showed that this loss is not due to other reason, but it loses its ability to bind tRNA. So, and then since we had a crystal structure, we wanted to design some inhibitors. And the easiest thing to do was to take antitoxin structure, make some peptides, and see if they can inhibit the reaction. So we took this antitoxin bound structure as a template. And this is the antitoxin sequence. We made 4 different peptides, overlapping peptides. And we now use the dose-dependent assays to see whether toxin activity can be inhibited. And we show all the peptides that we designed actually were able to inhibit inhibit toxin in a dose-dependent manner. So this was just an idea to get that these proteins are druggable, and if we can make small molecules, they can be targeted, basically. So briefly, so we were able to successfully determine the structure of this complex, and we have shown it's in vivo essential. And this is very important aspect, because if you're targeting any protein or a target in a bacteria, it should be essential for its growth, either in vitro or in vivo, basically. So what we propose from this study is basically a model where we say these WebBC systems are, especially the 11 and others, when you have normal growth conditions, they are not activated. Or so under when you actually design an inhibitor for this molecule, it kind of loses the ability to adapt in the cell. So it cannot change its transcriptome. So probably the host is the pathogen is not able to adapt in vivo conditions and is not able to survive well. Whereas when you have a normal expression of these toxins, probably some of these, what we have seen in our studies, it leads to a change in transcriptomics, which helps bacteria adapt better in vivo condition. Basically, it helps in slowing down its growth and probably also help in adapting under that stress conditions. So now moving on to another system which we have been working now. This is an unpublished story. So this is a DART-EG system in M. tuberculosis. We were actually interested in characterizing essential proteins from TB. We had targeted several of these. And one of them turned out to be antitoxin of this protein, which is DART-EG TS system, which was discovered in 2016. And we were working on the antitoxin part, which is actually essential gene, which we didn't know the function at that time. So DART-EG system is a new antitoxin system or a protein identified in a bacteria, which can actually do ADP ribosyl transferase activity on a single-stranded DNA. So this is the kind of, it is already known in eukaryotic systems, but this is the first report from bacterial system. So DART-EG is a toxin-antitoxin module, and it does ADP ribosyl it transfers ADP ribosyl on DNA, and the antitoxin was known to remove that modification from DNA. So this was the activity of this toxin. And our initial study, like as in case of other toxins as well, some of them, we could never clone this gene. Even if we clone antitoxin, overexpress antitoxin, still we are not able to get this toxin cloned in its active form. So this is a highly toxic protein, basically. So we made an active site mutant to do biophysical and structural studies. So we have shown that the role of, so this DART-G is made up of 2 domains, N-terminal and C-terminal domain, where N-terminal domain has an enzymatic function where it does removal of ADP ribosyl from the modified DNA, and DART-G C-term domain was not characterized. So we knew that DART-EG forms a complex. So we made constructs and we identified that DART-G actually binds physically to DART-G as well. So this is the pull down that we have done with DART-G overexpressing cells. And we have characterized its oligomeric state where DART is a monomer and DART-G forms a 1:1 stoichiometry complex using analytical ultracentrifuge techniques. We succeeded in crystallizing this complex. And while, just last year, a structure came from Thermus aquaticus homologue of DART, where they have shown how single-stranded DNA binds, how DART binds single-stranded DNA and does the modification. So we determined crystal structure of DART bound to the DART-G C-terminal domain, which is actually a protein antitoxin complex, toxin-antitoxin complex. And with this structure, we were able to assign now different functions. So DART is this from TB basically, and you have a DART-G macro domain which codes for ADP ribose, removal of ADP ribose from DNA, and this C-terminal domain which physically binds DART and inactivates this toxin. So interestingly, this DART-G, there is no structural homologue in PDB. So, but when we take small pieces of this protein, small domains, there are two domains basically, they mimic DNA binding structures. So this is very interesting. So the structurally, it shows very high structural homology with DNA binding structures, but if you look at the electrostatic potential differences, you see the DNA binding proteins will have a positive charge while this protein has negative charge. So it will not have any DNA binding property. So this was interesting to see how domains evolved from DNA binding proteins to protein-protein binding domains. So this interface is quite intense. There are numerous protein-protein interactions which stabilizes this complex. And you see these are three different loops which are involved in binding toxin and neutralizing. This is a very tight complex that we have observed. And so this was the interesting part, how DART gets inhibited by DART-G. So you look at this crystal structure here. So what we have shown here is superposition of DART from Thermus aquaticus, where this ball and stick is the DNA. And this is DART-G, where it exactly binds at the toxin, at the DNA binding pocket of DART-G, basically. And we also observed a mimicry where this protein DART-G actually mimics a single-stranded DNA structure kind of thing. So you see this is a highly negatively charged, DNA is highly negatively charged, and this is our DART-G binding region, which also mimics like a structure of DNA, basically. So we identified residues which are key involved in binding DART, and we made those mutants now, and we show that the three residues, this negatively charged residues, which is glutamate, aspartic acid, and another glutamate residue, if you mutate them to arginine, positively charged residue, the protein loses its ability to bind DART-G. And if you make double mutant and triple mutants, then completely the ability is lost. Same is shown here. So when we take a DART, we have done these studies basically with the DNA, single-stranded DNA, where when we take a toxin alone, it binds DNA. But when we take a DART-G complex, there is no binding with the single-stranded DNA. So from our studies, we have concluded that DART actually employs two-layered mechanism to inhibit toxin. So first is basically mediated by the macro domain, where it enzymatically reverses ADP ribosyl modification on DNA. And the second mechanism where DART actually blocks activity by sequestering DART. So this kind of a mechanism is not known in any TA system reported so far. And we believe DART is such a potent toxin that it probably has evolved this mechanism to keep it under check. So the role of toxins in bacteriophage or phage defense is emerging. And DART, since it modifies DNA, so it has also been shown that it actually, this is the model for DART working basically. So you have a DART GTA system where DART is a toxin part. It is blocked, inhibited by two mechanisms of DART-G. And another thing is that, yes, I'm almost done. Coming to the last slide. So it actually, if DART-G ADP ribosylates DNA, at least macro domain can remove that. And also during, if it is expressed, DART-G is also kept under check by binding to its antitoxin, which is DART. Okay. So, but what role it plays in bacterial physiology? One of the roles which has been established is that whenever there is a phage infection, this DART can actually modify the phage DNA, so it can stop its replication. So it's kind of a mechanism that bacteria uses for phage defense. And what role it has in other physiological aspects of TB, that we are not sure yet. I mean, that needs to be investigated. So to summarize, we have established that DART is inhibited by DARG by two distinct mechanisms, and we have determined a crystal structure of DARTG, which can be used for designing drugs and all. So this is the last slide I want to show, that toxins actually play a very crucial role in stress adaptation, virulence, and competition in bacteria. And in my lab, we have been working on two classes of those toxins, one which are cytosolic and the one which are expressed on the cell surface, which plays role in growth, in shaping the communities, basically. So another last thing I want to share with all of you is that TA systems are basically molecular brakes in a bacteria, and you can also call them as suicidal bags, okay? And our studies also suggest they are druggable drug targets. So basically, if we have to design some new molecules, new drugs, and we have these enzymes which are anyway keeping bacteria in check, and especially the ones like DART, which is highly potent toxins. And if you can design inhibitors for disrupting these protein-protein complexes, I'm sure that we can have very long-lasting drugs, which once activated can stay in a cell for longer time, and you don't need to give more and more doses of such drugs, basically. So toxin biology also offers you an opportunity to understand new molecular mechanisms like what I just shared with you, and to understand how bacteria adapts under stress conditions or provide phage defense, or how it regulates its other physiological processes. And this discovery also came from characterization of one of the hypothetical genes from TB. So definitely there are almost 1/3 the genome still not characterized well. So a lot of opportunities for understanding biology. So I would like to thank our director, current director, and past directors for supporting my research and providing us fantastic infrastructure. I mean, this work would not have been possible without active collaboration with Ramandeep Singh and funding from different agencies. And actual work was done by what I presented today by Amar, Japlin, Latika, and some contribution from Pushpanjali and Mahesh. Charan and Rishita are current lab members who are working in this TA systems. And Sony is the guy who initiated work on toxin projects in my lab. He worked on different toxins. And Latika is still working in this area in the lab. So this is my group in tech, and I thank you all for your attention.
So thank you, Chris. And so we have about 8 minutes for Q&A and if you could just,
I have just two small questions. One from Dr. Bachitra. As you said, humans are not able to produce certain amino acids like phenylalanine, but it is being found that in tuberculosis, the level of phenylalanine is increased substantially. So do you think any mechanism by which TB contributes to this level?
I will not be able to provide insights into phenylalanine.
Okay.
But as far as these amino acids are concerned,
yeah.
To begin with, TB takes it from the host before the adaptive immune response is on. So there are transporters for amino acids. Before the adaptive immune system is on, they take from the host. But the moment host realizes after the adaptive immune system is on through IFN gamma, it upregulates enzymes that could sequester the extra amino acids in the cell. So that compels MTB to make its own.
No, okay. But you said humans are not able to make these.
So we take from diets.
Okay, from diets.
We take from diets.
Okay.
All the 9 amino acids.
Yep.
Even I would say that arginine, it's not an essential amino acid, but the body makes so little. It's also considered as essential amino. Other 9 amino acids we take from the diets. We do not have the de novo biosynthesis pathway to make it.
Thank you. Yeah, thank you. And my next, just small questions, like, do you see, not from you, but from toxin and antitoxin systems, basically, do you see any regulation of human microbiome through these toxins? Can these toxins modulate microbiome? Because we find microbiome is playing a crucial role.
Yeah, so I talked about another family of toxins which are exported on the cell surface.
Yeah.
So they have been known to actually shape microbial communities.
Yes.
Okay, including some of the pathogens. It has been shown basically when they have to compete in existing niche areas where there is already commensals there. So they need to kill those commensals to establish infection. So that's the time when these pathogens actually are expressing such toxin, and they were discovered initially in EUPC, which is the Uropathogenic E. coli. They were first discovered in those bacteria. So they are really important for establishing infection also and shaping shaping communities.
Vinay, is there any variability known about the susceptibility or resistance to different drugs by various TB strains, such as S30-RV or Beijing, or so on and so forth?
Some of the Beijing actually develops a resistance faster, and that's kind of known. Like if you have a Beijing actually has a tendency to develop resistant fast is known. But with respect to the variability, the kind of depth study needs to be done to figure out what exactly is contributing. In fact, one of the reasons why Beijing may be developing is it has certain mutations in one of the repair pathways. It is not MUT5, but in another repair pathway, it has a mutation. So I think that needs to be looked at it much more. Yeah. So that much is at this point of time we are aware.
Okay, hi. I have a question from Ramandeep, sir. Sir, how are exopolyphosphatase levels regulated within MTB? And my second question is, what about the redundancy between the PKK1 and 2? In the absence of PKK1, can PKK2 take over?
Your first question was not clear to me, but for the second question, there is no redundancy because when we mutated PPK1, the polyp synthesis was really negligible. So there was no redundancy between PPK1 and PPK2 enzyme. And in terms of in PPX for the exopolyphosphatases, we have data which we did not show for the single mutant strain, but we showed data that they work in a cumulative manner. But I don't think much has been done for the regulation on the regulation of exopolyphosphatases.
Okay, thank you.
Thanks, Chair. Two quick questions. One to Vinay, one to Ramandeep. Vinay, in case of MUT2I, you had done a genomic analysis. Did you see any mutation for collateral sensitivity? And also about UARB that is mentioned in your paper. So I was just wondering how UARB mutant is in terms of the efficiency compared to MUT2I mutant for drug resistance.
Collateral sensitivity to
other drugs.
See, the thing is, to start with, the study was designed in such a way that we were looking for mutations that are found in the multi-drug resistant strains. So they were already drug resistant, and they also had mutations in certain, some mutations were like around 9 out of, 9%, I think 9 of them, we found mutations in one DNA repair gene or other. But they were already resistant. So the collateral sensitivity we did not look at because it's the data that was available is what we mined. I mean, we did not take other way around.
Yeah, quick question, Ramandeep. You said about accumulation of neutral lipids in THP cells, if I'm not incorrect.
Yeah.
It's only the neutral lipids or any other pathway was enhanced, like parachutonic pathway or anything?
So we had just checked for neutral lipids based on the host RNA-seq data. So I can't comment on the other lipids.
In the interest of time, I'll ask those with further questions to catch our esteemed speakers at the lunch hour. So I want to thank all 5 of you. and very stimulating session. Thank you very much.
I thank all the speakers for shedding light on different facets of TB drug discovery and challenges it throws at us. I request the session chairs to kindly felicitate the speakers. Professor Bill Bishai to Dr. Vijay Nandikuri. Professor Bill Bishai to Dr. Ramandeep Singh. Professor Gopal to Dr. Biswal. Professor Gopal to Dr. Gopal Thakur. I request Dr. Guru Prasad Medigeshi to kindly felicitate the session chairs. Professor Bishai. Professor Gopal. I thank the session chairs and the speakers for the smooth conduction of session 7. We now proceed to session 8.
We now move to session 8 and continue with 5 more talks on TB drug discovery and development. I warmly welcome Professor Debashish Mohante and Professor Vinay Nandikuri to chair this session. Dr. Mohante is currently the director of National Institute of Immunology and has expertise in computational biophysics and bioinformatics. In recognition of his contribution to the field of biophysics, he has received several prestigious awards, including the National Biosciences Award and the Rajiv Goyal Young Scientist Prize in Life Science. Dr. Vinay Kumar Nandikuri is currently the director of Center for Cellular and Molecular Biology, Hyderabad. He is well known in TB community for his path-breaking studies on the kinase-mediated signaling networks. He is the recipient of National Biosciences Award and elected fellow of National Academy of Sciences, India, and a recipient of NASI Scopus Young Scientist Award. We have 5 talks in session 8, each for 20 minutes. Following the talks, there will be brief Q&A session with all 5 speakers. I request the audience to hold their questions until then. I urge the speakers to please stick to their time. The buzzer will ring at 18 minutes. I request the speaker after their talk to be seated on the dais for the Q&A session. I now request the session chairs to kindly lead the session 8.
Thank you. And in the interest of time, we should quickly move. And the first speaker is Dr. R.K. Sandil from the Foundation for Neglected Disease Research. And he has rich experience in TB drug discovery at AstraZeneca for more than 20 years. He is co-discoverer of several candidate drugs led through clinical trials. In the interest of time, I will make the introduction brief and welcome him to start his talk so that we save some time. Thank you.
Good morning. Thank you, Chairs. Thank you very much for the introduction. And in the interest of time, let's get started. And the title of my talk is right here. That is on challenges in drug discovery and development. And I come from a industry, so I will give you a little bit of industry perspective of drug discovery and development. So first of all, let me thank our donors, granting agencies, funding partners, and especially AstraZeneca, Gates Medical Research Institute, Global Alliance for TB, the several governments, patients, our laboratory animals who do a lot of the job, and our employees, without which it would have not been possible for us to sustain as an organization, which is called Foundation for Neglected Disease Research today. My presentation actually has two parts. First is on basically in order to address the occasion of the World TB Day and the NTB commitments. So it's on the current TB pipeline and some discussion around that. And the second will be a story of a drug that we have taken from lab to the clinic today. So needless to say, lots of discussions have happened since yesterday that we have lots of drugs for the TB, of course, but still the TB remains a major problem with 1.6 million deaths every year. And if you come to India, we are one of the biggest players with 26% of the global TB, probably in India. The question is, why are we failing? Why are we not able to tame the TB? And if you look at the global portfolio today, how good it or bad it looks like, and are there new drugs available which will be compatible to combination therapy and so on. And finally, we ask the question, is it good enough to meet the target of NTB 2030? The second part of my talk will be on discovery of a TBS 7371, which was discovered by us while I was working at AstraZeneca. I'm one of the co-inventors of this drug. And finally, it's now being developed with certain other partners. I will talk in more details. And the next slide is, we know that TB is an old disease. And if you go back to history, we know that it was referred to as white plague. And a lot of history has been discussed, but let me briefly go through some of the interesting findings that happened in the history. Was 1867 was the first official international meeting of the experts where they talked about how to control this white plague, that is the TB. And then soon after that, Robert Koch described M. tuberculosis as a causative agent, and then rest is the history. And after that, the next important event happened in history was the creation of a body called the International Union Against Tuberculosis, which is one of the very pioneer bodies for TB control. And soon after that, we had the BCG as one of the first preventive measures, even before the drugs started being discovered. And finally, after World War II, is basically the era of the drug discovery. And you have seen, you have heard today morning from Dr. Tanjore Balganesh and many others that how the drug discovery have emerged from very primitive to combination therapies and so on. And the first first drug that was discovered was in 1943, streptomycin. Unfortunately, it was very much prone to resistance, and it started failing very soon. And then came the concept of combination therapy, and it was first in fact, streptomycin was discovered before the inception of WHO, which was constituted in 1946. So that old the TB discovery is. And the first combination therapy was shown by John Crofton in 1950 that combination therapy could successfully treat the TB patients. And in India, equally, almost at the same time, it was shown by Wallace Fox that TB can be treated at home, and that changes the whole scenario. So basically, we have discussed about all these things, and today we are at 2015 when we are talking about 2015, where the NTB strategy was launched, and the target remained 2050. And we are glad to see here the ambitious commitment of the government of India, NETP, is 2025. So that I think this describes our or the India government commitment towards eradication of TB. Now there are many drugs for the TB, but the TB is still there. If you look at the first half of the slide, from 1940 to 90, almost about 70, is called the golden era. That is where almost half a dozen TB drugs were, developed for the specific drugs for TB. And after that, there's something called innovation gap. But I believe it is not an innovation gap; it is the change of priorities. The change of priority happened to other bacterial infections, and TB probably lagged behind. So a lot of antibacterials were developed, but they were not TB drugs per se. Of course, many of them are used today for treatment of TB. And finally, around 2000, and since then till today, we have new drugs coming up, like we have a PA24, which is now called Pretomanid. We have Bedaquiline, Delamanid, and few more on the queue. So the bottom line is, I think one of the greatest revolution, I would say, as Dr. Kaul mentioned yesterday in a great detail, was discovery of Bedaquiline after 60, 70 years of a gap. There was no drug. Unfortunately, this drug is not widely used in a way that it has some safety signal. So it has a black box warning signal and all that, but it doesn't matter. It has still changed the way TB, or especially the MDR TB, is treated today. Now, question is how, if we have to discover a drug, how a TB drug should look like, and that is something called TPP. Again, and Dr. Bal Ganesh, who happens to be a mentor as well, I started my postdoc career in AstraZeneca with him, alluded about that is nothing but it's the profile of a drug, how should it look like. And that's what we do. So on the left-hand side is the target product profile, how a drug, what are the features that you would like to include in the drug, and right-hand side is what characteristics drug should have. Certainly, everybody will say we need to treat MDR, XDR, TB, and on the right-hand side, you will see it will happen only if you have a drug with a novel chemical entity with a novel target mechanism. It is bound to work on all kind of MDR strains. And shortening duration of therapy remains a challenge, but that will happen if you can kill bacteria faster. So your drug has to be bactericidal. Lowering dosing frequency needs a great pharmacokinetics and a long half-life. And the couple other properties is the reducing the pill burden. Today, if you look at the HREZ treatment, the 6-drug combination, it is about 2.5 grams of drug, or a chemical, I would say, that a patient is taking every day. And that's a problem. And if it is MDR TB, it may be probably more than that quantity, because the drugs increase from 4 to 6 or 8 or 10. So it is important then we have more potent and more selective drugs so that the drug burden could be reduced. And drug-drug interaction, because TB, the name of the game is combination therapy. And if a drug has to enter into existing combination, there's not going to be a way to treat it with a single drug. So the drug has to have minimum possible drug-drug interaction with the existing regimen, and also with those drugs which are commonly used by patients, for example, in HIV patients or diabetics, the comorbidities. So these are the kind of characteristics which define the TPP of a drug before you start. This is a review article which we wrote again with under the mentorship of Dr. Tanjol Bal Ganesh a couple of years back, which describes, and I will what it takes to make a TB drug, and I will not go through except for some of the points in the bottom, close to bottom. For example, there are so many new targets that have been described, the DPRE1 target of energy metabolism, like target of bedaquiline and electron transport system, and so on. And new combinations are really emerging, and you will see in the next few slides how it is going to shorten the duration of therapy of the TB patients. And finally, a bit on immunomodulators. It appears that there are several immunomodulators on the way, and that may be one way to treat TB probably in the near future, especially in combination with the existing therapies. And now if we look into the the improvements, the significant improvements have happened in the TB space. Infrastructure of TB has improved. BSL-3s, which used to be very uncommon, are very common today. There are probably dozens and dozens in India. Access to funding is better. There's a chemical repertoire, or a lot of chemical libraries are available, so you can start your screening with those chemical entities. Animal models are there. We have a lot of transgenics, gene knockouts, and everything, reporter systems. High throughput screening technology have grown from 96 well to even 384 well formats. Phenotypic screens are widely used. and a lot of target biology understanding, including AI, that has been inculcated into the TB research today. However, the challenges remain. The biggest challenge probably, as Dr. Soumya Swaminathan also mentioned yesterday, is the granuloma, which is the holy grail. Though we understand quite a bit about granuloma and the pathogen of granuloma, but there are many more things which we really don't understand. And another issue I see is the preclinical translation. And this means that there are a lot of the probability of getting compounds with MIC and MBC are quite decent, but when it comes to animal efficacy, more or less all of them fail. And that is the truth. And being in an industry, we have seen this day in and day out. So that's the translation issue. We don't really understand what is that driver, which will determine drug is going to work in vivo or not. And finally, the drug discovery path is really long. And just to allude onto that how drug discovery path is long, there is an example from a Nature paper, which was 10 years old, where they plotted the number of the drugs that are discovered, and you see the slopy graph from left to right. These are the number of drugs that were produced, drugs not TB, any drug for that matter, over a period of time from 1950 to 2010, over 6 years of time. And you see the cost, the $1 billion per drug cost, which was about 35 drugs in the 1950s. Today, it is probably one drug in $1 billion or less than that. So the cost has tremendously increased. And they give a very good analogy with a law, physics law called Moore's law, which is a law of exponential growth, which means that if you have, for example, cars, if you have a technology, you have a pieces A, B, C, D on the left-hand side, you assemble and you get a car. Very quickly, you can turn it around. You increase the left-hand side and you increase the number of the cars that you produce. But that is not the case in drug discovery. The drug discovery is not like making cars. Now, why? Because we are dealing with biology of the pathogen and biology of the human host. Another important thing, the drug is not a replica. It's not a car, another car in another car. The replica is not a replica, but the new drug has to be differentiated. It has to be somehow, some way better than the previous drugs. And that's how the drug discovery system works. And finally, it's like being better than Beatles. So if you have to make a famous music, you probably have a standard there, which is Beatles. So you have to beat the Beatles. And unfortunately, or fortunately, whatever it is, today's blockbuster is going to be tomorrow generic. So the companies will not make more money on that after the patent is over. Well, to cut the long story short, another interesting example that I put together to show why TB drug discovery is long and is it really long? And the answer is yes. I collected data on the 5 recent drugs, the bedaquiline, predominant delamanate, and some of the BTZ and the TBA73, our compound. And then based on a simple criteria, when the first publication was made on this, which was, so the first publication, for example, is in 2005. And then when this drug came to market, let's take example of bedaquiline. It took very short time, basically just about 10 years, because there were no drugs, so there were a lot of push. And this drug did somehow much better than others. But all others drugs are taking, let's say, the second, the predominant is about 22 years, delamanate is 15 years, and so on. And in fact, if I talk about our own compound, which is coming, TBA7371, we are already at 11th year, and it is just completed the phase 2A clinical trial. So yet few more years to go. So certainly it is going to be 15 to 20 years for all drugs. And very quickly on this occasion, I think if we have a look at the global portfolio of the TB, thanks to the group on the right-hand side in the bottom called the Working Group on TB. This is a group which basically keeps track of all the drugs that are coming in, and they keep updating this. And another thing you must note is the color combination. So the different colors denote different classes of compounds. For example, the blue colors on the left-hand side here, they are oxazolidinones. I don't think my marker is really working. If you go down in these verticals, you see the number of the drugs in every class. And finally, if you look at the late clinical stage development in the phase 2, there are several drugs already there. And in the phase 3, most of these drugs are being converted into combinations, either with the existing drugs or even totally new combination. And I would like to basically focus on the what is shown in the yellow, the BPAL, which was published recently. This is a ZNIX trial or NIX trial. This is one of the newest combination that has successfully treated TB in 6 months rather than 18 months or 24 months. And the reason for that is that these are all 3 new drugs. BPAL is bedaquiline, pretomanid, and linezolid. And more interestingly, all the 3 drugs are oral drugs. So there is no need to take even a single injection. That's the way I think the TB drug discovery and development and the therapy is revolutionizing today. Of course, this is one of the first hits. There are many more trials going on. It's a long way to go, but things seem to be doing much better. And finally, my view that there is strong drug pipeline today. We're coming closer to NTB commitments, but still a long way to go. And briefly, now I think time will not allow much. I will go through the discovery of our molecule called TBS-7371, which we discovered and has now completed phase 2A clinical trial in South Africa. So this trial is fully funded by Gates Medical, today is funded by Gates Medical Research Institute, and trial has just been completed in South Africa in few of the clinical trial sites. When we started with this program about 11 years back, we did the same thing. We went to TPP, and without rehashing, these are the same features that we wanted to bring into our compound that's orally administered, work on sensitive NDR strains, fit for combination, and so on and so forth. Basically, if I go back to a little bit into the biology of this compound, it's azindole class of compound, and it targets an enzyme called DPRE. The enzyme is DPRE1, right? And what this enzyme DPRE1 and DPRE2, that it basically converts the DPR, which is nothing but decaprenyl phosphorylate beta D ribose unit to begin with. It converts it into DPA, which is the arabinofuranose kind of the end product, which is the basic building block of arabinogalactan and lipoarabinomannan. So if you interfere this pathway, you basically interfere with arabinogalactan and LAM biosynthesis in the cell wall, and that's how this compound works. This target has been validated. In the bottom, you can see there are a couple of other compounds called BTZ and PBTZ. These are benzothioneone class of compounds. Now, a lot of molecular biology and initial, biochemistry has gone into this to show target essentiality. And just to show a very quick example here, on this table on the left-hand side, you see TBS-7371 and another compound which targets the same enzyme is BTZ. And these are the first is shown is the MICs, and then you have the we did it 2 ways. One is the overexpression strain, OE strain, and there were 3 different mutants that we made. If you look at the overexpression, both of these compounds, the MIC elevation happens from 0.7 to 100, and more than 100, and 0.005 to 12. That clearly says that you are hitting in vitro the target, and that's where the target, the MIC is elevating. But on the MIC of the standard drug remain the same. On the other hand, if you look at the target mutants, we have, MIC of for 7371, MIC is 0.7, 0.7, while it shifts with the BTZ, and vice versa is true in the last column where the BTZ MIC is 0.0015, but our compound shows MIC of more than 100. So what it tells is that we are hitting the same target DPRE1 by both the compounds, yet they work by different mechanisms. And we know detailed mechanism that our compound is indeed a non-covalent binder versus the other one is a covalent binder, and many other differentials. Finally, if you look at this chemistry, now you'll be surprised that this thank you. This compound started with a literature-based compound from a Korean group, but unfortunately, what had happened for them, they showed MIC, but there was no MBC, no bactericidal activity, and no animal efficacy. So they dropped it, patent collapsed, and we took from there. There are a couple of smart chemists on our team who took this challenge, converted the compound into a new scaffold, and created a patent space, and we patented this compound, right? The first compound, we brought in MIC and MBC, and you see the enzyme activity is about 14 nanomolar, pretty potent. And the compound in the blue is about almost, I would say, somewhere around 50, 60, after 50, 60 compound synthesize, which is nearly a final compound. But finally, it took about 200 compounds to be made to finally come to a clinical candidate, which went to clinic. And MICs, you can see the profile of this 0.7, DPRE1 inhibition is 5 nanomolar, it's bactericidal, efficacious in animal model. And I'll skip this chemistry strategy slide, how we achieved it. But if you look at some of the top compounds, the profile on the left-hand side, you see what it takes to make a drug. You know, you need physicochemical properties, you need drug metabolism properties, you need absorption, ADME, DMPK, bioavailability, and so many things. And to cut the long story short, you just look at the green, means they are ready to go from our perspective. And there were, of course, a couple of signals, like red ones are the signals which are not good. And one of the signals we had basically PD6 liability, which still went on, but finally it worked fine when we progressed the compound further. And this is the similar data, but shows most of the parameters. The key parameters for the drug discovery were green, and this is just the animal efficacy. So the top one shows the structural relationship, SAR activity between enzyme activity and MIC, which there were several compounds which went hand in hand. Good killing kinetics, time dependent, kill keeps to increase to several logs with time, and a decent pharmacokinetics on the right side bottom panel, which shows the free levels and the total levels. And the interesting thing, which is important for conversion, what we're talking about, how compound translates into in vivo efficacy, is had to have a decent pharmacokinetics. And here you see that our compound, basically the pharmacokinetics is above MIC, this blue line, and that's for about significant amount of time, for about 40% during dosing interval. It is above MIC in the, not only in the plasma, but in the target tissue also, we know, and it translated into efficacy. You see the green, the first blue are the control, and then you have this compound, the dose response to the two compound, and the red one is rifampicin. Finally, we did another study in the acute model. Again, you see compound is highly bactericidal, about 2 log kill, and also we did efficacy in another species in the rat. And again, you see here that the compound.
Sir, since you are running late, can you please?
Thank you. Complete dose response. And finally, based on these properties we achieved, it went for the clinical development. We found in this compound, the systemic toxicology study, there was no genotoxicity, no HERG, no repeat dose toxicity, and CMC challenges. Finally, we went to phase 1. And just 2 more slides, just to show that I don't have really, I'm not going to show any phase 1, phase 2 data, because still under, is happening. But just to show the design, how you do the SAD and MAD phase 1 studies. And the conclusion was, both in SAD and MAD, the compound was well tolerated, PK was dose proportional, and no safety signals or alarms. And finally, this is my last slide, showing the design for the phase 2 clinical trial. It is available on www.clinicaltrial.com. The number is there on the top. And this is a 14-day EBA study, phase 2A, dose escalation controlled randomized trial to evaluate safety, bactericidal activity, and pharmacokinetics. And the trial has been completed successfully. There were no major issues observed. Trial went through, and now fingers crossed. We are waiting for the results around mid of this year. And thank you very much for your attention. And I will be happy to take some questions. Thank you.
To introduce the second speaker, Dr. Mandira Verma. She's a professor in the Department of Microbiology at Vallabhbhai Patel Chasti Institute, University of Delhi. In addition to her academic role, she's also been recognized for her outstanding contribution to the field of microbiology. She has received several prestigious awards and international fellowships. She's interested in research interests include early identification of tuberculosis, and mechanism of drug resistance in patients of Mycobacterium tuberculosis. Please go ahead with the presentation. Thank you.
Thank you, Chairs. Good morning, everyone. And thank you, Dr. Ramandeep and the organizing team for having me here today. It's really an honor and a pleasure to be on this platform. So I'll be talking about, as the title suggests, about efflux pumps and the role in drug resistance. Just a little bit of the history, because it's already been covered, but we know that mankind knows that TB has been there. And even Aristotle said that the patients of consumptives got this disease because of bad air. But it took 1,000 years before Jean-Antoine Villemin proved that TB was contagious. And then, of course, we know the historic date of 24th March 1882, when Robert Koch told the world that Mycobacterium tuberculosis was responsible for this disease. And it was then that mankind realized that TB was not a punishment by God, but a disease that could be prevented and could be cured. It took a little, so then we started this era of sanatoria and treatment with rest and good nutrition. But then it took a little more than 60 years for streptomycin to arrive. And as Dr. Pal Ganesh said in the morning, that resistance to streptomycin arose soon after. So, so much so that tuberculosis is still a major health problem. Now, this figure has been shown multiple times, I think, in this forum. So just to reiterate that 45% of the cases of tuberculosis come from Southeast Asia, and India accounts for 28% of the global TB cases. So when we consider the global distribution of multi-drug resistance in the new TB cases, India has about, it's still 2 to 3% of MDR in new TB cases. Of course, it's much more in the previously treated cases. The good part of this graph here now is that we have information from most parts of the world, which was absent a few years back. So coming to drug resistance, how does it develop? And that's also been discussed. And we know that Mycobacterium tuberculosis, because of its cellular structure, is impermeable to certain drugs. We also know that the majority of the cases of resistance occur because of target mutations or canonical mutations in clinical isolates. And WHO has recently cataloged the mutations that Dr. Camilla also discussed in her talk. So this was in 2021 because of the plethora of mutations coming in because of WGS and NGS data. There was a lot of confusion as to which mutations are actually leading to resistance. So WHO has cataloged the canonical mutations and has also mentioned the mutations that would not lead to resistance. Nevertheless, what I'm trying to show here is that if we look at the right side of the table, we'll see none of these figures total 100. So the best result that's been obtained is for rifampicin, the RPOB gene, where 92 to 95% of the cases we find rifampicin, in rifampicin resistant isolates, we find RPOB mutations. So that means there are alternative mechanisms that would play a role in conferring resistance. And so one of those mechanisms are efflux pumps that Professor Nandikuri also mentioned in his talk. So the efflux pumps, of course, you know that they're translocases, they extrude toxic substances, and they have multiple substrate specificities, and a single drug can induce multiple efflux pumps. So they lead to a low level of resistance to anti-tuberculosis drugs. To any antibiotics, it's been seen in Pseudomonas, Acinetobacter, other organisms also. And these efflux pumps, when they throw out the antibiotics, they are, of course, not 100% efficient. So some of the antibiotic molecules stay inside, like this vagabond molecule here. And this leads to a sublethal concentration inside the cell, which leads to a constant stress to the bacterium. And then that bacterium then develops into a full-blown resistance through mutations. So that is basically the hypothesis of how efflux pumps act. There are some major families of efflux transporters. Just briefly, the ATP binding cassette, which is one of the biggest, and that acts through ATP hydrolysis. Then there are others that act through proton motive force. So coming to the ABC transporters, as I said, there's a large part, the 2.5% of the genome content is by ABC transporters. And a number of ABC transporters have been studied in relation to Mycobacterium tuberculosis, and it's been shown that they've led to or associated with resistance to a variety of drugs, including fluoroquinolones, rifampicin, and macrolides. The major facilitator superfamily has also been studied a lot. And of these, one of the most well-known papers from India is the Jeff A. That was published by Gupta et al. From JALMA. And they showed that Jeff A mutations could lead to tolerance to isoniazid and ethambutol. Now, there have been studies from clinical isolates also, like this one was from China. They studied the 1217 and 1218C genes efflux pumps. Then there were several studies from Iran. These two studies are from the same group. Again, they've compared the expression, the or upregulation of genes in drug susceptible and drug resistant isolates, but they compared it with H37RV, and they basically saw the basal level of efflux pump genes in both susceptible and resistant isolates. In the second paper that was published in 2019, they also studied efflux pump inhibitors. This paper is also from Iran by Shahi et al. And this again looked at 5 genes, and they also studied efflux pump inhibitors, and they saw that the efflux pump inhibitor CCP was more active for rifampicin than for isoniazid. So then we come to our experience about drug resistance in Mycobacterium tuberculosis in North Delhi. So these were the cases. So these were the isolates that we had obtained from patients of, we'd taken new smear positive patients, we'd taken previously treated patients and multi-drug resistant suspects. And we saw that we found a number of MDR isolates, and we picked up those for our study. And we also took up some of the polyresistant and rifampicin resistant isolates. So this was followed up by, we looked for canonical mutations by the sloppy molecular beacon assay and also partly by sequencing. We didn't have enough funds those days for whole genome sequences. So we looked at these genes by the sloppy molecular beacon assay. And for the rifampicin, we took up 108 isolates basically for the sloppy molecular beacon assay. And there were 3 probes. And the most common mutation was seen in 531. That is, we know that's the most common mutation. What is important was that 7% of the cases, we could not detect resistance by the SMB assay. For the isoniazid, again, we looked at the mutations. The most common was 315 in CADG. Again, 9% of the cases, we could not pick up the resistance. For ethambutol, again, EMBB306, this was looked at by the SMB assay and partly by sequencing. Sequencing was done for the 497 codon. And in this case, 36% of the cases, we could not pick up the resistance by this assay. And same for aminoglycosides, looking for RRS, RPSL, and EIS probes. And 20% of the cases, we could not detect the resistance, which again meant that we were missing resistance in a number of, the cause for resistance in a number of isolates, and what could be that cause. So we started looking for efflux pumps, and these are my students, Dr. Garima and Dr. Anshika. And so we looked at the clinical isolates. For this study, we picked up basically multi-drug resistant isolates and drug susceptible isolates. And we selected 10 efflux pump genes. So there were MFS family, ABC family, and the RND family. And these genes were picked up because one of our multi-drug resistant isolate had, we looked at the high confidence mutations, and then we picked up these. So 2 of these genes also were operonic, so we picked up all the genes here. Some of the genes were picked up through the literature, of which RV0842 was also one of the important ones. So first, we looked at the isolates, and because we went through a lot of publications about what sub-inhibitory concentration to give the isolates. So we first established that for isoniazid, half MIC was the best, and for the rest of the drugs, 1/4 of MIC. Then we looked for upregulation under rifampicin stress. Now, these are, of course, all multi-drug resistant isolates. So the most common mutation is, as I said earlier, 531, but there was one isolate with 511 mutation. Now, before this study, we also, we did those, the efflux pump genes, all these efflux pump genes that I'd highlighted, and we found there were 4 of them that were more important. So these extra isolates, we just picked up those 4. So for the 511, as you can see, 3 of those 4 efflux pumps were upregulated. And these were the sensitive isolates. Again, we saw some upregulation in the sensitive isolates as well. So just to summarize for the rifampicin, there was one high level resistant isolate which did not have any efflux pump genes, but the one which had efflux pump gene upregulation had a higher MIC, and there was efflux pump upregulation in the low level resistant isolate also. So then we followed it with isoniazid. Again, we took sensitive strains. Here, the first few rows are the sensitive strains and the resistant strains. There were, in fact, more efflux pump upregulated in sensitive strains than the resistant strains, but there was this one isolate where we did not find a canonical mutation, but there were several efflux pumps that were upregulated. And these 5 efflux pump genes were not upregulated in any of the susceptible. They were only found in the resistant isolate. So this is, again, to summarize what I've already said. So this was the important isolate where there was no canonical mutation that was found. This was then followed up by the ethambutol stress. And again, 3 of my students, Astha, Varsha, and Chanchal, who are doing this work. So for ethambutol, again, this time we could do whole genome sequencing also. So in addition to the upregulation, we also annotated the mutations that were seen, and we could also find out what strains, what lineage the strains belong to. So some of the mutations that we found were more were actually associated with phylogeny, and so were excluded. In fact, one of the RB0194, this mutation here, the 1098 codon, was deleterious, but it was only found in the Beijing isolates. So again, these are the resistant isolates that are highlighted here. The red boxes show the ones that were upregulated. And these are the susceptible isolates. Then these were, 2 of the isolates were East African Indian, and 2 were, the green ones were Central Asian strains. And we see that some of the strains had upregulation of efflux genes. But in this case, again, we see that there were high-level resistant isolates where there was more expression or upregulation of the genes, and the MIC that we saw was higher. In case of the lower level resistant isolates, again, there was upregulation of the genes. And there was this wild type isolate where we saw the upregulation of RV0842, which was also found in other resistant isolates. Then Chanchal and Varsha continued this on kanamycin stress. Here, again, as I had said, that we had looked for RRS and RPSL mutations. So the first rows are for the susceptible strains, and the latter rows are for the resistant strains. Here we see one of the susceptible strain has a lot of upregulation of efflux genes. The strain which had a mutation, resistant strain which had mutation, the canonical mutation also had upregulation of 0842. And there was one wild type strain which had upregulation of 0842. So this is just the same, that RV0842 was found in both. So after this, we, this time for the RV0842, we went in for cloning, and we used PVV expression vector and then electroporated it into H37RV. And when we put up the MIC, we found that for ethambutol, there was no change in the MIC in the overexpressed strain. But in case of kanamycin, the MIC increased several fold when we cloned RV0842. So just to summarize, for rifampicin resistance, we could see that there was a basal level of upregulation of efflux genes in both resistant and susceptible isolates, but that is true for all the antibiotics that we have studied. There was no definite association that we found, but 0676C and RV0194 were definitely found much more in the resistant isolates with a higher MIC. For isoniazid resistance, we saw these 5 genes that were only found in the resistant isolates. For ethambutol, again, we did not find, as I said, no definite link between RV0842 and ethambutol resistance, but RV0842 had been upregulated in several of the resistant isolates. So that could be that the RV042, the upregulation is maybe not enough to lead to an increase in the MIC, but it is definitely acting there. And there were 2 mutations that were found, the 37 nucleotide deletion in RV2459, which is the JEF-A, which was also shown in the JALMA study to be associated with the ethambutol and isoniazid tolerance. So we found this mutation in this gene, and we found another mutation in INEA, which is a novel mutation. The INEA has also been shown to lead to isoniazid and ethambutol tolerance, but no, this mutation had not been reported. But in our study, INEA was found in both resistant and susceptible isolates, so we need to study this further. For kanamycin, of course, RV0842 was not only upregulated, but it was also associated. We could define that it was associated with kanamycin resistance by our cloning experiment. So this is just there to say that if we are looking at maybe what we can see above the surface, and genomic mutations are the easiest to look at, but there might be much more hiding beneath the surface that we need to see. So the take-home message that efflux mechanisms may contribute, actually I should write may contribute in a major way here to intrinsic resistance. And they could also be a roadblock to discovery to novel drugs, because as we have seen the binaquilin, we know that efflux mechanisms are present. So we cannot really ignore efflux mechanisms. And about efflux pump inhibitors, there have been studies on verapamil and triple CP. But we need more research and much better options for efflux pump inhibitors. So with that, I say thank you. And these are my collaborators. This is our lab. And we've also taken help from the Rajan Babu TB Hospital, which is the largest TB hospital, and the New Jersey Medical School for our sloppy molecular beacon assay, and the Milan group for whole genome sequencing. So thank you very much.
Please. So our next speaker is Dr. Vivek Rao, and Dr. Vivek Rao did his PhD from University Delhi South Campus in Professor Tyagi's group. And subsequently, he spent a couple of years abroad in USA and UK for his postdoctoral work. In 2010, he started his own research group in IGIB, and his group is interested in immune response pathway that gives specific advantage to Mycobacterium tuberculosis, and he is using it to develop host-directed therapy. And without taking any further time, I would request Dr. Rao to start his talk.
Thank you, Dr. Mohanty, and for the kind introduction, and thank you, chairs, for chairing the session. And at this point, I'd like to thank Raman, Nishit, Krish, and Amit for giving me this opportunity to present the work that we have been doing over the last 4 or 5 years. I mean, barring the 2 years that COVID really made a dip in this progress. But yeah, so the talk is going to be, the main hero of the talk is going to be sertraline. And at this point, I'd like to give how the talk is structured. How did we select sertraline? And sertraline, by the way, is an antidepressant that's been used extensively in the human population and is quite safe to use this molecule as an antidepressant, even in adults and in the younger population as well. Further on, I'd like to say, what does sertraline do that we selected this candidate? Thirdly, what is the advantage it gives? How does it work is one of the things that we are pursuing now. Why do we need and why do we think it is a game changer in TB drug therapy? And finally, who does it matter and why does it matter to people? So further on, I mean, just to say how we reached at it. So this is basically a quote from We Are at War. We've been at war with TB for several decades. We won sometimes, we've lost as well. And we're still understanding the pathogen. And this is basically Chanakya Niti, which says that we should, as a person defending or fighting a war, we should not only understand ourselves, but also the enemy very well. And this is also true for the Art of War by Master Sun. And Chanakya goes further and says that we should not hate your enemy, but rather love him. And I think this population, this audience, everybody loves TB. And I think TB also loves us. That's why it's been with us for several years. So taking this forward, what we thought was, can we understand the communication that occurs at the host cell microbacteria interface? And can we understand the host pathogen dialogue better? And this is a work primarily done by Prabhakar, who is now moved on to the US for his postdoc. And along with him, Deepthi, she has moved into an alternate carrier. And looking at how the mycobacterium interacts with the macrophage, gets inside, sometimes it gets lysed by phagolysomes, sometimes it's not, leads to a plethora of transcriptional changes, induction of genes, and secretion of several molecules. So we wanted to start looking at the precise determination of immune cell pathways that are modulated by MTB, and that leads to immunopathogenesis. And one thing that stood out when we looked at this was the type 1 interferon pathway, which was very robustly induced as early as 1 hour, between 1 and 3 hours. And even at 6 hours, you could see a very significant increase in interferon beta expression. Not only this, we could see that the whole gamut of genes that are involved in restricting the viral invasion in cells is also induced significantly in MTB infected macrophages. Moreover, if you look, there is a clear dose response, dose dependence increase in type 1 interferons, and it requires an active infection in the form of live infection, because heat-killed infections don't induce this response. So we then sought to understand, can we manipulate this response? So we had access to these cell lines, which were basically null in the type 1 interferon response induced by pathogens, typically the DNA sensing mechanisms, which is CGAS as the receptor and STING as the adapter. And what we can then tested whether antibiotics would have an effect in these backgrounds. And as you can see, compared to the wild type cell, loss of either CGAS or the adapter gave better control of bacteria. And then we thought, okay, can we chemically manipulate this pathway? And we had an antagonist to this signaling pathway, which is called BX795, which inhibits the signaling at the last step. And therefore, we tested this, and we can see that it by itself can induce stasis, but in combination with the antibiotics, it can induce control the bacteria better. BX795 was not being used, so we checked for literature, and we came across, wanted to look at molecules that can effectively be used as drugs. And so we came across this paper in 2010, wherein they had used, they had identified antipsychotics and antidepressants as potent inhibitors of this response in macrophages. And two of the compounds, which is trifluoroperazine and sertraline, both antidepressants, showed the best effect in stunting this response. Having access to sertraline in our lab, we first tested whether it does the same, whether it can restrict MTB-induced type 1 response. And as you can see, as you increase dose, you get a significant reduction in the type 1 response. What does it do? And this is primarily the work of Deepthi. What does it do alone? I mean, when you give sertraline to macrophages, what does it do to bacterial growth? We asked this question, and again, as you can see, similar to BX, across 5 days of culture, what you can see is the input remains almost identical to what was given. Sorry, the bacterial numbers remain identical to bacterial. The input values, which means that sertraline alone was bacteriostatic. We then asked the question, what would happen in this situation if there were antibiotics as well? And we used, given our previous experience with the BX795 system. So we started looking at antibiotics at different concentrations, isoniazid and rifampicin. We gave a very effective concentration. And then just to mimic artificially situations where antibiotic concentrations were lower, we started in giving lower doses of antibiotics to macrophages. And as you can see, across as the dose decreased, the efficiency of isoniazid and rifampicin is lower. And at concentrations, you could see that they were not able to restrict bacteria. But addition of sertraline definitely enhanced the potency of the antibiotics to control bacteria, even at concentrations where they were bacteria were not being restricted in growth. You could see some level of restriction with sertraline. This is all fine, very exciting. Then we asked a simple question: does it work in alternate systems? So by then, Dr. Dyagi's paper on using vitamin C to develop dormant bacteria-like phenotype. We tested our combination in that model, and you see again, while antibiotics don't work as well, you end up getting at least nearly a log better killing by addition of sertraline. And S here is not streptomycin, it's sertraline. So this was also exciting. The real challenge was to test whether what would happen if you had drug tolerant bacteria. And again, we had a clinical strain that was tolerant to antibiotics. And again, antibiotics don't work as efficiently, but then you give sertraline to this combination, will be at THP1 macrophages or in monocyte derived macrophages. You could see that sertraline enhanced by more than 20 folds. Very exciting in macrophage models often. Then we ask a simple question: what happens in the in vivo model? So to test in the in vivo model, we used two models. One was a chronic infection, wherein we assess for bacterial control over time in tissues. And then, of course, tissue histopathology forms a part of the analysis. Then the other part being a very acute infection in a highly susceptible model, and then where you look for host survival and tissue histopathology. And given the time that acute infection can lead to results, I would like to show that first, because if you give a very high dose of 500 CFU to this mouse, which is C3HEB FEJ mouse, the Kramnik mouse, you can see animals die by day 30. Antibiotics can work very well in prolonging the survival up to 90 days. You decrease the doses of antibiotics and progressively you can decrease the survival. And the best part was when you add sertraline at the lowest concentration, you have now shifted the curve to, as you can see, the median survival time. You can see that the curve has been shifted to almost 10-fold higher of the antibiotics alone. So this was very nice, and we also have data about what happens in the case of the higher concentration of drugs when you combine with sertraline, and that's part of the poster that's being presented, I think number 20 by Anjali. Maybe you can go and pick her brains on that aspect. So this was fantastic. Now we looked at, and sertraline alone, surprisingly, didn't do much. It could not shade. It was as well as probably somewhere in between no treatment and the lower dose of antibiotic. Well, we looked at tissue histopathology at day 30 when almost all animals of the no treatment group had died, and this is one of the lungs, and as you can see, severe lesions in this, caseating lesions in these animals. Antibiotics at the lowest dose do not do much, but sertraline gives a significant advantage. This is okay. Then we asked what happens in the chronic model. We took the Black 6 model, which is a more resistant model of infection. You can prolong the infection to nearly a year. Mice survive, and all you follow is basically growth kinetics. The first question that everybody asked was, does it give better clearance in tissues? And this is a work by Anjali, who's presenting the poster as well. And this is with the 4-drug combination, which is isoniazid, rifampicin, pyrazinamide, and ethambutol. And as you can see, even 8 weeks post-infection in the lung and the spleen, you could see that almost all of the animals had around anywhere between 100 and 500 bacilli with the 4-drug combination. But the combination with sertraline, only one animal showed any evidence of bacteria, and that too was identical to the best animal which had responded, actually the best in the HRZE alone group. And in the spleen, we could not detect any bacteria in this animals with the combination therapy. Now, this was very interesting. Further on, what happens? 9 weeks, we started decreasing the dose because this was a very high. People started saying that it was a very high dose. So we looked at a dose escalation curve, whether this is true. again, as we excuse me, as we decrease dose of sertraline, you can see that you are able to see CFUs coming back in animals. But again, this is again one animal out of 5, and HRZE seems to be having animals even at this dose, but by 9 weeks, you have cleared the infection even at 3-fold lower doses that we started with. And in the spleen, at no dose, I mean, even at the lowest dose, we did not see any animals. So this was fantastic that we could lower doses and we could see much better clearance, even faster clearance in the preclinical model of infection. But okay, this is 8 and 9 weeks. What happens before? Does it start before? Does it start? What is the story before 8 weeks? And so we looked at 1 week and 3 weeks, and as early as 1 week, you could start to see differences between the HRZE and the plus S group in the lungs, and by 3 weeks, you have a significant reduction in the levels of CFU, and this was more reflected when you look at the histopathological sections. You could see that as you progress from 1 week to 6 weeks, this is the NT group, this is the HRZE group, and you can see the lesions have completely almost disappeared, barring a few in the combination therapy group. So given that addition of SRT speeds up bacterial clearance and tissue recovery. So, and again, the simplest question, we had a drug tolerant strain. Does it work in the scenario where the infection is by a drug tolerant strain? So again, you can see in the lungs and the spleen, you could get better clearance with the conventional antibiotics against when you combine it with sertraline. So this is all okay. Now we move on to the next step where we wanted to see what is the mechanism. We looked at the immune response generated and in the macrophages, and we see that TNF is not changed at the transcript level. In fact, it is decreased at the protein secretion level. But again, IL-1 beta transcription level, not much change. But what happens is a significant production of IL-1 beta protein that's being secreted out of these cells. And this immediately prompted us to look into the inflammasome, because inflammasome is a key component, innate component of the cell that is mediating release of IL-1. And just to give an idea of what inflammasome is basically a multimeric protein complex that is formed in response to infection and several stresses in macrophages. And these, it's a kind of an innate defense that's induced in these cells. And so there is this receptor molecule, which is an NLRP3 that recognizes the stress that gets activated. Primarily, it's NF-kappa B activation as the first signal. Then there is this adapter, which is the ASC. That gets activated with the first signal. Then there is a second signal, which is probably NLRP3 translocation and other things like ROS and potassium efflux that activates that ubiquitous, the ASC, and then finally all of these combine together along with the pro-caspase-1 and then form this multimeric complex, which leads to activation of the caspase-1 that goes and cleaves the pro-IL-1 beta that is induced upon infection, in this case, the NF-kappa B, cleaves it to the mature form of IL-1 beta. This also leads to the cleavage of this molecule, which is a gasdermin D. Gasdermin D is also induced upon infection or NF-kappa B activation. And now the N-terminal of this oligomerizes, goes to the membrane, and eventually this pore is responsible for throwing out mature IL-1 beta outside the cells. So this was how the inflammasome works. So having seen that we have higher IL-1 beta in the supernatrons, we asked a simple question, is inflammasome? So, I mean, and to tell you that there are specific inhibitors of this pathway, isoliquiritigenin, which is a global inhibitor of the inflammasome complex, but MCC 950 is now recognized as a specific NLRP3 inhibitor. So the first question we asked was inflammasome required for antibiotic-mediated clearance of bacteria and cells? And so we activated the inflammasome by using LPS and nigericin, which is well recognized as an activator. And again, you can see activation does decrease the growth rate of bacteria in the cells. Inhibition, you can see that both of these inhibitors, you can revert the efficacy of sertraline in potentiating antibiotics. So this tells you that the host inflammasome activation is an important component. Then we ask the question, what is the secondary signal? And so potassium efflux is one of the signals for activating the inflammasome. So if you now give excess potassium outside the cell, then you would be able to inhibit the inflammasome activation. And that's what we did. We gave a KCl from the cell. It does not affect the input bacterial numbers, nor does it affect the ability of antibiotics. But the moment you hit it with HRS, it kind of reverts the potency of sertraline to activate the bacterial clearance. So moving on, so this is basically the summary. We have seen that sertraline augments frontline TB drugs and in both macrophage and preclinical models. And now I ask the question, why? Why is it's an antidepressant? Is it safe? So there were only 2 papers where antidepressants were used along with TB drugs. And one of them was a 19-year-old woman. She was given isoniazid and sertraline up to 150 mg, and up to 8 months, there was no adverse effects. That's alone with isoniazid. And there was this paper from India where they checked the blood biomarkers of patients with multi-drug resistant TB, and then looking at see if sertraline alters the biomarkers. There wasn't any change in the biomarkers, suggesting that the combination could probably be very safe. So again, this is a story where we are now realizing that TB drug therapy and treatment adherence is a problem. Depression in people often leads to them not adhering to drugs, the drug regimen, and leads to, as you know, drug resistance or not clearance of the bacteria. So, and now recently it's been understood that depression is an invisible burden, and that we need to address this comorbidity in TB. And so I think that is where we have an USP, that we can now tackle both the TB-induced depression or treatment-induced depression and the TB therapy. So finally, these are the students, the previous lab members, the current lab members, and the funding that helped us to reach this place. And finally, I'd like to leave you with a trivia, which is basically that the first, after World War I, there was a lot of stockpile of the chemical called hydrazine. Hydrazine was taken up by Roche to develop the first TB drug based on this called iproniazid. And when it was done in a clinical trial in Staten Island, people immediately realized that the patients became euphoric, and it was called dancing in the halls, though there were holes in the lungs. So done, almost. So iproniazid was the first antidepressant that was clinically tested in patients. So TB drug going to antidepressants, and we are coming back the whole circle. Now we are taking an antidepressant for TB therapy. Hopefully we'll reach where sometime in the future we'll reach where Anil Kaul has reached. Thank you.
Thank you, Vivek. Our next speaker is Dr. Dinesh Mahajan. Dinesh Mahajan is a medicinal chemist and is an associate professor at PHSTI. He has experience in doing drug discovery projects and in various things like oncology, infectious disease, and metabolic disorders. He has 2 identified leads that are in phase 2 clinical development, and the other 2 drug leads are under IND enabling studies. So please start with your talk. Thanks.
Thank you, Chairs. So drug discovery is a team effort. So you start with clinicians who give you some understanding on the clinical need of the disease. And then there is a role of disease biologist who will study those challenges using in vitro and in vivo models. And then there is a role of structural biology and computational chemist. And then there is a role of medicinal chemistry. So I'm basically a medicinal chemist, and my lab is focused on medicinal chemistry and PK/ADME optimizations. We work very closely with biologists. So this is a focus area, and this is how my lab actually works. So as I said, we closely work with different biology groups, and the focus area of my group is disease agonist, because for disease understanding and in vitro, in vivo assay understanding, we believe and trust on biology colleagues. What we bring on table is conceptualization of the discovery projects and defining of target product profile. And we also work on hit identification to lead optimizations efforts, which involve designing synthesis and SAR development, drug metabolism and pharmacokinetic evaluations, and also doing some preliminary toxicology study to establish therapeutic index of the drug lead. So the idea is that this medicinal chemistry group that we have will translate the basic science that these disease biology groups are developing in their labs and convert them into drug leads for clinical evaluation along with research and training. So just to put context in place, why we need new treatment. So I will not go into details. TB is pandemic. There is a high death count. So the drug-resistant TB is more difficult to treat, and there is a lower success rate, and also the economic burden of drug-resistant TB is significantly higher compared to drug-sensitive tuberculosis. So question arises, what kind of new treatment? So we are hearing this since this morning and also yesterday, that the current treatments have their own liabilities. They are long, complex, and this leads to compliance issue. And also there are issues like negative interactions and very high cost. So the straight one-line answer is we need simpler, short duration and less toxic treatment. So there are two different kind of efforts to develop these kind of ideal treatments. The one effort is based on development of new therapeutic regime using existing drugs, including TB and non-TB drugs. The other approach is to have new drug discovery to identify novel drugs having novel mechanism of action. So this leads to the point that what are the clinical needs that are there, or clinical challenges that are there to facilitate NTB goals. So if you focus, or if I conclude all the sessions, the talks of all the sessions. So there are three key needs that are required to facilitate NTB goal. So we need to have a shorter treatment and or reduce supervised doses. That's the primary need probably. We need to have improved treatment for drug-resistant TB. And there is a role of latent TB infection also if we want to end TB from the globe. So this comes to the idea of therapeutic product profile. This is something different than the target product profile. Target product profile is more based on your ligand, and therapeutic product profile is more based on your clinical need. So ideal therapeutic product profile is that the treatment should achieve at least one, new drug lead should achieve at least one of the above three objectives as a standalone or combination. And also the new drug lead should be compatible with existing TB drugs. So based on this, we have a few drug discovery programs at THSTI. So broadly, we can categorize those programs on new chemical entities development. And also we have interest on pharmacological improvement of existing drugs to enhance their tolerability or efficacy. So I will focus more on new chemical entity. So these are the different approaches which are focused on new chemical entity development. I will not go into details. Most of us, we are aware of. For today's talk, I will stay focused on phenotypic screening assays. So most of the work that I'm going to talk here, not most of, everything is done in collaboration with TB research group. So I'm leading medicinal chemistry group, and the TB research efforts are being led by Dr. Raman. So the work that I'm going to present here is a joint effort of these two research groups. So we have complementary skills. So TB research groups, they have understanding of disease biology, they have in vitro and ex vivo assays, and also they have disease animal models. So what we bring as a complementary skills on the table is design and synthesis expertise, PK/ADME, and bioanalytical and exploratory toxicology. So based on this, we have, and we believe that we have a complete pipeline to support preclinical drug discovery, starting from disease model hit to lead identification, PK/ADME optimizations. What we are missing is probably IND enablings and filing support, and which we are focused on industry collaboration. And just to give you idea of two leads that we have right now at THSTI, one is SPR113. These both are small molecules. SPR113 is an advanced lead, which is into IND enabling stage. I will not go into detail of SPR113 today, but I will stay focused on DR536. But briefly, one slide on SPR113 is a small molecule co-invented by THSTI and a biotech company, Sphera Pharma. We have granted patents, globally granted patents on this molecule. The mechanism of action is based on host target. There is a GPCR involved in mechanism of action. The molecule is equipotent in macrophages infected with drug-sensitive and drug-resistant strains, demonstrated efficacy standalone as well as combination in infected mice. So it's orally bioavailable. We have complete detailed PK profiling in mice, rat, and dogs also. It's very selective. There is no interaction with either of the human kinase, ion channel, or key enzymes. There is no hERG liability. The molecule is not mutagenic. We also done with MTTDR studies, and the NOL based on this study is 1200 MPK, which is way beyond than the EC50 in infected macrophages. So we also done with CNS and respiratory tox in rats. The CMC chemistry, manufacturing, and control is in place. We have also synthesized a GMP manufacturing batch, and that was used to do part of GLP tox studies. But there are few GLP tox studies which are pending, and that will eventually lead to IND filing and phase 1 clinical trials. So that's on SPR113. But as I said, I will talk or deliberate more on development of DR536. So these are the kind of assays that TB research group has at THSTI, and we use these assays, phenotypic based approach and infected macrophages based assays, and the chronic in vivo disease model for screening purpose and drug discovery efforts. So this is a kind of workflow. So medicinal chemistry coupled with primary assay, which is mostly liquid culture assays, and secondary assay, which is mostly infected macrophages assay. We use these two to identify hit and to optimize hit into lead molecules. Once we have a lead in a optimized hit in place, we went for ADME, PK, and animal efficacy, and to ensure drug-like properties in that molecule. And once we have that, we are focused on preliminary or exploratory tox and selectivity profile to establish therapeutic index of that molecule to make a lead candidate ready for further investigation or detailed investigation in the animal models. So this is what Raman actually identified based on his phenotypic approaches. He has a pool of molecules showing activity in liquid culture or in infected macrophages. He shared few structures with me. We picked few structures and we explored these two structures, and based on initial SAR, we prioritized MMV254 because of its chemical and metabolic stability. And very quickly, we developed an SAR and of approximately 150 NCs, and we end up with 514 and 536, having MIC 1x to 2x to INH, established animal efficacy, and also active against INH resistant stains. We have some idea on mechanism of action, and these are molecules or series in novel IP space. So this is the profile of identified hit MMV254. It showed dose dependent activity both in vitro as well as ex vivo assays. It also did not show any kind of negative interaction with the frontline or most of the clinically used TB drugs, which is an important point if you want to develop a drug, because any new drug will go for clinical evaluation in combination with the existing drugs. We have some kind of mechanism of action understanding. We believe that there is a pathogen enzyme amidase C, which is involved in the cleavage of this molecule. And we have this confirmation based on LCMS metabolite experiments, but we don't have absolute understanding on the target profile yet. So that is a work in progress. But unfortunately, when we dosed this identified hit to animals, it was well tolerable, but it did not show any efficacy either standalone or in combination with INH. So we went for MedChem optimizations. We developed a series of molecules, and with high chemical purity, we evaluated those molecules both on liquid culture as well as ex vivo infected macrophages. And we identified a couple of leads having MIC 1x2, 2x2 INH. And so this is the data on these two leads. And one of the lead, DR514, which is 2x2 INH in infected macrophages. But it was also found active against CATG mutated bacteria in infected macrophages. And that was one of the key outcome of this screening. We also evaluated 514 in different kind of clinical stains, well-characterized clinical stains, and we found that 514 is equipotent in drug susceptible as well as MDR and XDR clinical stains. So again, similar to identified hit MMV254, we did interaction study of DR514, which is the first lead, and we found that it did not interfere with in vitro activity of any of the first line and second line TB drug. Unfortunately, this time we managed to have efficacy at 50 mpk. Not only the standalone arm of DR514 showed comparable efficacy to INH, but in combination that was significantly more than a standalone of INH also, both in lung and spleen. And the follow-up, two other follow-up molecules having better bioavailability than 514, we saw similar kind of trend. And also we evaluated one lead 536 as a combination with the existing drugs by replacing one of the drug from the combination isoniazid. And you can see that there is a better clearance of MTB in animal experiments using that combination when you remove isoniazid with 536. So kind of summary, I will not go into detail. So we have a defined SAR. The molecules are active against drug-resistant stains also. These are orally bioavailable molecules. We demonstrated animal efficacy. This is our plan. We need to do detailed DMPK profiling of the leads. We have planned to do drug-drug interaction using naive mice pharmacokinetic studies. And we will evaluate two molecules in dose ranging animal efficacies, both in mice and guinea pig, using drug sensitive as well as MDR infection stains. So acknowledgements, as I said, most of the work is done by two different groups, TB research group and medicine chemistry group. So Jashwant and Sonu Gupta, they both helped us on metabolite experiments using LCMS. Unfortunately, we did not manage to get external funding till now, but we are surviving on intramural grants. So that's a different story. On SPR 113, so there are three financial contributor as well as owner of IP of SPR 113 involving THSTI, Wellcome Trust, and Sphera Pharma. Thank you. Thank you for your time.
Thank you, Dinesh. Please come.
Yeah, now we'll go to the question and answer session. Yeah, please.
Excuse me. Hi.
Hello, can you hear me? Yeah. Yeah. So I have one question for Dr. Mandira. So this, how are these efflux pumps have evolved? Means they were there with the bacterium ever since it was there, or with respect to time, it has evolved to efflux the drugs?
Well, that's a very good question, but we've not really seen how far back they go. But ever since people have started looking for efflux pumps, they have been there. And as we all saw that there's a basal level of efflux pumps that's been acting. It's there in sensitive, it's there in resistant. Some of the studies, the ones that I showed earlier studies, they just saw the basal level of efflux pumps. But what we saw in our study was even when we gave stress to a susceptible organism, there was upregulation. So which means that it is there in the organism, and it is probably there to prime the organism to fight against stress. And this is what we have to look into. Is it also contributing to or lead to resistance in the future?
Okay, thank you. Next question to Dinesh. Dinesh, it was nice. Yeah. So all these molecules that you have found, SPR 113, MMB 254, DR 514, and so many. How were these molecules to begin with identified? Through small molecule screening or computationally? How are these molecules?
Yeah. So SPR 113 is based on Dr. Kanuri Rao's initial work based on siRNA based macrophage screening.
I see.
So, and that was a target based approach. He managed to come up with a target involved in a host target involved in pathogenicity and sustenance of MTB in macrophages. That was for SPR 113. The other all molecules are based on phenotypic screening that we started with the library first rather than target. And we actually don't know the absolute target right now. So Raman is working on that.
Yeah, yeah. My question is for Vivek. Hi, Vivek. Very nice work. I just have a query regarding using sertraline in conjunction with INH. What was the rationale of introducing INH in the combination when sertraline is inducing bacteriostatic effect, because INH don't work on bacteriostatic?
So we tested sertraline in both combinations of the 4 drugs, HRZE, and the 2 drugs that are followed in the extensive therapy, which is isoniazid and rifampicin. So there's nothing like, we want to test against one, because in reality, whatever adjunct, it's going to be a major, it's never going to be possible that we could say, okay, we will replace isoniazid from TB therapy, and that would be impossible with respect to TB therapy. So I think if at all I have to use an adjunct, then it has to be in the presence of both the drugs. You're right in this. That's why we chose both kinds of therapies.
Yeah, Professor Tyagi after that. Yeah.
My question is to Dr. Shandil. The most important molecules which you showed in the last part of your talk, where do they stand in the clinical pipeline?
The last one, TBS 7371. So there was a series that was being developed, and it took about 225 compounds were synthesized. So when I showed on slide the 4 or 5 compounds, that was a stage where we were around the final stage of the compounds. Some further modification done, and basically the TBS 7371 also has all these characteristics which I showed in the early leads. All the physicochemical properties, pharmacokinetics, efficacy, and everything. So yes, it was a gradual development through lead optimization program.
I have one question to Vivek. This is regarding sertraline. So you showed the effect in combination with antimicrobial drugs, but if you see the mechanism is not specific to the TB drug because it alone doesn't show. So are there studies done, or you have studied any other bacterial infection resistant to antibiotics, because its mechanism of action is more general, inhibitor of uptake?
That's partly true. Sertraline has been shown to be used as an adjunct for multiple different bacterial infections. And primarily we think, it's not specific for TB, definitely. But yes, wherever there is involvement of, I would think at this point, my guess would be restricting the type 1 response. Then sertraline and similar molecules, like a lot of antipsychotics could play an important role. And yes, it is not specific for TB alone.
So my question is for Vivek, sir. Sir, you showed the therapeutic efficacy of sertraline with anti-TB drugs. Did you also do any reactivation studies or relapse?
No. Correct, good question. This was asked by in one of our monitoring meetings, as even Anil suggested that we do this, and then COVID in a way helped us because we had a set of mice that we had infected and we're trying to treat. COVID lockdown, we just missed a couple of time points and then said, okay, let's leave it for a week and let's leave it for nearly a year. And this is a very small set of mice. So we had 5 animals in each group. And then the treatment was taken after 7-8 weeks and left for nearly a year in C57 Black 6. And then after this time, the 4-drug therapy gave higher number of colonies in the lungs as compared to what was seen at the time point when he had shut down treatment. But there was no colony that came back in the sertraline group. So we think it is also preventing reactivation. But as I said, this is something that we had to leave and we haven't done a detailed study on this as of it.
Thank you.
This is the last question from there.
I have a question for the Vivek. So you have blocked the type 1 interferon. So if you block the type 1 interferon, you compensate on the gamma interferon, correct?
Yes.
So if so, then you are sensitizing the system for the TH17. So from that angle, my question is that how this sertraline is, changing the granulometric lesion. Have you analyzed the granulometric lesions in these mice?
We haven't done in-depth analysis of how the cellular profiles or the granulomatous lesions are changing. But what we know is, it also has probably anti-inflammatory properties of its own. So we know for sure that granulomas are, I would say the lungs are much better in terms of lesions, in terms of recovery when you give sertraline to the animals.
So how these macrophages are turning, they're turning more like a CD14 dim, CD16
We don't know that. We haven't analyzed anything into that.
Any
level.
scavenging macrophages, like the
Sorry?
scavenging phenotype. Are they acquiring the scavenging phenotype under the influence of the type 1 interferon blocking?
We haven't done those kind of studies here. We haven't gone too much into the depth of the mechanism because we are now focusing on trying to take this forward. And the mechanism part is a little lagging behind at this point.
Thank you. You guys can have the discussion during the lunch hour with the speakers that are available. Thank you for stimulating session and thanks all of we would like to thank all the speakers.
I thank all the speakers for shedding lights on different facets of TB drug development and on the challenges it throws at us. I now request the session chairs to felicitate the speakers, Professor Mohante to Dr. Shandil, Professor Mohante to Dr. Mandira Verma, Professor Nandi Kori to Dr. Vivek, Professor Nandi Kori to Dr. Mahajan, thank you, sir. I now request Dr. Nishit Agrawal to felicitate the session chairs, Professor Mohante and Professor Nandi Kori. I thank all the speakers and the session chairs. We will now break for the poster session and lunch. Lunch is arranged in the second dome area, that is to the right of you. The posters are placed in the first dome area near the main entry of the auditorium and the corridor that connects first and second dome area. We request all the poster presenters to be present at their posters for discussions. I request all of you to gather back in the auditorium 3:45 p.m. for the start of the next session. Thank you very much.
Hello, everybody. I request you all to please get settled. We'll be starting the session 9 momentarily. If someone has lost their laptop connector, please contact the registration desk. I repeat, if someone has lost their laptop connectors, please contact the registration desk. I request everyone to kindly settle down. The primary accepted norm in the bacteria-driven infectious diseases is to target the pathogen for the therapy. However, understanding the host and its response to pathogen dynamics and pathogenesis can also lead to targeting the host in conjunction with the pathogen. In this context, on behalf of the organizing committee, I warmly invite Professor William Bishai to deliver his keynote lecture titled Glutamine in the Granuloma, Reversing an Immunometabolic MTB Virulence Mechanism with a Novel Host-Directed Therapy. Dr. William Bishai is currently a professor of medicine at Johns Hopkins University, USA. He also serves as the co-director for the JHU Center for Tuberculosis Research. He also served as the founding director of the Howard Hughes Medical Institute-funded KwaZulu-Natal Research Institute for TB and HIV in South Africa. Dr. Bishai's interest involved TB pathogenesis and animal models of pulmonary infections. His work has led to second-generation BCG vaccine, which is being advanced towards clinical trials. He was recently in the review panel of WHO Stop TB Partnership Coordinating Board. Following the keynote address, there'll be a 5-minute question and answer session. In the interest of time, I request the audience to keep your questions brief so that there is opportunity for others. Sir, I request you to kindly share your valuable insights with us.
Thank you very much for that very kind introduction. I too want to join the chorus of those who've thanked the organizers. It's really been a wonderful meeting. It's especially nice to be back here to see a lot of old friends, including some trainees who I think are still friends, as well as their trainers. And so it's a wonderful audience. I want to say a word about this institution. One of your early faculty members, Nishith, was with me, and he came to me in about 2010 and told me about this opportunity. And I was one of the many times I was wrong. I said, you know, Nishith, you have such a great potential. It's a plot of land in the countryside, and worse than that, it's promises by politicians. Wisely, he didn't listen to me. And look what you've accomplished in 12 years. It's truly remarkable. And I warmly congratulate you. So I work in a cancer research building. And one of the things I've learned over the years is that, first of all, there are a lot of similarities between the solid tumor, a ball of cancer cells and immune cells, and the TB granuloma, a ball of bacteria and immune cells. I've also learned that the cancer guys are usually about 5 to 10 years ahead of us. So sometimes it pays to go to their seminars. And the story I want to tell you this afternoon is related to that. So you've all probably had to study the well-known Warburg phenomenon about the tumor microenvironment. Tumor cells, it was found by Warburg many years ago, change their metabolism from a balance between oxidative phosphorylation and anaerobic glycolysis to one that's dominated by anaerobic glycolysis and the production of lactate. And more, some have called these tumors glucose addicted for this reason. It's also been shown more recently that tumors become glutamine addicted. Some tumors, due to this MYC oncogene, upregulate this channel ASCT2, which promotes the uptake of glutamine, and therefore over time they become glutamine addicted. So among cancer immunologists, they've come to this kind of thought process of the glutamine and glucose addicted tumor cell, which is heavily anabolic. The tumor cells are growing, but the use of glycolysis leads to nutrient depletion, hypoxia, acidosis, and the expansion of immunosuppressive myeloid-derived suppressor cells. This in turn leads to the effector T cells being exhausted, apoptotic, and having reduced effector function, and as I mentioned, MDSC expansion. This gives the tumor the advantage. Well, I went to a seminar way back in 2019, and I heard about this story from these two cancer biologists. We should say that Jonathan Powell is a cancer immunologist, whereas Barbara Slusher is a very talented medicinal chemist. And they had wisely read the old literature about glutamine and tumors, and they were aware of this glutamine analog, diazoxynorlucine, or more easy to understand to say, DON. And as you can see, DON is glutamine, but for the sidearm, the amide has been replaced by this diazoxo warhead that is capable of covalently inactivating an enzyme that binds to this molecule. DON had great activity against cells, but was very toxic in animal models, so was abandoned. And Barb Slusher said to herself, I know what to do. And so she came up with a DON prodrug and named it JHU83, and took the very interesting approach of decorating DON by putting an isoleucine at the N terminus and an ethyl ester on the carboxyl group. So it's a very simple modification, but in characterizing this drug, they showed it had very potent anti-tumor effects in mice, and that the effect was driven by immune modulation, not by direct tumor killing. And so this led to a model where JHU83 treatment reversed all of those negative effects I just mentioned. There was nutrient abundance, reduced hypoxia, reduced acidosis. The MDSCs now were exhausted. The T cells were reinvigorated, more effector function, and this in turn led to killing of cancer cells. They also did a second, well, one of many smart things. JHU83 was their second best drug. The best drug was licensed by this company, Drayson Pharmaceuticals. Its structure is now published. It's called zerpiglenistat, and it's in phase 2 clinical trials for solid tumors. So one of the advantages I have of all this gray hair is, even though the light bulb is dimmer, every once in a while, because I was around so long ago, I remember stuff. And when I heard Jonathan Powell and Barb Slusher's story, I remembered these papers from Marcus Horowitz, who spent many years characterizing a glutamine synthetase called GlynA1 in M. tuberculosis. He published 10 papers over an 11-year period from 94 to 2005. And to oversimplify a very complex story, GlynA1 was one of 4 glutamine synthetases in the MTB genome, but it was extracellular. He could show that it was inhibited by this glutamine analog, methionine sulfoxamine, that it was essential for growth in vitro, but the strains would grow if you supplemented with glutamine, i.e. it was a glutamine auxotroph. And then when you took the mutant, the glutamine auxotroph, and put it in animal models, it was highly attenuated. So interestingly, an essential gene that was extracellularly secreted. And to my knowledge, Dr. Horowitz has never come up with a good explanation for why this valuable metabolic enzyme was thrown out of the cells. So anyway, thinking about things, we put together this hypothesis. M. tuberculosis, with its extracellular GlynA1, makes glutamine in the extracellular environment. And upon infection, potentially a gradient of extracellular glutamine accumulates, and then as the granuloma assembles, there's the same phenomenon that my cancer colleagues observed, excess glutamine, T cell exhaustion, immunosuppressive myeloid-derived suppressor cell expansion, and the microbes have the upper hand, and there's a granuloma progression. So our hypothesis, our plan, was to test JHU83, a wonderful anti-tumor agent through an immunologic mechanism as a host-directed therapy for TB. So luckily, right around the same time, I had a very talented postdoctoral fellow from Manjula Reddy's lab in Hyderabad named Sadia Parveen. And we had several compounds to test. There was the prodrug I've been telling you about. There was the hydrolyzed prodrug, the naked one, Dawn, as well as Marcus Horowitz's drug, methionine sulfoxamine. And we've tested a number of host-directed therapies over the year, and I've learned the hard way that you have to do things in the right order. And we have the so-called commandments on how to test a host-directed therapy. If you're claiming it's host-directed, make sure it doesn't have an MIC against the microbe. So Sadia dutifully did those experiments, and lo and behold, JHU83 had an MIC of 1, which is pretty good, microgram per ml. It was better than MSO, so did Dawn. As you can see, this approaches the MIC of our traditional drugs like rifampin. It also was capable of killing MTB, or at least Dawn was capable of killing MTB inside macrophages. So Huxley said, the great tragedy of science is the slaying of a beautiful hypothesis by an ugly fact. Well, the honest truth is we had a grant, so we weren't going to let this stand in our way. The grant said we were going to test it in animal models and whatnot. So Sadia bravely persevered on. So she set up some traditional models, many of which you've heard about over this wonderful 3-day session. We started with immunocompetent mice, happened to use 129S2s, and we infected them with about 200 CFUs and had sacrifice dates at 2 weeks and 5 weeks for these parameters, which I'm going to spend the rest of the talk telling you about. And importantly, knowing that this drug JHU83 had an antibacterial effect, we used a true antibiotic, rifampin, which has no known host-directed therapies. So first experiment in this mouse model gave a compelling result. The mice that were treated with JHU-83 for 5 weeks survived statistically significantly longer than PBS-treated mice. I don't generally like curves like this. I want them to have both curves die, but we looked at it every which way, and there was really no way that if she had gavaged mice for longer, the p-value would have changed. So JHU-83 prolonged survival of mice when given as an oral agent. She looked at the lung CFUs, and interestingly, for the first 2 weeks, the drug-treated mice, shown in green, proliferated to the same degree from 2 logs to 6.5 logs. And there was not a statistically significant difference, but there was a difference between PBS-treated and rifampin, as indicated by the gray asterisk. At 5 weeks, the JHU-treated animals looked very much like the rifampin-treated animals, indicating a bacterial containment effect. And as when we looked at the pathology, as you can see, JHU-83 lungs looked better than the PBS lungs. I wish I could say more than that. I'm so jealous of Audrey, who can actually really interpret these H&E stains. And there were fewer granulomas. We went to the trouble of checking a separate mouse model, the so-called Kramnik mouse model. And as you can see, what we observed in 129S2s was also observed in the second mouse model, the Kramnik model. The next thing was to test the drug in SCID mice. Here you saw, we observed what we hoped. The cancer therapy JHU-83 treated animals and the PBS treated animals all died at about day 40. There was no beneficial effect of the drug, whereas the rifampin treated mice survived much longer. And the lung CFUs reflected this as well. Very high counts in both PBS treated and JHU-83 treated. Rifampin lowered the count. So we were somewhat encouraged by the fact that there was no therapeutic efficacy in SCID mice, and this gave a little fuel to our beautiful hypothesis. At least it lined up with a potential host-directed mechanism. We started looking at immune cell populations by flow cytometry. Here you see that at week 2, JHU-83 led to a statistically significant increase in total CD4 cells, naive CD4 cells, pro-inflammatory CD4 cells, TNF-alpha expressing, interferon gamma expressing, but immunosuppressive T cells expressing IL-10 were reduced. And when we looked at subpopulations using this terminal differentiation marker, which correlates with T cell exhaustion, there were fewer of these potentially exhausted CD4 cells and more of these CD62 ligand expressing CD4 cells, suggesting perhaps better central memory expression in these T cells. And I just want to point out one thing. This all was done at 2 weeks when the CFU counts between JHU-83 and PBS treatment were approximately the same. So the argument that there were fewer bacteria and that led to the beneficial effects on the CD4 populations was somewhat mitigated because there were the same abundance of bacteria at that time point. We looked at CD8 cells. They really didn't differ at week 2. We saw fewer potentially exhausted CD8 cells and slightly more central memory cells, but it was hard to get too excited about this. Then we turned to B cells. Yesterday, Deepak told you about some of these interesting follicular helper B cells in the SIG-H vaccinated animals. And we saw more B cells at week 5. But also RIF did the same thing, more memory B cells at week 5, more of these follicular helper CD4 cells at week 2, and also follicular helper CD8s at week 2. What about myeloid cells? So total CD11B, really no difference between PBS and JHU-83 treated. But then we looked at alveolar macrophages, which are thought to be the lung resident permissive cells that are infected and can't contain bacterial growth, significantly reduced population of these permissive alveolar macrophages, but the interstitial macrophages, which are thought to be MTB restrictive, which are recruited from the periphery, were more abundant at the week 5 time point. And then looking at some of the submarkers on the alveolar macrophages, there were more of these co-stimulatory T cell ligand expressing cells, CD86, fewer myeloid cells that express CD206, which is thought to be inflammation resolving, suggesting that the myeloid cells were still revved up to contain, there were fewer of these anti-inflammatory cells. And similarly, when we looked at both monocytic myeloid-derived suppressor cells and granulocytic myeloid-derived suppressor cells, fewer of these immunosuppressive types of myeloid cells. So get to the punchline, Bill. What did it do to the metabolome? So we used the entire lung of these 3 groups of mice to look at metabolomics. And our metabolomics detected 144 metabolites. 48 were higher. JHU83 was higher than PBS. 96 JHU83 was lower than PBS. And here I'm just showing you the PBS JHU83 comparisons, not the rifampin comparisons, but we had those as well. What about glutamine levels? So there was a 23% reduction of glutamine in the total lung homogenate in the JHU83 treated lungs. However, this did not achieve statistical significance. Rifampin, as you can see, looked very much like the PBS, same levels of glutamine. What about some other metabolites? We decided to investigate amino acid pathways. And here you see the tryptophan metabolism metabolites. We were able to capture 11 metabolites, and you can see the heat map here, but only 2 were statistically significantly different between PBS and JHU. Quinolinic acid was lower, and hydroxyindoleacetic acid, 5-HIAA, was higher. And you see that data here. So mapping out this series of pathways, and I'm just showing you the 2 metabolites that were statistically significantly different with this green arrow and this red arrow. As you know, there's a bifurcation. One direction leads you to 5-HIAA, another down to quinolinic acid, with 2 important things in the middle. Indolamide dioxygenase, which you heard about very eloquently from Smriti Mehra yesterday, and kynurenine, which she also touched on. And here are the papers that Smriti told you about on the left. She showed very beautifully when you use 1-methyltryptophan, an inhibitor of IDO1, the non-human primates showed a significant benefit from TB progression. And as is also known, kynurenine is an immunosuppressive metabolite that has been identified as a marker for progressive TB. And here's a nice paper from Vidya Mave from Pune and Johns Hopkins, showing that in pediatric tuberculosis, kynurenine elevation compared to tryptophan is a marker of tuberculosis. We also looked at the arginine pathway, and here we captured 8 metabolites, and 6 of the 8 had statistically significant differences between JHU83 and PBS. Citrulline levels were elevated. The other 5 shown in red were relatively reduced statistically significantly. And again, I'm just showing you the ones that were statistically significant with the colored arrows. How does one interpret this? Well, you don't have to be a rocket scientist to know that nitric oxide synthase producing NO has potent antibacterial activity, as has been elegantly shown by Carl Nathan and his colleagues over the last 20 years. And so we felt that potentially JHU83 had the ability to shift arginine metabolism away from polyamines down the citrulline pathway, where one would expect higher levels of the antibacterial nitric oxide. So our working model, we feel that I've shown you today that the administration of JHU83 to mice is restrictive for MTB proliferation. There are higher levels of T effector immunity, lower levels of the permissive alveolar macrophages and myeloid-derived suppressor cells, higher levels of the restrictive interstitial macrophages, and lower levels of IDL1 and kynurenine in the tryptophan pathway, and higher levels of the NOS side of the arginine pathway. Potentially, these are all adding up to greater bacterial containment. So to summarize everything that I've told you, good evidence that JHU83 is a potent immunologic drug against cancer. We had this quote unquote beautiful hypothesis, some of which I've proven to you today and some of which remains a little bit nebulous. But we had the hypothesis that inhibiting glutamine metabolism could be host beneficial. I think the data show that the drug also has direct antibacterial activity, but has these pathogen TB disease progression markers. And the fact that the efficacy of the drug is lost in SCID mice mitigates towards a host-directed therapy as being more important, at least in the SCID mouse model that I showed you, than the antibacterial action. The flow cytometry showed higher CD4 cells and higher macrophage activation markers. Again, the fact that all of this occurred at the early time point when the CFU counts were about the same gives the host-directed therapy hypothesis a little more horsepower. And I made some arguments from metabolomics that tryptophan metabolism shifts away from the immunosuppressive IDO1 kynurenine pathway, and the arginine pathways shift towards antibacterial nitric oxide production. I would say this is a plausible mechanism, but more data are needed. So I want to close by saying immunometabolism is a growing area. The cancer immunologists have been on it for the better part of a decade. As you heard from Smriti Mehra, IDO1 is a target, and 1-methyltryptophan is a beautiful immunometabolic inhibitor that is proven very clearly in the non-human primate model. So I think immunometabolic modulators deserve a little more attention. They have potential as host-directed therapies, and hopefully going forward, this will be a promising pathway. Thank you to Sadia Parveen, who did all of the experiments I showed you, and our wonderful collaborators, and thank you for your kind attention.
Thank you so much, sir. The floor is now open for questions.
Okay, I'm going to take friendly questions. Deepak, I'll get to you next, Govardhan.
Well, congratulations to you and Sadia. This is wonderful work. So I have a question, but before that, it looks like to me that the you and Smriti to write an MPI R01 grant, inhibiting both glutamine and tryptophan catabolism. So my question is that your glutamine levels were not statistically down, they were only down 23%. But what about enzymatic activity? Is there an assay that you can do to look at the enzymatic activity? And my second question is, have you thought of looking at cells that, immune cells that are changed, like interstitial macrophages, for example, by both rifampin and JHU83 treatment, to see if there are different correlates there?
Yeah. I think we were a little bit overambitious to go to your first question. What about the enzymes themselves? A whole organ is a tough thing to do metabolomics on. And, obviously we're looking at metabolites from dozens of different cell types, including the lung architectural cells, as well as the immune cells. And the cancer guys have an advantage because they do their metabolomics on excised tumors that basically have just tumors and immune cells. So we need to dig down. We have begun a very large single-cell RNA-seq project to try to figure out what the transcripts for all of those enzymes are, and be the first to agree we need to look at the enzyme levels as well. To your second question, yeah, I think we kind of went straight to the mice, and we haven't looked at the drug's effect in isolated immune cells, and there's probably a lot more running room there. Govardhan.
Yeah, Bill, nice talk. I have two small questions. Number one, if it is a host-directed therapy, why not combine with the conventional antibiotics and look at how it works? Because it may show you additive effect. And my second question is, if GHU 83, I didn't really understand how it's increasing the naive T cells.
How is influencing the naive T cells? Yeah, so the cancer model is that it's all about the milieu, that a lot of those cells are there already, but they're suppressed because of the local tumor microenvironment. But they're just looking at the tumors, isolated tumors. Here we're looking at the whole lung, and so we're looking at what happens with the entire mouse system. So our feeling was that potentially the microenvironment was such that the myeloid cells could now attract more T cells, and that led to T cells being there, potentially recruited from the bone marrow. But it's a little wishy-washy as to why they're naive and not further differentiated. Nishith.
Yeah, so very nice talk, Bill. So I have two questions. One is that, did you try checking those cytokines in the absence of infection, treated and not treated with the GHU drug? And secondly, in your metabolic analysis, you showed that this drug affects the carbon metabolism, right? The glycolysis pathway. So did you see those metabolites which are related to the carbon metabolism also?
Yeah, so to answer your second question first, 144 is not very many. So we had some of the TCA cycles, some of glycolysis, and the right way to do it is fluxes. And I don't think fluxes is very amenable to an entire organ. So we're thinking about ways we can actually look at the glycolysis versus OXPHOS pathways in a more defined system where we're not taking just a whole organ, homogenate, and doing the metabolomics. Yeah, your first question is, we did not test the effect of the drug against naive mice. We're only looking at the lung here. Our colleagues have done extensive safety studies on the better of the two inhibitors, and it was quite safe. My guess is it probably wouldn't do much in a healthy lung where there are not very many immune cells in the first place, but I don't actually have the answer to that. Yes.
Okay. So very nice work. Thank you. My two questions. First is, you mentioned about a glutamine gradient. I would like to hear more about that. And second is, would you have expected these effects on the tryptophan and arginine metabolic?
Would I have expected the effects? No, I did not. And I read Powell's papers, and they did not have they showed that kynurenine accumulated. But I guess the ARG pathway was somewhat anticipated by Powell's work in the tumors. But our findings in the tryptophan pathway were not predicted by the tumor. So what was your first question again? The glutamine gradient. Oh yeah, so yeah, thank you. So that's unproven. We need to look in the whole granulomas. That's a part of what I would call the beautiful hypothesis. It's hypothetical, unproven, needs more work.
Yeah, excellent talk. Two quick questions. One is your KLRG1 staining, essentially the T-cell exhaustion story. I know JHU 083 used for CAR T-cell therapy enhancement. So I was wondering whether it is in case of co-infection with HIV and T-cell exhaustion could be a tricky issue, can be used. And number one. Number two, one of the byproducts of arginine, alpha-ketoglutarate, actually it changes the epigenome actually, because essentially H3K4 becomes hypomethylated. I was just wondering whether in those cells where there's a T-cell exhaustion is reversed, do you think there are some epigenetic changes because this is supposed to act as antagonist?
Yeah, so the answer to that question is I don't know. We're hoping that the single cell will enlighten us. Perhaps we need to dig down on the flow as well. I love your idea about exhaustion and TB HIV, and I think that grant that it looks like Deepak and I are going to be writing, that would be a aim 3 maybe. Okay, but thank you for that helpful suggestion.
Yeah, hi, it's a nice talk, So my question is all about the immune metabolic programming of the alveolar macrophages. So if we actually have to define that this glutamine is really immunometabolic programmer, so can we rescue this phenotype which you have in the condition of low glutamine by the glucose treatment? If that is the case, how this metformin can also be a player in polarizing these M1 macrophages to the M2 and vice versa?
Yeah, so I don't know enough about metformin and TORC inhibition to really speculate on how glutamine metabolism could intersect. I think it's important to recognize, and my colleague Powell stresses this, that this is not a glutamine synthetase inhibitor only. Any enzyme that has glutamine as a substrate or a product could potentially bind it and be inactivated. So there are going to be pleiotropic effects. In the tumor microenvironment, that clearly leads to good stuff. But the precise mechanism, I think, is going to take years to work out because there's so many enzymes that use glutamine.
One last quick question. So is there an impact of this glutamine and tryptophan rheostate on the maturation of these dendritic cells and the macrophages? Considering from the tumor immunology platform, this is very much important, that these cells need to be mature before they see the mycobacterium, even in the comorbid conditions. So whether is there, did you analyze the impact of the glutamine rheostate on the CD83, 86 marker reshedding and all?
Yeah, so I think you're suggesting that we vary the treatment temporally, pre-treat and then infect to try to reset the immune system before it sees the TB. And we have only done this one model. It's a good idea. Okay.
Till the time.
So you talked about memory cells. So as far as I know, in humans, it is effector memory and central memory. So did you check for both? Is it the same in mouse?
We didn't do effector memory, just those central memory markers, but
Okay.
we should. But again, I'll come back to single cell RNA-seq is supposed to give you everything, so maybe we'll find it there.
Another unrelated question is actually we are doing some work in household contacts of TB. So we were comparing arginine levels along with nitric oxide levels in PTB patients and household contacts. So we found that arginine levels as well as nitric oxides were raised in PTB group, somewhat in household contact compared to controls. So we were actually clueless, why is it so? And you are showing that, so with this thing, arginine levels will increase and so nitric oxide levels will increase, something like that you showed.
Yeah, so I didn't actually, we didn't find that arginine levels were statistically significantly different between drug-treated and PBS treated. They had modest differences, but it wasn't statistical. Again, in this whole long mishmash. So I'm only showing you the metabolites we found. We didn't find them all. and the ones that were statistically significantly different. So arginine itself, I can't say whether, we only have PBS and drug treated. We don't have pre-treatment arginine levels in these mice. So I couldn't really speak to your point about the household context.
Thank you.
Yeah, hi, Bill. Thanks for a nice talk. I just want to know your molecule inhibited granuloma formation, but still you see a bacteriostatic phenotype, which is mainly seen in granulomas. So what could be the mechanism or reasons for having bacteriostatic phenotype when
the antibacterial
in spite
effect.
there is
Yeah.
a less granuloma?
I can tell you, so you asked a good question, and I was expecting it as the first question, but Deepak was too nice. You really want to convince me that it's a host-directed therapy, make a mutant that's resistant to the drug, eliminate the antibacterial effect, and show me that the drug still has activity in these animal models when it's resistant to the drug. So we tried. And the mutants, it's a very low frequency of resistance mutation, somewhere north of 10 to the 9th. They're not easy to elicit. None of them are in glutamine synthetase, although with methionine sulfoxamine, right smack in the active site of glutamine synthetase. And one was an aspartate transporter, maybe related. The other four, we have no idea how resistance to the drug occurs. The sad part of the story is we tested all 5 of those and they're attenuated in mice. So we were sort of blocked, at least with those 5 mutants, from testing a resistant mutant head-to-head in this model, because if you have a 3-legged dog, it's going to lose the race to the 4-legged dog, and an attenuated mutant is not a fair comparator. We only did MABA. What you saw is microplate Alamar blue, which is really looking for cidal activity rather than statics. We haven't carefully analyzed the drug mechanism of killing. Thank you.
Thank you so much, sir, for such an enlightening and thought-provoking lecture. I now request Dr. Nishit Agarwal to kindly felicitate Dr. Bishai. Thank you, sir. We now move to our last, but one of the most important sessions, session 9, that dwells in the host-pathogen interaction. Understanding host-pathogen interaction is central to design of vaccines, therapeutic agents, and biomarkers. I take this opportunity to warmly welcome Dr. Narayan Swami Balaji and Dr. Deepak Kaushal to chair this session. Professor Balaji is currently at IISc Bangalore, has expertise in understanding intricate dynamics of host signaling pathways with epigenetic machinery upon distinct pathogenic insults. Professor Balaji is a fellow of all 3 science academies in India and is recognized by various awards from the Government of India and Government of Karnataka. Of late, Professor Balaji's group has forayed into understanding host responses to other distinct pathogenic fungi, such as Candida albicans and Cryptococcus neoformans. Dr. Deepak Kaushal, currently the director of Southwest National Primate Research Center at Texas, is recognized for developing the macaque model of TB. He also oversees development of macaque models of COVID-19 and testing of vaccines and therapeutics. We have 4 talks in session 9, each for 20 minutes. Following the talks, there'll be a brief question and answer session with the speakers. I request the audience to hold their questions until then. I urge the speakers to please stick to their time limits. The buzzer will ring at 18 minutes. I request the speakers after their talks to be seated on the dais for the question and answer session. I now request the session speakers to kindly take charge of the session.
Welcome to this last session. Last but not the least, we have some really impressive speakers here. It is my immense pleasure to first invite to the stage to deliver a talk, Dr. Dheeraj Kumar, Group Leader of Cellular Immunology at ICJB New Delhi, whose research focuses on frontier areas to understand the pathogenesis of tuberculosis with a very unique perspective towards developing novel host-directed therapies. His group utilizes a multidisciplinary approach involving various cell biology, molecular biology, microbiology, immunology, and computational biology tools. His particular work on autophagy highlights the need to balance inflammatory and metabolic aspects while developing HDTs using this pathway. At the same time, his work on infection-induced alternative splicing has opened up a very new field of investigation for host pathogen interaction. At cellular level, his works have uncovered unconventional cellular niches of TB that act as confounders towards the efficacy of drugs and vaccines. His research contributions have been highly cited and recognized through various national and international awards and grants, including the distinguished Shanti Swarup Bhatnagar Prize in the year 2019. And Dheeraj will be talking about stitching together in a board for the survival, for its survival by MTB. Welcome, Dheeraj.
Thanks, Deepak. It was a long, and I think whatever was written initially was spoken about. Anyway, thanks, and thank you, organizers, Raman, Nishit, and THSTI for organizing such a wonderful meeting. I had to miss some sessions and some talks, but that's different matter. So what I'm going to do, this is the last session, and one advantage of talking in the last session on this tuberculosis symposium is that I can really skip the introductory part of tuberculosis that has been done and dusted over various sessions so far. This everyone is aware of. This is the tuberculosis granuloma, which I'm showing here. What I'm trying to show today is, different ways through which bacteria actually brick by brick builds its abode inside the host. That is at the molecular level, at subcellular level, and at the cellular level. And I'm sure, you all appreciate that there are multiple mechanisms which facilitate tuberculosis pathogenesis in the host. So the broad research goals in my lab actually looks into all three aspects that was shown in the previous slide. We look at the cellular level, where we look at granuloma compositions, different cell types which are recruited and kind of harbor the bacteria and how they respond to different kinds of treatments, vaccinations, and things like that. We look at subcellular levels, including autophagy and inflammation, different compartments and trafficking and signaling inside the infected cell types, immune cells. And at the molecular levels, we look at gene regulation, particularly nowadays focusing on alternative splicing upon MTB infection, which has emerged over the last 7-8 years as a very, core, research focus in my lab. So what I will do today is talk a little bit about this work, the alternative splicing, and a little bit about the unconventional cellular niches that we have been doing over past few years. So a few years back, we had shown through our RNA-seq studies that there is massive alteration in the pattern of RNA splicing in MTB-infected macrophages. And the important point was that all important functional classes, the innate immune classes, which you would expect to be involved in antibacterial host responses, were being affected through this process of alternative splicing. And to simply summarize the overall observation through this study was that if this is the full length splice variant of a protein, which is part of one of the innate immune pathway, which can lead to bacterial killing, what's happening during bacterial infection, you get a splice variant which is truncated, that may not form a functional protein, that may get degraded very fast and doesn't get translated, or basically, you will not have this protein sufficiently, and that will lead to bacterial survival. So while we were talking about this, and basically, if this is how the host response looks like, this is what the MTB impact does, it just based on your host response. So while this study was being done, there were a lot of questions asked by reviewers at that time. What is the mechanism? And looking at the process of RNA splicing, we were a little, kind of convinced that it has to do something with, some bacterial protein coming out and doing something with the host RNA splicing process, considering it is a really multi-step process involving large multimolecular complex assembly that is called a spliceosome. So we came up with a high throughput and candidate-based approaches to understand the mechanism. And this was supported by India Alliance to carry on. It was a challenging work. But what I will not get into is how we reached here. But we did high throughput experiments. We identified interactions between mycobacterial secreted proteins and host RNA splicing proteins, and we identified interactors from there. And then from there on, built on that, what is the mechanism through these interactions are actually impacting host RNA splicing. And what we actually observed, and for that we need to go back to biochemistry class 101. On how RNA splicing happens. There is a complex called snRNPs, which is small nuclear ribonucleoproteins. It's both small nuclear RNAs and combined with certain proteins, which together forms the snRNPs. And these are the fundamental part of all spliceosome complexes. So for snRNP assembly to happen or biogenesis to happen, these snRNAs are synthesized in the nucleus. These are transcribed in the nucleus, and then they are exported to the cytosol. And in the cytosol, they interact with SMN complex. It's a multimolecular complex, forms a symbioosome, and then that leads to assembly of SM core ring around it. And this forms a kind of snRNPs. And these snRNPs then goes back to the nucleus, where they participate in the spliceosome complex formation, leading to RNA splicing event. So what we actually observed is that a lot of MTB proteins, and again, through yeast to hybrid, through co-immunoprecipitation, and through functional studies, what we showed is that these lot of MTB proteins are actually interacting with SMN complex and SM core ring components. And these interactions were actually happening in the cytosol because snRNP biogenesis requires these RNA to come out to the cytosol and then interact with the snRNP components there. And just to give you some examples, one of the important aspect is of this USNRNPs, you have these snRNAs, which binds to SMN complex and SM core rings. We found direct interactions between one of the snRNP SM core complex protein with mycobacteria, beautifully decorating the bacteria inside the cytosol. Again, this is happening in the cytosol. And also, this is fish with one of the snRNAs, and you can see even snRNAs directly binding with bacteria. So what we actually concluded from there is that for the snRNP biogenesis, bacteria has, the RNA has to come out into the cytosol. That's where it gets sequestered by certain bacterial proteins in the cytosol. And once that happens, where snRNP biogenesis is disrupted, and when snRNP biogenesis is disrupted in the field of RNA splicing, it is very well known that that will have global impact on splicing of those genes which are getting actively transcribed. So, we identified some proteins, and we have, we identified some proteins and asked Vinay, who is a collaborator and great friend, to make a knockout of one of those proteins. We showed experimentally that once you make the knockout of that protein, so one of the protein we named as HSR1, which is host splicing regulator 1. So when you make the knockout, you lose the impact of RV-induced RNA splicing that happens in the wild type cells. In the knockout, you don't see that splicing event. And at the same time, it also shows the decline in the survival inside THP1 macrophages, as well as in the animal models. So suggesting that once you lose the capability to alter host RNA splicing, you also lose to some extent your survival and virulence inside the host. Not only that, we actually went to human tuberculosis granulomas and looked for one of these snRNP complex proteins that we're identifying directly interacting with bacteria. And as I can show here, is that it's, tuberculosis granuloma is strongly stained with one of these SNRP complex proteins. And, in fact, multinucleated giant cells, they are showing cytosolic retention of this SNRP complex protein in the tuberculosis granuloma, something that we are also seeing in vitro in macrophage models. So again, very, excellent, correlation observed here. And finally, we went to a small cohort data of, pulmonary tuberculosis and healthy controls, around 20 or 19 odd, individuals, and looked at monocyte RNA-seq to understand what kind of splicing events happen there. And it was amazing to see that the splice variants were able to distinguish PTB versus healthy controls across all the functional classes that you would see: RNA splicing, metabolism, immune response, and inflammation. So, what basically we conclude from here is that MTB has found a way to alter host RNA splicing by disrupting the SNRNP biogenesis. And this is amazing because SNRNP biogenesis, for it to come to the cytosol, is an evolutionary conserved mechanism. In all the eukaryotic system where splicing happens, SNRNAs have to come to the cytosol. That's a quality assurance mechanism. And MTB utilizes it and exploits it to alter the host RNA splicing. So what is the general host response where you could actually get a fully functional proteins in case of in the presence of MTB infection? What actually you see is an altered splice variants, and that actually eventually helps the bacteria to survive and grow. Something like actually stitching the host responses the way the bacteria desires. So, that was very quick summary of the RNA splicing thing that we have done. Moving on. Basically, what we are trying to show is that host RNA splicing is a new paradigm in host pathogen interactions. And we also have worked on some ways to block the interaction between MTB and host protein, which are regulating the RNA splicing, to see whether you can achieve, some therapeutic targets through that. So the other aspect that I will quickly touch in last 2-3 slides is some work that we published few years back, where we showed that mesenchymal stem cells were a preferred niche to Mycobacterium tuberculosis. And what we had shown there is that the stem cells, when the bacteria is present inside these cells, they actually tolerate drugs like isoniazid and rifampicin. They are also tolerant to inflammatory cytokines like IFN gamma and TNF alpha. Having known that how different vaccines work, we had this question that, whether that would, one of the reasons why it impacts efficacy of different vaccines, which are at different stages of clinical development. And this we also showed in human lung biopsy samples that you do see, bacteria in mesenchymal stem cells in the lungs. But one observation that we had overlooked initially, and later on went back to understand what's happening, was the role of neutrophils in tuberculosis granulomas. And when we started looking at it, if this is how your bacterial CHU looks like over the time, we took out the lungs, made single cell suspensions, and sorted different cell types from the lungs of infected animals. So these markers are not very well visible here, but this is where the PMNs are coming, which is Ly6G, CD11B positive. And then we did CFU from individually sorted cells to see that which compartment has more bacteria or less bacteria. And I don't need to say that where you see the maximum number of bacteria present in the lungs. They're actually the PMNs, which are very high. If you look at the scales, so it's almost a log order more bacteria present in PMNs. So we, of course, had a question that does high neutrophilia associates with disease pathology? And also, can our neutrophils really the Frankenstein within, which actually kind of promotes more bacterial pathogenesis rather than helps the host to control the infections? So we went further and did a couple of experiments. So one of the experiments that we did was BCG vaccinations, where animals were vaccinated intradermally, and then we followed over certain times and looked at bacterial population again in the 3 different populations that we had looked for. And to our surprise, what we found, of course, the bacterial population were declining in BCG vaccinated animals across all the groups, whether it is PMNs, macrophages, or stem cells. But you can see is that there is still maximum number of bacteria remaining in neutrophils rather than in macrophages and stem cells, suggesting that neutrophil infiltration could behind the the reduced efficacy of BCG among adults, something which one needs to test it. I know we have only animal data. So following that, we took another model of severe pathology in high neutrophilia, which is IFN gamma knockout mice. So we took IFN gamma knockout mice and again followed them for for 10 weeks and just observed at how bacterial distribution across different cell types happens. And one of the easiest thing was that you see a lot of neutrophil infiltration. Sorry for this, I think it has lost the resolution. We could see through histopathology, through immunostaining, as well as single cell sorting. You could see maximum number of PMNs almost going 1.5 log higher in hyphen gamma knockout mice. We were asking the question that what drives the neutrophil infiltration? And we did a Luminex assay to see, and that was done from the lung homogenates of these infected animals. And what we observed among many cytokines that we looked for is that IL-17 was very high in these animals, the hyphen gamma knockout animals, which were infected. And then we actually developed an experiment where we neutralized IL-17 using IL-17 neutralizing antibody. We had also used COX-2 inhibition. I'm sorry, because of time constraint, I didn't get into how we went to COX-2. So there is data to support why we use COX-2 inhibitor. And we started with IL-17 neutralization 2 days before infection and followed up exactly in 2 arms, one which got the neutralization antibody, another which didn't. And then we had plus minus celecoxib group to look for that. To summarize the whole observation, what we observed is that you can reduce neutrophil recruitment, you can reduce neutrophil resident bacteria, and you can enhance animal survival, even if it is hyphen gamma knockout mice, if you can block neutrophil recruitment. That's the level of impact neutrophil recruitment is happening. And the driver is actually IL-17. We are trying to understand from where this IL-17 is coming. We have some data, but still it's under progress. While we were doing this, a month back, we hosted a Report India meeting at ICGV New Delhi, and I was listening to a talk from Dr. Subhas, who given a talk on day 1, and he was talking about a cohort study where he looked at successful treatment cases and treatment failure cases in a large cohort. And the treatment failure cases were interesting because the treatment failure was not because of drug resistance. So certain immunological parameters were what driving these treatment failure in these individuals. So I asked him to share the data, and he was excellent. I mean, he shared the data immediately that what we were looking for. And the observation is that the cases where you see treatment failure at baseline, before they were started on the treatment, just when they were diagnosed, they had very high IL-17, very high absolute neutrophil count and neutrophil to lymphocyte ratio, suggesting that IL-17 neutrophil axis actually works tremendously in tuberculosis pathology, and it actually provides a new, a dimension which can be targeted for host-directed therapy. So to summarize, I didn't talk about the subcellular aspect where we look at autophagy related thing. But what we certainly know is that bacteria really meticulously craft its abode by manipulating at different levels how it has to set up the granulomas inside the host. And if we can unravel these cellular, subcellular, and molecular mechanisms, there are tremendous opportunities to develop novel host-directed intervention approaches. So I will stop here. I will acknowledge the people who have worked. Some of them are not seen in this picture, which is on excursion on the Himalayan side in winters, so that we do some snow trekking. And collaborators, of course, a wonderful group of collaborators who help us at different stages, like Vinay and Amit Singhal. Subhas has been wonderful in sharing this data, which he has generated over the last few years, and funding from different funding sources, including India Alliance. Thank you very much for your attention. We'll be happy to take questions.
Questions for Dheeraj?
Oh, okay.
Sorry about that.
Okay. So
Dheeraj, come have a seat with us. We're going to move on to the next speaker and do a bunch of questions later. It is a pleasure now to invite to the stage Dr. Sheetal Gandotra. Sheetal is an accomplished scientist in the fields of immunology and microbial pathogenesis, serving as a principal scientist at CSIR IGIB and assistant professor at the Academy of Science and Innovative Research. She's made significant contributions to the scientific community through her research on TB. Actually, her paper in Nature Medicine from 15-ish years ago with Sabina Ert is, in my opinion, one of the more important papers in the field of molecular genetics of TB. Her current research focuses on lipid metabolism, immunometabolism, and microbial pathogenesis. She's received numerous grants and awards for her work, including the prestigious Swarna Jayanti Fellowship in 2019 from the DST, and has published more than 30 papers in her career. Welcome, Sheetal.
Thank you. Thank you to the organizers. Thank you, Nishit, Raman, Dinesh, Krish, and Amit for organizing this wonderful meeting. It's great to meet the fraternity here after such a long time. So thank you for this opportunity. So my lab works on these beautiful intracellular organelles, the red ones, which are called lipid droplets. So this is what we're all accumulating after that lovely badam, of that lovely dal halwa. And if I fall while giving this talk, it's the food coma that you can blame. All right, so I'm going to start off by telling you why it is, actually my whole talk is going to be not about these organelles per se, but why we study these organelles in the context of TB infection. So the way I think of tuberculosis and the challenges in tuberculosis is thinking about it as immune response, particularly the inflammatory response gone too far. So in a normal self-resolving infection, you would have the infection rising, and then there is a control of infection. And alongside that, you have recruitment of inflammatory cells that peak, and then there is a resolution phase. And there are lipid mediators like leukotrienes and prostaglandins that help in making this development of this immune response, and then lipid mediators like lipoxins and resolvins that help in resolving this immune response. In case of tuberculosis, things go awry. So there is in susceptible individuals, there is a high neutrophilic infiltration, which is very well known. There is a burst of pro-inflammatory cytokines which promote that neutrophilic response. And really what we are trying to do is, by studying these immune responses, here, I mean, the relevance of why we study these immune responses is because there is a relationship with the inflammatory response and this condition called pulmonary impairment after TB. So in individuals who have been treated for tuberculosis, across different studies, there can be anywhere between 50% to 80% individuals showing pulmonary impairment with scars, restrictive ventilation or obstructive ventilation, nodules, and generally a poor quality of life. So inflammation is the reason for why these patients have to suffer for the entire life. So what the questions that motivate us are: what drives tissue pathology in TB? And are there active manipulations that can be reverted? So we go back to looking at the granuloma, which has been talked about by a lot of people. I'm just going to bring your attention to these cells called foamy macrophages. So the reason why they're called foamy is because when you do an H&E section, what you see are holes like you see in a foam when you cut the foam. So these holes don't stain with either hematoxylin or eosin. And they're common not only in conditions of primary tuberculosis, like the examples shown up here, but also in post-primary tuberculosis, where you have the development of necrotic granulomas. These necrotic granulomas can be standalone. They can be coalescing, caseating granulomas. Some of them can also be present as a fibrotic wall, which is what probably gives the scarring in TB lesions. In post-primary tuberculosis, you can also have bronchial occlusion or alveolar occlusion by these granulomas. And just this could, these occlusions could be filled with just foamy macrophages. There could be fibrin accumulation, or there could be caseation within the alveoli. So the granuloma in primary or post-primary tuberculosis, it contains these fat-filled foamy macrophages, but they can be present in many different formats. So because TB genome is enriched with genes that have that show that the organism is a fat-loving organism, there has been a lot of interest in understanding whether it's TB-derived metabolites or TB that is driving the formation of these foamy macrophages. And over the years, a lot of studies have actually been focused on understanding this. So very early on, trehalose dimycolate, which is a complex lipid on the cell surface of mycobacteria, was shown to induce this foamy macrophage phenotype when injected in vivo. Ketomycolic acids have also been shown to induce this phenotype in macrophages, and work done in InTech in Dr. Pavan Gupta's lab showed that the nuclear receptor TR4 was important for this ketomycolic acid-driven foamy macrophage formation. Work done at ICGB in Kanuri Rao's lab showed that ESAT6, by driving production of ketone bodies via cyclic AMP production, lead to inhibition of lipolysis, and that's what leads to the lipogenic program in the host cell. A lot of extensive work done from K.N. Balaji's lab at IISc showed several epigenetic factors which favor lipogenesis in macrophages as a result of MTB infection. However, what came up intermittently while all of this was also getting published was the fact that TB alone in some models was not able to induce foamy macrophage formation. So this is work from Kolatakuddi's group, where he shows that you need hypoxia to be able to generate foamy macrophages in TB-infected in the TB-infected condition. Sarah Stanley's lab showed that you need interferon gamma, which via a HIF1 alpha response leads to the foamy macrophage formation. And then more recently, TNF alpha was shown to be responsible for this lipogenic response. So there's one phenotype, but too many contenders. Around the same time, we also were working on this. And what we found is that in THP1 macrophages, and also in human macrophages, we didn't see this response. So the work that I'm going to talk to you about today is work done by two graduate students, Neetika Jaisinghani, who is right now in Jessica Seeliger's lab, and Stanzin Dava, who is in Emory, also working on tuberculosis. And the questions they asked were, what are the determinants of foamy macrophage formation on TB, and are foamy macrophages relevant to the outcome of infection? So as I mentioned, when you culture blood-derived monocytic cells into macrophages in vitro, these cells spontaneously develop lipid droplets. And when we infected macrophages with Mycobacterium tuberculosis at a low MOI of infection, this is now data from 4 different individuals, we didn't see a lipogenic response. We didn't see lipid droplet, an increase in lipid droplet size as a result of infection. We did the same thing in THP1 macrophages, and lo and behold, in MOI1 and MOI5, we don't see such a lipogenic response. But interestingly, what we started seeing is if we infected bacteria at very high multiplicities of infection, so this is 50 bacteria per 1 macrophage, we start seeing an increase in the lipid droplet abundance. So what you're seeing here is a lipid droplet size binned on the x-axis and the frequency of finding these droplets. So as you move from left to right, you have an increased abundance of lipid droplets of the larger size, and that's happening at MOI 50. So that was also validated at the level of rate of triglyceride synthesis, as well as total triglyceride levels. So no change at the lower MOIs, but an increase at MOI 50. And you know what happens if you infect macrophages with very high multiplicity of infection is you get necrosis. So we had a very nice correlation between the percentage of necrosis happening as a result of infection and the amount of triglyceride that was accumulating in cells that withstood that necrosis. So we're talking about a bystander response now. So we wanted to validate, or we wanted to address whether necrosis was the reason. So we decided to use a strain of Mycobacterium tuberculosis, which is unable to cause necrosis. So if you have propedium iodide positive cells, you have necrosis happening. If you have annexin 5 positive cells, you have apoptosis happening. So H37RD1 deletion mutant is a strain which is known not to cause necrosis and instead induce apoptosis. So in our hands as well, we found apoptosis happening with this strain and less necrosis. So we asked if this strain could also induce, could also lead to the necrosis-induced phenotype that we were seeing in case of the wild type. So at high multiplicity of infection, this strain was unable to cause increase in lipid accumulation in the bystander cells. We also tried a pharmacological method, and I have to say that this was a marginal decrease in necrosis that we could achieve with this inhibitor. So this molecule is IM54, and it inhibits ROS-mediated cell death. There was a marginal decrease on cell death, and there was somewhat recovery of the triglyceride synthesis that was happening as a result of high MOI. So with this, we concluded that MTB-induced necrosis triggered lipid accumulation as a bystander response. So next, we wanted to see whether this was happening in an in vivo infection. So for this, we turned to the guinea pig model of infection, and Dr. Tyagi and Garima helped us get these mouse tissues. So if you see, so we looked at 2 time points, and the choice of these 2 time points was because we had an understanding from their previous work that the granulomas at week 4 are more solid granulomas, and the granulomas at week 10 are usually, there's a shift from solid to necrotic granulomas at that point. So we stained these lung tissues with Olredo, which is a stain for neutral lipids. And what we found was there was a beautiful correlation between the presence of necrosis, whether at week 4 or week 10, with Olredo positivity. So very good correlation. And this actually was also found in a previous study in lymph node tissues. So in vivo, there is a strong correlation between foamy macrophages and necrotic granulomas. So we wanted to model this, and to do that, Neetika chose a cellular model, which was followed up later by Stanzen and using an in vivo model. So several of the phenotypes that I'm going to talk about, the upper part is going to be the cellular model, and the lower part is going to be the in vivo model done at 2 different times. What Neetika developed is a very simplistic model. So in this model, we cultured THP1 macrophages, we harvested them and made a necrotic supplement from those by mechanically freeze thawing them. And these necrotic cells were then added back to healthy macrophages. And what we found is there was a time-dependent increase in the neutral lipid accumulation. So the neutral lipid here is being stained by this dye called Budipi, just like the previous slide. And we saw a time-dependent increase in the lipid accumulation. So we asked whether we could use an in vivo model that would be more easily tractable than a guinea pig model for any intervention studies that we want to do. So we turned to the C3HEB-FEJ mouse model, and using the high dose of infection, we achieved necrotic granuloma formation by day 28. And I do want to point out that it's not that every granuloma at this stage is necrotic. We have as low as 2 granulomas in a single lung lobe being necrotic, while there might be 10 to 8 other non-necrotic granulomas, whereas there would be mice in the same group which may have even 12 to 20 necrotic granulomas. So there is huge heterogeneity, but we start seeing the development of necrotic granulomas by day 28. And when we stain these granulomas with Olredo, we find that the infiltration that is happening at day 14 is completely Olredo negative, whereas we start seeing the development of Olredo positivity at day 28. And you get these beautiful structured granulomas, which look like human TB granulomas. So now the question is, what is the role of this stored triglyceride during infection? So we knew from the macrophage model that if you knock down DGAT1 gene, so DGAT1 is a diacylglycerol acyltransferase 1, converts diacylglycerol to a triacylglycerol, and this happens at the ER membrane. So if we silence this gene in THP1 macrophages, we essentially ablate the cells of lipid droplets, and we can decrease the amount of triglyceride. And even when you supplement the cells with NCS, these DGAT1 silent cells do not store as much triglyceride. So we wanted to address whether DGAT1 was important for lipid storage in the mouse model. And for that, first of all, we looked at the expression level of 2 DGAT genes that are present in the mouse, which is DGAT1 and DGAT2. We see an increased expression of DGAT1, but no change in the expression of DGAT2. These data are at day 28 post-infection. So we decided to use a pharmacological inhibitor of this enzyme DGAT1, which was previously described. And there's been a lot of interest of the pharma industry, particularly, to look for DGAT1 inhibitors. And that's because, there is a lot of investment in the area of obesity. So you want to have lipid-lowering drugs for that purpose. But anyways, we chose this molecule because of its very good bioavailability properties, but we chose to do an intravenous injection, and I can talk about that later. So we treated mice between day 15 and day 28 and found that indeed there was a lowering of the Olredo positivity in the T863 treated animals. The name of the compound is T863, and that's quantified here. So great. So now we know that DGAT1 is at least important in vivo in producing this foamy macrophage phenotype in the MTB infected lungs. So now coming to the million-dollar question, impact of triglyceride on intramacrophage MTB growth. So in our THP1 model, you're looking at the left, the NAFM model. So that's the necrosis-associated foamy macrophage model that I spoke about. There is no difference in bacterial growth, whether the cells are the normal macrophages or they're induced to accumulate more triglyceride. Even if you knock down DGAT1, there is no impact on TB growth. So we decided to do this in mouse bone marrow derived macrophages, and it didn't matter how you stimulated the macrophages, whether you used IL-6, interferon gamma, or IL-4. Basically, the levels of triglyceride did not matter to the intracellular burden of Mycobacterium tuberculosis. But things got interesting in vivo. So what we found is that about 50% of our mice had about 25 to 97% reduction in the bacterial burden. So there is a huge heterogeneity, but it's statistically significant because about half the animals are giving this stronger phenotype. All right, so now we were interested in finding what's happening in these mice such that they're responding to treatment in a certain way, whereas these mice are not. Or what are the correlates of the decreased bacterial burden? So we decided to do RNA-seq, this is of whole tissue, and we looked for, so we decided to bin the data. So we had the vehicle-treated animals, we had the TH63-treated animals. We binned the data into the high CFU group, which we call the TH63 high group, and the low CFU group, which we call the TH63 low group. And when we compared the TH63 versus the vehicle group, we found no differentially expressed genes. So TH63 on its own was not doing anything universally different than just the vehicle control. However, there were differences, and very few differences were found actually between the TH63 high group and the vehicle control group. But the TH63 low group versus the vehicle group or the TH63 high group, we found quite a lot of statistically significant differences at the level of gene expression. So what are these differences? So we had decreased number of genes or decreased expression of genes involved in neutrophil degranulation, signaling by interleukins, genes involved in innate immune signaling, activation of matrix metalloproteases, and an upregulation of genes involved in elastic fiber formation and muscle contraction. So all the determinants of pathology were actually downregulated in the TH63 low group, whereas all the determinants of the healthy lung tissue were upregulated in the TH63 low group. So we got very excited about the kind of genes that we were finding in the signaling by interleukins and the innate immune system. And these were cytokines. We got excited because this is exactly what Neetika had found in the cellular model. So increased triglyceride level, you get increased TNF, you get increased IL-1 beta, you get increased IL-6, you decrease the levels of triglyceride, you get a decrease in these cytokines. The same was happening in the TH63 low group. Look at the stark difference within that group. But what you can see is there is a high heterogeneity here. There are animals that have as low levels of cytokines as this group, but then somehow this group is different, right? Because it's controlling. So there must be other determinants as well. So we went back to looking at neutrophils. And again, 50% of the animals responding by lowering the levels of neutrophils, and in the same animals, lowering the levels of the acid-fast bacilli. So this is the cyber gold staining that I was referring to on the first day of this meeting. This is to look at bacterial populations by acid-fast staining. And there was a very nice correlation between the cyber gold stain and the myeloperoxidase stain, which is used to stain the neutrophils. It's very well established in using outbred stains of mice that the resistant mice like the C57 black 6 mice have very low levels of neutrophils. It's more a lymphocytic granuloma. One more minute, Deepak. And the super susceptible or the susceptible mice have an increased abundance of neutrophils. And it was shown by Anna Gara's group that if you neutralize neutrophils, you get a decrease in bacterial burden. So we were very happy to see that the neutrophil burden was associating with bacterial burden. So what leads to neutrophil production in the susceptible mouse model has been described as a type 1 interferon response. We looked at the interferon beta levels, that was indeed lower in the TH63 low group, but then there are other determinants of neutrophil recruitment such as eicosanoids. And triglycerides being storage houses of fatty acids, they could be determinants of the levels of eicosanoids. And indeed it's been shown that macrophages produce certain cytokine, certain eicosanoids dependent on the triglyceride levels. So we were very keen to see whether that was happening in vivo. So we used a mass spec-based approach to identify several eicosanoids. And to cut a long story short, we did not find a correlation between TG levels and eicosanoids. On the other hand, we found that the triglyceride lowered groups actually had a higher level of some of the eicosanoids. And interestingly, it is the differentiation within the TH63 high and the TH63 low group at the level of these eicosanoids that might be playing an important role, because these are pro-neutrophilic eicosanoids. So to sum up, we think that foamy macrophages are formed during the course of infection as a result of necrosis that is induced by MTB, and they help in promoting the inflammatory response by producing pro-inflammatory cytokines in the type 1 interferon response that leads to recruitment and activation of neutrophils. So is that a great strategy or a great way forward to lower triglyceride levels? I think not, because DGAT1 has a very important role to play in the human gut, and DGAT1 inhibitors have lost out on the race even for obesity because they cause diarrhea. So what's the alternative? The alternative is to target eicosanoid imbalance with non-steroidal anti-inflammatory drugs. And there is a huge trial going on in Europe on that. Targets host signaling involved in inflammation. So this compound was mentioned yesterday, which is a phosphodiesterase 4 inhibitor and lowers inflammation. You heard Vivek's talk today on lowering the type 1 interferon response. But what my lab is focusing more specifically on is macrophage lipid droplet related functions. And the lipid droplet is not just a blob of fat made by triglycerides. There are a lot of proteins that decorate this. And we found that the lipid droplet surface is actually actively manipulated by MTB. And the kind of questions that we're addressing now is how are these proteins localizing to the lipid droplet differentially during infection? And what role do they play in lipid metabolism and innate immune response? So with that, I'd like to thank the funding that we've received very generously over the years. I'd like to thank all the members of the SGWave lab. I'd also like to thank all the facilities that have helped us do this work, and Dr. Tyagi and Garima for the guinea pig tissues, and David Sherman for the RD1 deletion mutant. And I'm sorry to cross over my time.
Hi, and the next speaker is Dr. Varadarajan Sundaramurthy. He has a long-standing interest in host pathogen interactions. He started his lab in NCBS, that is in Bengaluru in 2014, and his research interests have been centered around understanding the alterations in fundamental host cellular processes during intracellular MTB infection, and use this knowledge to host-directed therapeutic approaches against TB. So Dr. Varda.
Thanks, Balaji. And yeah, it's thanks for the organizers of this wonderful meeting. It's really good to see all the TB people in one room. And thanks to everybody for staying for the last session of the last day. So today I'll talk to you about modulating mycobacterial physiology in an in vivo context during infection. So this work was started by Manisha when she was in my lab, and continued by Nawaz and Kushi. And we have collaborations with Praveen, Sadamini, and Shandil. And funding agencies, I thank them for their support, and the fantastic facilities we have in NCBS, BSL-3 imaging, and other facilities. Right. So the major theme of my lab is to understand pathogen-mediated reprogramming of host cell functions. And we study this at multiple levels, looking at tissues and organs, cell, subcellular, and molecular levels. And over the years, most of our work has been targeted on the cellular pathways, with some focus on the tissue scale and tissue scale modulations during the pathogen infections. Right. A lot of speakers before me have given wonderful introductions to the systems that we are studying. And just before me, Dheeraj spoke very nicely about how these multiple levels are coming together layer by layer to generate the pathogenesis mechanisms and outcome. So some of the previous work in my lab, we have studied looking at the cellular determinants of Mycobacterium tuberculosis, uptake, intracellular survival, and balance. We showed that there is a significant heterogeneity or a cell-to-cell variability in the endocytosis of the host cell. And that is something that is subsequently determining which cell is going to get infected. So we all know that in a phagocytic assay, some cells get infected, some cells don't. And we could show that this is directly related to the cell-to-cell variation in the endocytic capacities. And this is also somehow directly related to the cellular content of the lysosomes. Now, we all know in the mycobacteria field that in cell culture models, Mycobacterium tuberculosis avoids the delivery to lysosome, and the mechanisms of the phagosome maturation arrest is very well studied. What we have also seen in our lab is that beyond the confines of these mycobacteria containing phagosome, there is a global reprogramming of the lysosomal machinery during infection, right? So we could show that there are specific mycobacterial components, which I'll come to in a bit, which is directly modulating the biogenesis of lysosomes. So we see a lot more lysosomes in the MTB infected cells compared to the non-infected cells. So that is something that is also summarized here, something that we published some time back. What we see is that mycobacterial infection itself is inducing the biogenesis of lysosomes. And this is happening using mycobacterial surface lipids. Some of the prominent lipids, such as sulfolipids or PIM6, alone on their own in purified form, are able to induce the biogenesis of lysosomes. Right? Now, the mutant mycobacteria, which do not make sulfolipid in this case, this enhanced rewiring does not happen. And what is very interesting for us is, as a consequence of that, there is a increased survival of mycobacteria. So these mutant mycobacteria, they survive much better. Actually, this is something that was also shown earlier, but we are seeing this in the context of the lysosomes. So there is also a reduced co-localization of the mycobacteria to lysosomes in the case of the mutant mycobacteria. So we think there is a kind of a reciprocal influence of mycobacteria and the host cell lysosomes. Now, as we were doing this, we really got interested in this phenotype of enhanced mycobacterial survival. And that is something I was really happy to hear talks from Amit Pandey, that there are also several mutants in the absence, some genes in the absence of which mycobacteria are surviving better. So we started thinking that, we have heard over and over in the course of the last few days that there are so many mechanisms that are responsible for the survival of mycobacteria inside the host cells. And we are thinking that these kind of phenotypes, which are pro-infective phenotypes, can offer us significant clues about the mechanisms that are operating in the host cell that are restricting the survival of the mycobacteria. What can we know about these mechanisms? So that's a sort of a broad question that we are addressing. And we also, many years ago, Amit is here, there is many labs started developing fluorescent reporter strains that are giving us significant insights into the physiology of the mycobacterium. So here, Amit's lab nicely showed that they developed a redox sensor that is giving us the redox status of the mycobacteria by measuring the fluorescence. And you can see at different subcellular locations, the redox stresses encountered by mycobacteria are different. And similarly, few more examples from David Russell's lab using different kind of fluorescent reporters, they can get a sense of the kind of pH and chloride stress or nitric oxide and hypoxia stress that individual bacteria are experiencing in their different niches. So these reporters are also giving us fantastic tools where we can start addressing not just whether a particular compound or a drug, how much it is killing bacteria,
but also in what ways they are altering the bacteria, right? I think these are important points that need to be addressed. We also know from several screens that have been done with, for example, here is a host-based high-content screening platform where typically after infection chemical libraries are added, and then the phenotype is measured by either automated imaging or a kind of a plate reader. You get a variety of phenotypes, and the most preferred and the most common phenotype that is followed up are the kind of phenotypes where the bacteria are killed. But in many screens, you also end up with phenotypes which leads to enhanced mycobacterial survival. Usually these compounds are sort of ignored. But for example, here in the screen that we did long ago, we can see that there's a whole bunch of compounds which is resulting in a enhanced mycobacterial survival. So a possible idea is that given the prevalence of these compounds, are there so many mechanisms that are restricting mycobacterial survival in vivo survival? And can we use these compounds as tools to start understanding these mechanisms? And if we combine them with these fluorescent reporter strains, can we start also inferring the effect this is having on the mycobacterial physiology? So that's the kind of a broad motivation of what I'm going to talk to you today. So that's what I said. So growth restriction is a major player in control of TB. And of course, understandably and correctly so, we all need compounds and phenotypes that are drastically reducing the mycobacterial CFUs. But for the reasons that we have been hearing through these days, and what Anil Kaul nicely mentioned in the challenges that we face in eradicating TB, in the context of the different heterogeneities that we encounter in both the host as well as the mycobacterial, the factors like the accessibility of the drugs to the different sites of location, the different stresses that are induced by the host system at cellular, subcellular, tissue niches. All of this has distinct impact on mycobacteria very, very locally at the level of individual mycobacteria. And CFU assays are going to tell us that there is this much reduction, but readouts of higher sensitivity and readouts at single bacterial level, which are going to also tell us about growth restriction phenotypes, we think are going to be quite important, right? So what we are trying to do is to use a panel of reporter lines and examine the status of some of the compounds that are coming out of various screening approaches, and look at the correlation between the CFU and the physiological status at the infected tissue level. So this is a work that was started by my student Manisha when she was in the lab. So we took a bunch of compounds from previously published screens that were sort of hinted to have a pro-survival phenotype, and we screened them in a intracellular mycobacterial infection assays. These are 2 replicate screens. And you can see here that these 2 compounds were showing an increased mycobacterial survival phenotype inside the host cell. So we also then confirmed this by CFU assays, and also could validate them in an in vivo infection model, where after infection, you take the single cell suspensions and you could see that there are a lot more infected cells in vivo. And also by CFU, we can see that there is a pretty significant increase in the intracellular, in vivo survival of mycobacteria, right? So then, of course, the question is, what are the mechanisms that are driving this? And what can we infer from these pro-survival compounds? So we did some preliminary docking studies, and looking at a conventional target like COX-2, for example, we could dock the structure to its known target. But what was also interesting was that such a kind of a binding motif was also predominantly present in several other proteins. So this suggested that maybe there are additional potential cellular targets. And we all know that although we would ideally like to talk about single drug and single target, polypharmacology is very common. So we thought that maybe what is the most directly relevant target in this context. So to look at that, we took a biochemical approach. We biotin tagged the compound and pulled down the biotin with the streptavidin beads. And here you could see that with both the versions of the tagged compounds, we could pull down specific bands. And then by mass spectrometry, we could identify a bunch of proteins that were binding to them. Now, this protein ASS1, which is an enzyme in the arginine biosynthesis pathway, arginosuccinate synthase, was coming up as the most prominent target. And going back to the docking studies, we could see that this is the ASS1, and these are the substrates, two substrates, citrulline and aspartate, and this is the ATP binding pocket. And we could see that this compound could bind very nicely to the ATP binding pocket of ASS1. And to validate this, then the pull down lysates, we could confirm the interaction with an antibody. And so there's a negative control that did not show any strong binding to the beads. And with a purified recombinant ASS1, we could see that there was a strong inhibition when we added this compound. And similarly, from the macrophage cell lysate, when we treated with the compound, there was a significant reduction in the activity of ASS1. So we think that this compound is significantly having an inhibitory effect on the ASS1. Right? But as I told you that there could be multiple additional cellular targets. So we wanted to now check if the ASS1 is directly involved in the phenotypes that we are interested in. So by silencing ASS1, we could see that there is an increased mycobacterial survival. And also by CFU assays for the silencing, we could see that there is an enhanced survival of mycobacteria. So clearly there is the effect, ASS1 is restricting intracellular mycobacterial survival. And to confirm this again, we took an independent inhibitor of ASS1. And when we did the CFU assay with this inhibitor MDLA, again, we can see a substantial increase in the survival of mycobacteria. So what then is the link with the enzyme and the phenotype that we are seeing? As we know, and we heard from Bill today, arginine is crucial for macrophage function and immunity. And we know that arginine is the major or the only nitrogen donor for nitric oxide. And in macrophages, in fact, ASS1, then the subsequent enzyme arginosuccinate lyase and nitric oxide are often seen together. So we thought that maybe inhibiting ASS1 can have an effect on nitric oxide. And that is exactly what we see, that there is a reduction in the nitric oxide. And if this is inhibited, there is also going to be the impact on the other arm, which I will come to in a bit. So this immediately made us to think that, you know, the reporter line that David Russell has published, HSPX GFP reporter that is very sensitive to nitric oxide stress. So we infected mice. So this is the way this reporter works is this is HSPX, the promoter is sensitive to nitric oxide stress, and that is driving the GFP expression. So there is, if there is, it's inversely correlated. So less GFP means, sorry. So in this case, you see that there is a reduced expression of GFP, which means that the bacteria are less stressed here, right? So that's exactly what is happening with our compound, that it is reducing the stress that is encountered by mycobacteria in vivo, which quite likely could be the nitric oxide stress. Now, around this time, there was also a paper that came which suggested that there is some kind of a relationship between the ornithine and other metabolite levels with autophagic pathway, and this had something to do with mycobacterial survival also. And going back to Bill's point, we also took some hints from the literature from the cancer field. And again, there were significant associations between ASS1 and autophagy. So we know, given what we also know about autophagy and mycobacteria, we also looked at the autophagic phenotype, and there was something quite surprising happened. So here we have macrophages treated with autophagy inducer taurine. You can see a significant induction of autophagy. And when you do this in the presence of our compound, there is a suppression, there is a inhibition of the autophagy. Now, autophagosome formation. To confirm this again, so we did LC3, GFP LC3 experiment where macrophages are starved, and you can see a significant induction of autophagosomes upon starvation or rapamycin treatment. Now, when we do this in the presence of this compound, you can see that this is not happening. So between starved and non-starved, there is no difference. So ASS1 is also playing a crucial role in autophagosome formation. And we also now have a compound that is actually inhibiting the formation of autophagosomes. And an independent inhibitor of ASS1, MDLA, is doing the same thing. So in vivo, what is the happening? So we went back to our mouse sections and we immunostained with autophagy marker LC3. And now we could clearly see that there are bacteria which are co-localizing with autophagosomes and there are bacteria which are not. And now we could split the bacteria into ones which are in autophagosomes and which are not. And clearly you could see a difference in the levels of stress that they are encountering. So the guys which are not co-localizing with autophagosomes are less stressed, right? So now what is happening when we add our compound is that we see that due to decreased autophagosome, there is a substantial reduction in the association of mycobacteria with autophagosomes in vivo. And correspondingly, as I told before, there is a reduced stress response that the bacteria are encountering. And here again, the data with untreated and the compound treated mice, you can see that there is, when we look at the bacteria which are either inside or outside the autophagosomes, there is a significant difference there. Now, if this is indeed true, because autophagy is also a pathway that can be nicely pharmacologically modulated, we wanted to see if we can modulate it the other way. So before that, just a quick thing. So the guidelines for use and interpretation of autophagy, they explicitly mention the challenges associated with looking at autophagy in vivo, particularly if you're using immunostaining techniques. So they say that we have to substantiate it with measuring lysosomal functions. So these are single cell suspensions from infected mice looking at different types of lysosomal function, and we can see that they are all reduced by the action of these compounds. So going back to modulating autophagy in a different way, here is a different compound that we had identified earlier, which is modulating autophagy, which is inducing the autophagy machinery. And we could nicely show that because of this, there are mycobacteria which are going inside the autophagosomes. So what will happen if we treat infected mice with this compound? So this compound is called nortriptyline, which is also an antidepressant. You can clearly see that qualitatively there is an increase in the formation of autophagosomes in vivo. And what is really important here is that the reduction that is happening in the stress levels of mycobacteria is getting reverted now. So by tweaking the autophagy axis, we are able to alter the physiological status of mycobacteria. And of course, you can see this also in the mycobacteria which are in or out of autophagosomes. So what I showed you today is that we did a systematic screening for pro-infective phenotypes. We identified and validated the target, and we can establish a relationship between the subcellular localization of mycobacteria in vivo and distinct physiological states, and something that we can alter by pharmacological modulation, right? So I think I will stop there. Thanks to David Russell for the reporter plasmids, and thank you all.
Thanks, Dr. Varda. So our next speaker is Dr. Ved Prakash Dwivedi. Dr. Ved Dwivedi works as a group leader of immunology group at ICGV since 2019. His primary focus has been better understand the TB immunopathology with primary emphasis on host immune responses to TB. And he had provided first evidence of enhanced protection by Th17 cells in the lung against TB. Also, he had developed a novel host-directed therapy for TB by suppressing Th2 cells as well as Treg with therapeutic compounds. And he's also most interesting is recently his group has generated an immunology Immunogenic complex against TB pathogen consists of promiscuous T cell epitopes as well as TLR components. And of course, this involved intranasal delivery of these molecules. And I think, interestingly, this molecule has been patented and presently in national phases of various Southeast Asian and African countries. With this established research work and new insights, we welcome you for today's talk.
Thank you, Professor Balaji, for a nice introduction. So first of all, I would like to thank all the organizing committee member for giving me an opportunity to present my work here. Today, I'll be discussing about some novel vaccination and immunotherapeutic approaches to combat tuberculosis. I will also discuss how the immune modulation can particularly strengthen the long-lasting protective memory responses. As we all know, understanding the host protective immunity against TB is really very complex. It is still unclear which antigen or which immune cells provide protective immunity against this deadly disease. Every day we are increasing our understanding to unscramble the complexity of this disease as well as associated immune responses. So I can skip this slide, but for just freshen up the memory, I'll go quickly. So tuberculosis is the pandemic disease. As per WHO, one fourth of the global population is latently infected with Mycobacterium tuberculosis. That is the causative agent of tuberculosis and is responsible for millions of deaths each year. It has spread worldwide, but more prevalent in Southern and Central Africa, South Asia, and Latin America. Mycobacterium tuberculosis mainly infects through respiratory tract, where the alveolar macrophages engulf airborne bacilli. Bacteria replicates inside the macrophages. Macrophages secrete some cytokine and chemokine in response to bacteria to activate the lymphocyte. Once they get activated, they migrate to the site of infection and form granuloma. The main function of the granuloma is to prevent the spreading of bacilli from lung to other organ, as well as concentrate the immune response at the site of infection. If the immune response is stronger, then bacteria will be retained, granuloma will be reduced, and characteristic will be asymptomatic. However, if there is weak immunity, then bacteria will replicate, granuloma will caseate, rupture, and spill the bacilli into the airway to infect others through coughing and sneezing. So depending upon the cytokinic environment, T helper cell gets differentiated into Th1, Th2, Th17, and T regulatory cells. So previously, we and others have reported that Th1 and Th17 cells provide protection against this deadly disease. However, Th2 and Treg cell assist in disease progression. So thus, simultaneous redirection of the immune responses are required in such a way that one has to Th1 along with Th17 simultaneously suppress Th2 and Treg cells for the better therapy of tuberculosis. So TB is the present on the earth since ages, but it is still considered as global pandemic. The problem lies both in its prevention, wherein we have only one vaccine, that is BCG, which is very effective in case of disseminated and meningeal TB, but it has questionable efficacy against adult pulmonary TB. On the other hand, it's a treatment which consists of multiple expensive antibiotics with no emphasis whatsoever on the host. So, however, there are multiple T cell subsets which are known to play a crucial role in containing MTB infection, but still the exact correlates of host protective immunity against this deadly disease is not known. So our lab is primarily focusing on the development of novel vaccine candidate as well as novel immunotherapeutic strategy by utilizing various host factor, bacterial factor, and epigenetic modulation. So to develop a vaccine candidate, first we should understand why BCG doesn't work. So for this, we designed a very simple experiment wherein C57 Black 6 mice were infected with virulent strain S37RV and vaccine strain with BCG. And at different time point, we took out the lung to look at the bacterial burden as well as immune responses. And interestingly, we observed that initially both strain replicated in similar manner, but at later time point, growth of BCG got gradually diminished. So it might be the reason that adaptive immune response plays an important role for the clearance of BCG at the later time point. Then we looked at the immune response, and we observed that both strain induces comparable amount of IL-15 gamma producing Th1 cells. However, BCG was unable to induce IL-17 producing Th17 cells. So what is the reason behind this? Why BCG is unable to induce significant amount of IL-17 producing Th17 cells? So as compared to virulent strain S37RV, BCG possesses multiple deletion mutations, and these mutations are known as region of differences and RDs. And among these RDs, RD1 is the most prominent, dominant, and important one for providing virulency to Mycobacterium tuberculosis. Then we asked whether RD1 has anything to do with TS17 differentiation. For this, we deleted RD1 from S37RV and put back RD1 into BCG. And then we looked at the bacterial burden and immune response after infecting mice with these strain. And interestingly, we observed that RD1 deleted S37RV was behaving like BCG. However, RD1 knock-in BCG was behaving like S37RV in terms of bacterial burden as well as immune responses. Then we looked at the IL-17 and TS17 differentiating cytokine in the ball fluid of infected animal, and we observed that RD1 deleted S37RV and BCG were unable to induce TS17 differentiating cytokine, that is IL-6 and TGF-beta. So now we know that RD1 has something to do with the TS17 differentiation. So within RD1, there are multiple proteins, and ESAT6 is the most prominent and has been extensively studied. So we thought whether ESAT6 has anything to do with the TS17 differentiation. So for this, we took out the femur bone marrow of the mice, differentiated into dendritic cells, treated with ESAT6, and then co-cultured with CD4 T cells derived from OT2 transgenic mice. And then we looked at the cytokine level in the culture supernatant, and we observed that ESAT6 was able to induce TS17 differentiating cytokine in TLR2 and MYD88 dependent manner. Then we looked at the molecular mechanism of this phenomena of the induction of the TS17 differentiating cytokine, or you can say differentiation of TS17. So long back in 2017, Baltimore group has first time discovered that microRNA regulates innate immunity. And so taking cue from that study, we screened many microRNA, and interestingly, we observed that ESAT6 significantly induces VIR146A and allows the induction of IL-6, which is again required for the differentiation of TS17. So with this data, we proved that S37RV even can provide better protective immune response than BCG. But for obvious reason, we cannot use S37RV as a vaccine candidate. So therefore, we thought to prepare a molecule that can be immunogenic without causing any pathogenicity. So for this, we screened many MTB peptide for this, for their efficacy to induce T cell activation as well as protective cytokine responses. And then we combined together with TLR ligand and adsorbed these molecules on the surface of liposome to facilitate their entry into the lung of mice through intranasal injection. And we named this molecule as PTL, and we just patented this, and this is now in the national phases of various African and Southeast Asian countries. Then we asked whether this PTL molecule can enhance the vaccine potential of BCG. For this, we designed a vaccine experiment wherein BCG immunized mice were subsequently boosted with this PTL. And then after a month of rest, these mice were infected with MTB through low-dose aerosol infection model. And then we took out the different organ to look at the immune response as well as bacterial burden. And interestingly, we observed that PTL co-immunization significantly reduced the bacterial burden, and you can say significantly enhanced the BCG-induced antitubercular immunity, as evidenced by reduction in bacterial burden by more than 2 log. And we also observed that PTL co-immunization significantly induced polyfunctional cytokine responses and memory responses, which further led to the reduction of rate of TB reactivation. So now we proved that intranasal immunization with PTL complex enhances the BCG vaccine efficacy in murine model of TB. This study just got published in JCI Insight. Then we also observed an increased population of double positive T cell in the lung of PTL co-immunized mice. And these double positive T cells are very novel subset of T cells. And they are initially thought to be produced in the thymus and are involved in the generation of single positive T cell, which eventually migrate to the periphery and provide protection against different disease conditions. So there are various reports showing the presence of these double positive T cell in the periphery in a number of virus infection as well as cancer by technique flow cytometry as well as confocal microscopy. So when we analyzed in detail, we observed that these double positive T cell produced increased polyfunctional cytokine responses and memory responses. And upon adoptive transfer in rag knockout mice, we observed that these double positive T cells displayed increased anti-tubercular immunity as evidenced by reduction in bacterial burden than their single positive T cell counterparts. So in order to look at the mechanism of the infiltration of these double positive T cell in the lung of MTB infected mice, we performed proteomics and transcriptomic studies to understand the differences between these T cell subsets, as well as proteins and pathways involved in the generation and maintenance of these double positive T cell during MTB infection. So fortunately, Professor Kaushal is here. So I discussed this data in detail, and now he's agreed to provide some NHP sample to for the further validation of this data. So moving on to the next term of the lab, where we are involved in the formulation of the novel immunotherapy. As we all know that ATT having multiple antibiotics, it is very lengthy, and due to length of treatment, once the patient start feeling better, they withdraw from the therapy, and that is the main cause of emergence of drug-resistant variants. It also causes various side effects like hepatotoxicity, and most importantly, it causes immune impairment response, thus making host vulnerable to reactivation, TB reactivation and reinfection. And we think that these shortcomings and side effects can be overcome by addition of certain immunomodulator in the treatment regimen. So for this, we screen many immunomodulatory compound, and here I would like to show berberine as an example, which is an alkaloid having a very weak antibiotic property. People also have shown berberine as an efflux pump inhibitor. It has anti-diabetic nature and can reduce the hyperlipidemia, and there are many reports showing its anti-cancerous property. So we thought whether berberine has any immunomodulatory activity in case of MTB infection. For this, we took TSP1 cells, that is TSP1 human monocyte, and peritoneal macrophages, and infected with MTB S37RV and treated with berberine. And interestingly, we observed that berberine reduces the bacterial burden in these macrophages. And most importantly, we observed synergistic effect when we added berberine along with INH. And therefore, we can say that berberine can potentiate the antimicrobacterial potential of frontline anti-TB drug isoniazid. Then we translated this research in vivo model of tuberculosis, wherein black 6 mice were infected with MTB through low-dose aerosol infection model. And after 15 to 20 days, we started treating these mice with berberine, isoniazid, and combination therapy. And at different time point, we took out the lung and spleen to look at the bacterial burden and immune responses. And interestingly, we observed that berberine treatment significantly reduces the bacterial burden in lung and spleen of the mice, not in case of only drug susceptible strain, but also in case of drug resistant strain. And most importantly, we observed that berberine treatment, when we observed the additive effect when we used combination therapy of berberine and isoniazid. Then we looked at the immune response after berberine treatment, and we observed that berberine treatment significantly induces macrophages activation, T cell activation, B cell responses, and significantly heightened the stem cell memory response, central memory response, and tissue resident memory response. These memory responses are required for the long-term protection against tuberculosis, or you can say any disease, any infectious disease. Then we looked at the mechanism of the heightened memory response. As we all know, naive cell upon stimulation gives rise to stem cell memory, and that further gives rise to effector memory, central memory, and tissue resident memory responses. That memory responses are required for the protection at the tissue specific site. So for this, we took human PBMC from 4 PPD positive individuals and treated with complete soluble antigen of Mycobacterium tuberculosis and berberine. And interestingly, we observed that berberine drives the differentiation of naive T cells to effector memory and tissue resident memory T cells, which are again required for the long-term protection against tuberculosis. So in order to find out the mechanism of the increased resident memory T cell, we performed the proteomic studies, wherein we observed that berberine treatment significantly upregulated the proteome landscape of CD4 positive T cell, resulting in the upregulation of diverse signaling pathways which are required for the protection against tuberculosis. And interestingly, NOS signaling and glycolysis pathways were upregulated, which are required for the generation and maintenance of these memory responses, particularly effector and resident memory T cell responses. So ironically, NOS signaling activates PTEN molecule, which further inhibits AKT and which activates FOXO1 by dephosphorylating it, and then which is and also BLIMP1, which is required for the transcription of TRM specific gene. Then we further validated by doing RT-PCR studies, where we observed that berberine treatment significantly induces that gene which is required for the transcription of these resident memory T cells, that is FOXO1, NOS3, BLIMP1, etc. We also observed an increased glycolysis, which is after berberine treatment, which is required for the effective function of T cell by performing lactate assay in the culture supernatant. And these data were further validated with the Bioplex data, where we showed that berberine treatment significantly upregulated almost all the pro-inflammatory cytokine, which is required for the protection against tuberculosis. So now we prove that berberine induces long-term protective immunity by generating effector memory and tissue resident memory T cells. So, and previously we and others have reported that BCG immunized animal primarily develop effector memory T cell and fails to induce central memory T cell, which is the major precursor of memory cells, which are required for the long-term protection against tuberculosis. So it might be one of the reasons for the failure of BCG. So we thought, then we asked whether berberine can enhance the vaccine potential of BCG. For this, we performed a vaccine experiment wherein BCG immunized mice were subsequently treated with berberine. And after a month of rest, we infected these animals with MTB through low-dose aerosol infection model. And then we took out the lung to look at the bacterial burden and immune responses. And interestingly, we observed that berberine treatment significantly reduces the bacterial burden in BCG immunized animal, and also it enriches the resident memory T cell in the BCG immunized animal. So now we have proved that BCG has both immunoprophylactic as well as immunotherapeutic activity. So now in the previous slide, I discussed that ATT makes host more vulnerable to TB reactivation and reinfection. So we thought, what would be the effect of this berberine treatment on the TB reactivation and reinfection? For this, we designed a reactivation experiment, and interestingly, we observed that berberine reduces the rate of TB reactivation and also reduces the bacterial burden in case of MTB reinfection. So now on this note, I would like to conclude this study by saying that berberine treatment significantly induces the expression of PTEN and FOXO1 molecule that can induce the immunological memory against tuberculosis. So this study just got published in PLOS Pathogen. And another in similar line, we also published a report where we showed that vidofludimus, that is active ingredient of Vidania somnifera, protects against primary and recurrent TB by modulating mycobacteria specific host immune responses. So on this note, I would like to thank my lab members, my collaborators, ICGB core facility, and of course funding partner. Without their help, I could not be able to generate this data. And this data was mostly done by one of my hardworking and brilliant PhD student, Isha Pahuja, with the help of these two collaborators, Dr. Ashima and Dr. Jaswinder Singh from ILBS. Jaswinder Singh provided all the PBMCs and performed the proteomic studies for this study. Thank you.
So first of all, on behalf of Professor Deepak Kaushal as well as myself, this is the only opportunity for us. Thank the organizers for the generosity to invite us here, and we really enjoyed all the talks as well as being here on this. And also we thank all the speakers today, who is really all of them very illuminating. And for us, it's a learning curve, or at least for myself, to learn each of you. So thank you all. So now the podium is open for questions. sorry.
So this is for, oh, sorry. This is for Ved. So with respect to the ESAT-6 story, I was just wondering, there are a lot of clinical isolates which basically doesn't make ESAT-6. So there are a lot of clinical isolates, they don't make ESAT-6. So would that mean there would be some difference with respect to the TH17 responses?
Yeah,
That is
it might be because that is why BCG is unable to induce TS17.
I'm talking about the clinical isolates.
That we can study, and that we can, without doing study, we cannot say, because here it was one of the factor, but there are other factors which can be responsible.
But the reason I'm saying that is because these are clinical isolates which basically have established infection and caused pathogenesis. So I was wondering what is happening with that. That's one thing. And the second one is with respect to your RD1 in BCG. Was this the entire RD1 or is it just the ESAT-6?
No, this plasmid was given by Professor Stuart Cole, that was named as 2F9. Yeah.
Right. Yeah. Thank you.
I'm just curious to know, like, how does BCG evade this immune mechanism which is initiated by berberine? Because at what time you are planning to give berberine post vaccination?
So yeah, we gave immunized mice, and then after 2 weeks of immunization, we started treating with berberine with 2 doses.
So by that time, you expect it to induce a memory response, or
no? Then after treatment, we kept these mice for rest for another month, and then we infected these mice with MTB, and then after 60 days we profiled. So it was around, from immunization to immune profiling, it was around 120 days. So generally we expect a memory response after 45 days to.
Yes, I mean, berberine is not,
in mice.
Berberine is not going to affect the viability of BCG.
Viability of BCG?
Yes. Because if it is inducing antimicrobial immune response.
No. Actually, it doesn't have any antimicrobial activity direct.
No,
like
anyways, even through immune mechanisms I'm talking about.
So it is just inducing immune response in the cells.
For mycobacterial clearance.
Yeah, mycobacterial.
But BCG is also mycobacteria.
Yes, but we are giving again 15 days interval.
Okay.
After
to set up the immune
yeah,
response.
Yeah. That time we expect that immune cells are there.
Okay, thank you.
Professor Dheeraj, it was a nice talk. So my question is basically, you are looking at about the SNRNPs that interacts with numerous mycobacterial proteins. So do you look into the classes of the different mycobacterial proteins? Like there are different proteins like soluble or membrane bound, membrane associated mycobacterial proteins.
Yeah, so all the proteins that we started with for the screening work was all the established secreted proteins from mycobacteria. So these are all secretory proteins, and quite a few interactors were found, but of course everyone was not followed. Some of them were followed. Not much known about those proteins in terms of what they do in mycobacteria. So some of the functions that we are revealing as kind of novel function, which are in the host, something they should do also in bacteria. We don't know what they do in bacteria. Yeah.
Just like said, there are different proteins of the type 7 secretion pathway. So does your group also looks at those classes of proteins, that whether they might interact with SNRNPs or not?
A part of them is, so, the secretome is characterized by both, whether they are type 7 secretory or, they have TAT kind of or SEC kind of secretion system. So the secretome comprises of all those proteins, right? It's for not selective that you took only one and then talk about it. So it's a combination of all. And I think what I remember is that you got interactors from all independent classes of secretory pathway that are present in bacteria. Yeah. Thank you, sir.
Yeah. Oh, hello. I have a question to Sheetal. Excellent talk. Just out of curiosity, I'm asking, there is this DGAT1 enzyme target. So why was it chosen as, because if you inhibit DGAT1, you'll have DAG levels to increase, and that may have also, because it has a messenger role also. So I was just curious to know why was it a good candidate?
So this was an academic question. What is the role of lipid accumulation in macrophages? Simple. We were not looking for host-directed therapies. Yeah.
So
So
it
we
was not a
very well
target
knew,
per
we
se.
very well knew that targeting DGAT1 as a host-directed therapy was not an option. But to begin with, do these macrophages that have lipid accumulation play any role? And the only way to address that is not by looking at other correlates, but by looking at triglyceride accumulation. So that is why we chose this.
Thank you. And if I could ask a simple question to Dheeraj, which the cohort study that you mentioned where you showed that the TL17 levels were very high, IL-17. So as Dr. Dwivedi was suggesting about adjunct therapy of immunomodulator, so do you think it should be given to the patients? I mean, it should be checked before giving treatment?
I could see in Ved's presentation that there are a lot of things which we find independently in terms of what's happening upon TB infection in terms of IL-17 and, it's certainly, we can discuss more. This data, I think I haven't discussed with Ved also earlier because it's something which is very new, it's unpublished. We look at mostly the innate immune part. So I haven't looked into how T cell responses get affected under all these conditions. So there are a lot of things which can be, evolved. So I think your question was what Ved's observation is in terms of IL-17. Is that what?
My question is that cohort study where you showed that about 133 patients who were successfully treated and those who were not successfully treated. So in those patients, the IL-17 level was found to be high.
Yes.
So, I mean, that means that they already had the levels high. So that's
They
why
have
perhaps they
high-
did not respond to
They
the
have
treatment.
very high IL-17 at a time of recruitment. So when
Yeah.
those patients were recruited,
Yeah.
they had very high IL-17.
Right.
And those were the ones which failed treatment.
Exactly.
Yeah.
So that's what I'm saying, that since it was known that they had low levels at the time of recruitment and then they did not respond to the therapy. So I was wondering that if they already had elevated levels of IL-17, what adjunct therapy can be given?
Sorry, I didn't get the question. Can you be a little loud? I didn't get the question.
I'm saying that, okay, I was just curious to see that data from the cohort study where you had shown about 133 patients who got treated well and successfully treated, and about 60 plus were not, they did not respond to the treatment. And at the time of recruitment, only it was seen that their IL-17 levels were high. And perhaps there's a link between the high level IL-17 and not responding to the treatment. Did I get it right?
So, I'll tell you, the baseline data is for entire panel of cytokines and CBC, all the data is there. I was very selective in what I was talking about because that correlates better with the mice data that we had. So the observation is that at the time of recruitment, before treatment started, those patients which had very high IL-17 levels were the ones which failed treatment, right? And these failed treatment were unique. I mean, interesting because they didn't have drug, because treatment failure can be because of drug resistance also. So those were excluded out so that you don't, combine drug resistance versus immune, compromised state and things like that. So that's.
Unfortunately, this would have to be the last word.
Hello, sir. I have one question. So I have the question for Sheetal ma'am. So ma'am, what I understood was foamy macrophages are part of granuloma and microenvironment. So why is it that few of these macrophages turn foamy and not all of these? So is it like something TB wants hosts to make so that it can easily scavenge lipids from it? Like, what are your views on it?
So actually, I'd like to make a correction to that. It's not that only some macrophages are. So if you look at the caseous granuloma, you have a central core of necrosis, and that's just dead cells, a lot of neutrophils in that area. You come out of that, there are more neutrophils, and then you have this zone of foamy macrophages. So essentially, that entire rim contains foamy macrophages. Having said that, it's not that all macrophages in a granuloma are foamy macrophages. So sorry, what was your question? That is bacteria, sorry, say that again.
So is it like bacteria wants host to make this foamy macrophages so that it can easily scavenge lipids from it? Or is it like a
So that's a very good, sexy idea, but not proven, I think, because like our data shows, even if you inhibit triglyceride synthesis in a macrophage, that's not sufficient to inhibit growth of bacteria. Right? So I think it's not simplistic, and that's why we're still working on factors that regulate lipid mobilization in cells.
Thank you, Sheetal. And I'd like to once again thank all the speakers of the host pathogen interactions session.
Thank you, speakers and chairs, for illuminating the importance of host-pathogen interaction. I kindly request the session chairs to felicitate the session speakers. Professor Balaji to Dr. Dheeraj Kumar. Professor Balaji to Dr. Sheetal Gandotra. Dr. Deepak Kaushal to Dr. Sundara Murthy. Dr. Deepak Kaushal to Dr. Ved Prakash Dwivedi. I now request Dr. Ramandeep Singh to felicitate the session chairs. Professor Balaji. And Dr. Deepak Kaushal. Thank you, sir. Today and yesterday, we witnessed several posters, and it's time for the Best Poster Awards. There'll be 9 consolation prizes and There'll be 9 consolation prizes and 3 independent prizes.
I request Dr. Anil Tyagi, sir, come on to the dais. Bill, Audrey, Vinay, Balaji, onto the stage. Thank you. So you may be seated. You may be seated. You may be seated in the dais. You may be seated there. Okay, go ahead from the bottom. So be careful because you might post it and this is Balaji. Dr. Balaji to basically give away the consolation prize for poster this number.
I request Dr. Balaji to kindly award. So I'll be going from the consolation prizes to the main independent prizes. So the first consolation prize is poster E2.04, Kirti Medreta. Kirti, if you're here or if someone is here to receive the award on her behalf.
Just make sure that you don't spend the entire money.
Next consolation award is poster E2.03 Archana Singh. The next poster is poster 1.53 Sidra Khan. Balaji, yeah. Thank you, sir. Moving on, I request Dr. Vinay to kindly give away the consolation prize to poster 1.25, Manisha Priya. Sir, I'll also request you to hand over the prize to poster 1.11, Ankita Arora. Poster 1.11, Ankita Arora. Poster 1.10, Amitava Sinha. Amitava Sinha. Amitava, okay. He's not here. Yeah. Thank you, sir. I now request Dr. Adri to kindly give away the consolation prize to poster 1.8, Pratibha Sharma. Poster 1.8, Pratibha Sharma. Poster 1.5, Linus Augustine. And finally, the last consolation prize goes to poster 2.38, Neeti Singh. Thank you, sir. Now I invite Dr. Bill to give away the third prize, poster 1.59, Taruna Sharma. The second prize goes to poster 1.13, Bhupinder Singh.
Come this side, come this side.
Thank you, sir. Now the first prize will be given to two candidates. I invite Dr. Tyagi to kindly give away the first prize to poster 1.12, Arun Sharma. And poster 1.31, Munmun Bhasin. Thank you, sir. Thank you, everyone.
Thank you.
With this, we conclude our three-day symposium on Towards NTB, Achievements, Challenges, and Future Directions. I request all poster attendees to kindly collect your posters from the display boards. Now I request Dr. Amit Kumar Pandey to deliver the vote of thanks.
So, wow, that was it was an interesting meeting, and I thoroughly loved it, and I hope everybody enjoyed the meeting, both professionally and, of course, we tried our best to take care of your logistics and comfort. So I would not say that everything good has to end, but I must say that we have to take a pause here, and the show must go on. So on behalf of the organizing committee, I would like to extend my heartfelt thanks to each and every one of you who attended and participated in this symposium. It was an honor for us to host such a diverse and accomplished group of speakers and delegates who shared their expertise and experiences with us throughout the symposium. Their contributions and invaluable insights for their contributions in invaluable insight, we are grateful, and we are also grateful for their time and effort in making this event a success. A big thank you to all the keynote speakers, session chairs, national and international speakers for sharing their valuable insights. Your contribution was greatly appreciated, and we are grateful for the time and effort you took to be with us today or for the past 3 days. We express our heartfelt gratitude to all the senior mentors, including Professor Talwar and Dr. Swaminathan, for gracing this occasion. Your presence has been has motivated and inspired all the participants. We would also like to express our gratitude to Dr. Rajesh Gokhale, Secretary DBT, for his constant support and valuable insight, and for accepting the invitation to be one of the keynote speakers despite his busy schedule and engagement with the G20 meeting. As always, we would like to thank the Department of Biotechnology, Dr. Jyoti Lagani, Dr. Vashne Singh, Dr. Alka Sharma, and other senior advisors for their constant guidance and support. We would like to express our gratitude to Professor Pramod Gar, the Executive Director of THSTI, for conceptualizing this meeting and for all the motivation and unconditional support extended to the organizing committee. It would have been impossible to organize the meeting without the support of all the faculty and scientists of THSTI, Mr. Santo and Mr. Rajkumar, the administrative officers, volunteers, administrative staffs, engineering, IT, security, and housekeeping staffs who worked tirelessly behind the scene to ensure that everything went smoothly and efficiently. I take this opportunity to thank all the chairs and members of the different committees that were constituted for smooth conduct of this event. We are very grateful to all the event sponsors. Without their financial support, this symposium would not have been possible. We are grateful for their contribution and support. Lastly, we would like to thank you all once again for your active participation and engagement throughout the symposium. Your enthusiasm and energy added value to the event, and it made it a memorable experience for all of us. Thank you all for coming, and we hope to see you again in the near future. Very good night to you. I hope we meet sometime soon and have a good TV talk. Thank you. Thank you very much.
Let us now end this